Doctor Peñalosa. Good morning, Preston authorities, ladies and gentlemen, good morning. We begin the workshop Flowing Through the Net, top-tier cellular analysis using next-generation cytometry. We welcome and especially greet the authorities, Dr. Marian Krause, Acting Rector of the Pontificia Universidad Católica de Chile, Mr. Francisco Chaguán, Senator of the Republic, Dr. María Fernanda Pérez, Dean of the School of Biological Sciences of the Pontificia Universidad Católica de Chile, Dr. Felipe Heuser, Dean of the School of Medicine of the Pontificia Universidad Católica de Chile, Dr. Sergio Lavanderos, President of the Academia Chilena de Ciencias, Dr. Ignacio Negón, FOSI's President, Director de Research, INSERM, Emerit Nantes, France, who will be joining us online, Dr. Robert Banderas, Vice President of BD Biosciences, Dr. Alergis Kalergis, Director of the Millennium Institute on Immunology and Immunotherapy. We also extend a warm greeting to the national and international researchers who will join us today. We thank the organizing entities for this event, the Millennium Institute on Immunology and Immunotherapy, FOSIS, and BD Biosciences. We begin this session by inviting Dr. Marian Krause, Acting Rector of the Pontificia Universidad Católica de Chile, to give her welcome address.
So, good morning to everyone on this rainy day. Yes, I welcome especially the authorities. I'm not going to repeat all the names because we know who is here. I'm very happy to have you here. So we'll start immediately with some words. It is a true pleasure to welcome you on this workshop called Flowing Through the Net, the top-tier cellular analysis using next-generation cytometry. Today, we gather for an event that brings together leading researchers, academics, students, and professionals, all united by shared commitment to advancing science through innovation, collaboration, and the pursuit of new knowledge. I would like to sincerely thank each of you for being part of this important occasion and for contributing your expertise and vision to this vibrant exchange. And I see this full room, so it will be a vibrant exchange. Flow cytometry has historically been a cornerstone technique in the scientific development of the School of Biological Sciences at the Pontificia Universidad Católica de Chile. Over the years, it has empowered us to drive forward research in key areas such as immunology, cell biology, environmental microbiology, and preclinical studies. Our state-of-the-art cytometry platform has not only fostered scientific excellence within our university, but has also contributed to projects of great public relevance. Among these are the clinical studies that supported the emergency approval of COVID-19 vaccines and the development of the UC vaccine against respiratory syncytial, you have to tell me how to pronounce that, virus. Yes, clear examples of how cutting-edge technology, when combined with dedication and scientific rigor, can have profound impact on society. Today's workshop has two main objectives. Firstly, it seeks to highlight and further strengthen the role of our flow cytometry platform as a hub for scientific excellence and innovation. Secondly, it aims to forge and deepen connections between our institution and leading academic centers worldwide and with the scientific industry represented here by today's distinguished participants. I would like to extend special recognition to Dr. Hernan Peñalosa and Dr. Alexis Kalergis from our university, and to Mr. Robert Balderas, Vice President of BD Biosciences. for their outstanding leadership and commitment in making this event possible. Their efforts reflect the transformative power of collaboration across disciplines and sectors. The presence of Dr. Valderas with us today also underscores the critical importance of building effective, lasting bridges between academia and the global scientific industry. Such connections are vital for opening new paths of research, technological innovation, and scientific advancement that responds to the challenges of today and anticipate the needs of tomorrow. This workshop is not merely a showcase of our current capacities. It is, above all, a forward-looking space, an opportunity to envision and build future collaborations, foster new research networks, and position us as a key node in the international scientific community. At the Pontificia Universidad Católica de Chile, we reaffirm our deep commitment to a science that is at the service of society, a science with a real public impact, grounded in robust international networks and guided by a clear and ambitious vision of the future. We believe that science is fundamentally about community and dialogue. It is about openness to new ideas, to new technologies, and to new ways of understanding the world and life itself. May this workshop inspire us to continue exploring, researching, collaborating, and dreaming big. I wish you all a highly productive, inspiring, and rewarding workshop. Welcome and much success.
We sincerely thank Dr. Marianne Krause for her words. We now welcome the Dean of the School of Biological Sciences, Dr. María Fernanda Pérez.
Buenos días a todos. Voy a hacerlo en español porque no recibí la instrucción en el momento adecuado y ya no alcanzo a hacer un cambio en el discurso. Bueno, primero que nada, muchísimas gracias por la presencia, senadores, un placer que esté con nosotros, nuestra nueva prorectora, Marianne Kraus, nuestro presidente de la Academia de Ciencias, por supuesto, los académicos, Alexis Calergi, Hernán Peñalosa, Susan Bueno, Pablo. Y también, por supuesto, a nuestro decano de la Facultad de Medicina, con el que hemos estado haciendo muchísimas colaboraciones para poder tener este tipo de instancias. Como ustedes saben, uno de los objetivos principales de la universidad es incentivar el desarrollo de investigación de impacto internacional, pero que también tenga relevancia pública. Y ahí la relevancia de que usted esté aquí, senador. Y eso significa abordar desafíos globales que son cada vez más complejos y que requieren interdisciplina y requieren un equipamiento de altísima complejidad. Y para instalar esos equipamientos, para que esos equipamientos se usen con toda la potencia que eso significa, se requiere liderazgos académicos importantes y se requiere articular esfuerzos relevantes, esfuerzos que vienen de las facultades, de los académicos, de toda la universidad y, por supuesto, del Estado y también del sector productivo. Así que agradezco enormemente también la presencia del vicepresidente Robert Valderas, que está aquí presente. Este es una iniciativa particularmente interesante porque no solamente involucra dar a conocer la plataforma de citometría, sino todos los usos potenciales. Hoy día, y aprovecho nuevamente la presencia, no solamente es equipar una universidad, sino que ese equipamiento se utilice en un país que ese esfuerzo es extraordinariamente relevante. De aquí una actividad que involucra difundir el uso de una plataforma tan sofisticada como puede ser la citometría de flujo, es hacia dónde debemos ir. Y eso involucra que la puedan utilizar investigadores de nuestra facultad, de todas las facultades de la universidad, pero también investigadores de todos los de Chile y de una región que no requiere o que no tiene necesariamente equipamiento. La citometría de flujo, bien lo explicó Marian, mejor que yo probablemente, permite caracterizar funcionalmente la célula y eso tiene una serie de impactos y una serie de potenciales extraordinariamente relevantes, no solo en áreas de la biomedicina, sino también, por ejemplo, en el área de la ecología. Ya se destacó el uso que tuvo para lo que significó los estudios clínicos en el momento de la pandemia del COVID-19, lo que puede significar también y lo que está significando para el desarrollo de vacunas contra el virus sincicial. Pero tiene impactos también que son muy relevantes en el ámbito de la ecología, con el monitoreo de las microalgas y particularmente con el monitoreo de la marea roja, que conllevó un movimiento social muy importante en la isla de Chiloé no hace tanto tiempo. Así que la cantidad de usos potenciales de una plataforma de esta dimensión como citometría de flujo son muy relevantes al desafío país, al desafío de las universidades y por supuesto al desafío de la región. Así que nuevamente yo agradezco al doctor Peñalosa, al doctor Alexis Calergi por habernos invitado y por haber hecho este esfuerzo de tener a representantes de las distintas entidades, tanto del sector público, del sector productivo, del sector académico, y nada puede ser tiene significado si además no hay un grupo de estudiantes que se estén formando en estas tecnologías. Así que nuevamente muchas gracias al equipo por esta actividad.
We thank Dean Perez for her words. Next, we welcome the Dean of the School of Medicine, Dr. Felipe Heuser.
Buenos días a todos y todas. Voy a partir saludando muy brevemente a las autoridades, particularmente a nuestra prorectora, Marian Kraus, al honorable senador Chaguán, al doctor Sergio Lavanderos, al profesor Valdera y particularmente a los organizadores de este simposio, ¿no? Alexis Calergi, Hernán Peñalosa, pero a todos los académicos, académicas, a todos los participantes de este simposio, desearles una muy cálida bienvenida. Welcome to this workshop. From the Faculty of Medicine, I would like to express my joy and satisfaction with the organization of this workshop, primarily for two reasons. First, because of the central theme of the workshop, flow cytometry, a technique pioneered over 50 years ago, actually 55 years ago, that allows the study of cells and particles in suspension through the simultaneous detection of multiple physical and chemical characteristics. This technique, thanks to the technology, has developed over the years to include versions that have expanded its potential, such as spectral flow cytometry. Has made an extraordinary contribution to basic research, translational research, and clinical research, significantly contributing to the biomedical research in general, but particularly to the clinical diagnosis and to the development of advanced therapies. There are numerous investigations carried out using this technique that have had and will continue to have for sure a major impact on the advancement of medicine and in particular on the health of the population. Secondly, this workshop brings together experts, researchers, academics, and people from different universities in the country and around the world, from our neighbors in Argentina to China, in a spirit of collaboration, sharing their knowledge and experience. It is clear how collaboration leads us to better science, more impactful results, and ultimately, more importantly, to our society and to the health of our population. Finally, I would like to thank all the presenters at this workshop for their generous participation, and especially I would like to thank the organizer at the Millennium Institute of Immunology and Immunotherapy. I wish you all a very productive day and that we emerge with new motivations and new ideas to continue contributing to the people's health through basic translational and clinical research. Thanks a lot.
We thank Dean Heuser for his contribution. We invite Dr. Sergio Lavanderos, President of the Academia Chilena de Ciencias.
Okay, good morning, everyone. It's a real pleasure to be here today. But I switched, originally was in English, but I prefer to switch to Spanish to keep a balance. Muy buenos días a todos. Realmente es un honor estar acá, saludar a la nueva prorectora, al senador Chaguán, al representante de Beckton Dickinson, el decano y otras autoridades, y especialmente a los organizadores. Está claro que estamos viviendo tiempos muy, muy complejos. Conversábamos antes del inicio de esta presentación, cambios geopolíticos de proporciones, duda acerca del rol de la ciencia en la sociedad. Con gran preocupación hemos visto lo que está ocurriendo hoy día en Estados Unidos y he tenido la oportunidad recientemente de asistir al meeting, al Global Science Forum 52, organizado por la OECD, en que primero creo que es un privilegio que Chile esté en esta organización. Viendo el vaso medio lleno, aquí hay un vaso, así que lo voy a colocar para que se vea que efectivamente hay un vaso. Viendo el vaso medio lleno, efectivamente un orgullo que Chile esté en esa instancia. Eso es lo positivo. Lamentablemente me tocó, o sea, lo positivo, representé a Chile en esta instancia como asesor científico y pude ver lo que hacen otros países. Me llamó poderosamente la atención de que la organización de la OCDE cambia de gobierno y cambia todo. De hecho, dentro de este gobierno han habido varios cambios internos. Me tocó estar sentado al lado de Canadá, al lado de Corea, y ver cómo ellos toman este desafío como una mirada de Estado. Pero lo que me hizo recordar esto, y un tema que se trató, cuáles son las prioridades que tiene la OECD para la investigación. Varias, primeras, algunas totalmente insospechadas para mí, como es la computación cuántica. Pensé que todavía era algo medio etéreo. Es una prioridad absoluta, además de la inteligencia artificial. Pero uno de los temas que tratamos profundamente fueron de las plataformas tecnológicas. Cómo hoy en día los avances que tenemos en biología celular, en biomedicina, cómo ellos hoy día se pueden ser enfrentados. Porque hoy día sabemos que tenemos una tecnología nueva, desafiante, pero que dura muy poco. ¿Cómo somos capaces de ir actualizando nuestro conocimiento? Y precisamente el tema que se habló es cómo las universidades, los países, son capaces de enfrentar esto. Por ejemplo, un tema que salió fue el de la inteligencia artificial, en que si muchos países, universidades, empresas no se unen, no van a poder competir. Y hablamos precisamente de avances como los que estamos presenciando hoy en día, de todo lo que es la transcriptómica. Nosotros el proyecto más importante del área biológica y medicina es lo que se conoce como el Human Cell Atlas. Es lo que nosotros vamos a conocer de todo nuestro organismo, célula por célula, lo que hace. Por ejemplo, el cerebro lo estamos desarmando, célula por célula, conociendo qué es lo que hace en condiciones normales y cuando se enferma. Y por lo tanto uno prevé, y aquí para Felipe, un poco, cuando uno vaya, me imagino, al médico 10, 20 años más, las tecnologías que vamos a tener disponibles van a ser muy diferentes a las que tenemos hoy en día. Entonces la pregunta es cómo nos preparamos. Y en varias presentaciones he visto, graficado esto, que viene un tsunami tecnológico de proporciones. Y la pregunta, ¿dónde queremos estar en ese tsunami? ¿Queremos esperar la ola que nos reviente? ¿Queremos estar arriba de la ola? ¿En qué parte? Primero tenemos que hacernos la pregunta como grupos de investigación, centros de excelencia, universidades, como país. Por lo tanto, son desafíos. Y lo otro que estamos observando claramente es cómo nos vamos adecuando a estos tiempos. Hoy día probablemente los científicos vamos a tener que estar recirculando nuestro conocimiento mucho, especialmente por todos estos cambios. Y por lo tanto ya no solamente vamos a tener que pensar en la gente joven, vamos a tener que pensar en los académicos intermedios o académicos un poco más longevos, de cómo son capaces de ir actualizando, sobre todo a la luz de todas estas tecnologías. Por lo tanto, creo que este curso, este simposio, tiene esa finalidad que los organizadores tienen, actualizar el conocimiento. Y probablemente esto no va a ser un evento que va a ocurrir cada ciertos años, va a comenzar a ser muy frecuente. Así que en nombre de la academia que dirijo y del Instituto de Chile les doy la más cordial bienvenida, pero también los invito a pensar en estos desafíos, ¿ah? De cómo somos capaces de enfrentarlo, y creo que por eso felicito que los organizadores nos hayan invitado a ser parte de la organización junto con el Centro Milenio, con la Universidad Católica, sus distintas instituciones, Becton Dickinson y otros, para cumplir con este propósito de ir actualizando el conocimiento. Aprovechalo. Felicitaciones.
We thank Dr. Lavanderos for his words. Now, let's receive Mr. Francisco Tawan, Senator of the Republic.
Good morning, everyone. I didn't receive the instructions but I prefer speaking in Spanish. Primero, saludar a la prorectora, saludar, por supuesto, a los decanos que hoy día nos acompañan, ambos, saludar también al presidente de la Academia de Ciencia. Quiero decirle, quiero notificarle que a partir de ahora usted es miembro de la Comisión de Desafíos del Futuro y por tanto tiene que ir todos los días lunes a las 3:30 al Parlamento. Ustedes saben que nuestra comisión, la Comisión de Desafíos del Futuro, que fundamos primero en la Cámara de Diputados, un grupo transversal de diputados, Marco Enrique Ominami, Esteban Valenzuela, también el que habla, fundamentalmente viendo e imitando el ejercicio que hizo Finlandia, que fue capaz de dar el salto cualitativo y cuantitativo al desarrollo, y que constituyó finalmente esa comisión en Chile, en la Cámara de Diputados, pero que lamentablemente no ha podido ser integrada, porque la Cámara de Diputados está en la política de mirarse el ombligo y cuesta justamente construir una política pública de largo alcance desde la Cámara de Diputados, y luego la constituimos en el Senado el año 2010, que buscaba justamente lograr que Chile tuviera un espacio institucional para el diálogo social y que permitiera justamente generar una política prospectiva que permitiera justamente ese salto cualitativo y cuantitativo al desarrollo que tuvo Finlandia en su momento, en la década de los 90, y que tuvo también otros países que pusieron énfasis en el diálogo social. Por supuesto, saludar a Robert, gracias por estar presente, Alexis, que ha sido un compañero de labores del trabajo, no solamente cuando trabajamos junto a la Comisión de Desafíos del Futuro y Salud, que integro además ambas, y me tocó presidir también ambas, en la época de la pandemia. Y agradecer tu trabajo, y ha sido y será nuestro candidato a Premio Nacional, y espero que venga ese reconocimiento en su momento por parte del Estado de Chile. Contarle en qué estamos, porque el presidente de la Academia de Ciencia me hizo inspirarme y dejar atrás el discurso o las palabras que tenía planteadas para hoy día. Es muy relevante generar el clima para que finalmente la innovación y la transferencia tecnológica y el conocimiento finalmente opere en nuestro país. En el día de ayer aprobamos en general, en segundo trámite constitucional, justamente el proyecto que establece justamente la transferencia tecnológica para lograr generar un clima de colaboración, primero entre las universidades y también entre la sinergia público-privada que permita justamente tener tecnología y ciencia aplicada. Y este seminario es justamente un paso relevante en esa dirección. Pero también estamos trabajando en otras materias que son relevantes desde la Comisión de Desafíos del Futuro. Yo fui coautor junto con Guido Girardi en la protección de los neuroderechos. Somos el primer país del mundo y pionero en la protección de los neuroderechos para proteger la indemnidad mental de las personas. Hoy día esa legislación ya ha sido replicada en el Reino Unido, ha sido replicada en algunos estados de Estados Unidos, y se está mirando con mucha expectación en Europa. Logramos sacar adelante, también soy coautor del proyecto y principal impulsor del proyecto de telemedicina, justamente para lograr finalmente establecer condiciones, modificando la ley de derechos y deberes de los pacientes, que permitiera justamente la posibilidad de llegar con especialistas a cada rincón y también favorecer y facilitar la colaboración en temas que dicen relación con la atención, particularmente a pacientes que están en lugares remotos. También estamos sacando adelante, bueno, el proyecto de ley de transferencias tecnológicas y de conocimiento. Estamos sacando adelante la primera ley de inteligencia artificial de las Américas, que está, además, que es la única que tiene adoptado el modelo RAM, que fue recomendado por la UNESCO, y que va a distinguir entre sistemas de inteligencia artificial de riesgo inaceptable, aquellos que, por ejemplo, los controles biométricos en la vía pública, y que solamente se van a permitir para efectos de seguridad, y para los efectos que las policías o el Ministerio Público los pueda utilizar. Y lo hemos regulado específicamente para ciertas materias con leyes especiales, como por ejemplo el aeropuerto. Hoy día se está aplicando controles biométricos en los aeropuertos, y se está iniciando el trabajo experimental en algunos de estos en nuestro país. También se está trabajando finalmente en establecer en este mismo proyecto sistemas de inteligencia artificial de riesgo alto que para poder ser utilizados requieren a lo menos cumplir con 10 condiciones. Primero, que los sistemas de inteligencia artificial sean manipulados por personas y deben estar al servicio de personas, precaviendo fundamentalmente los peligros de la inteligencia artificial autónoma, y por supuesto los sistemas de inteligencia artificial de riesgo moderado, estableciendo un sistema, un laboratorio en nuestro país que va a permitir justamente el ejercicio y el trabajo de los sistemas de inteligencia artificial. Esto además va unido a que fuimos el primer país de América Latina y el Caribe en sacar adelante la primera ley de ciberseguridad, y que sin lugar a dudas nos permite estar preparados y ser pioneros justamente en este horizonte. No solamente ciberseguridad, no solamente inteligencia artificial, también estamos sacando adelante, soy coautor de ese proyecto, de la protección del genoma humano, para los efectos de prohibir la manipulación y la clonación. Es un proyecto que esperamos despachar del Parlamento dentro de poco, y estamos sacando adelante también un proyecto que regula las plataformas digitales en nuestro país. Así que usted, presidente, va a tener que ponerse al día rápidamente y estar en nuestras sesiones los días lunes para ir colaborando en este ejercicio de avanzar en estos proyectos. Pero en este mismo sentido, yo quiero felicitar este trabajo que está haciendo Alexis y Robert, porque esto finalmente implica cómo logramos que la tecnología aplicada finalmente logre desde Chile finalmente ser pioneros y hacer una contribución a la ciencia global. Y por tanto, este workshop que hoy día está invitando la universidad es una tremenda experiencia que espero sea aprovechada por cada uno de ustedes. Señalarle simplemente que estamos trabajando también con el CERN en los temas que dicen relación con los algoritmos cuánticos. Me tocó participar inicialmente en el acuerdo que Chile logró con el CERN, primero con un ex rector de la Católica de esta universidad y de la Universidad de Santa María, ya el año 2007, cuando concurrimos al CERN y firmamos un convenio para que estudiantes de esta casa de estudio pudieran estar y ser protagonistas del proyecto de aceleración de partículas. Fue así como finalmente doctores en ciencia de esta casa de estudio y la Universidad de Santa María finalmente llegaron a trabajar al CERN. el CEN ahora ha dispuesto, y eso es parte también de las conversaciones que hemos tenido con ellos, que ahora no solamente vayan al CEN doctores, sino que estudiantes de pregrado y también de carreras técnicas a perfeccionarse. Y básicamente el convenio que estamos realizando con el CEN dice relación fundamentalmente que por cada aporte en recursos que haga el Estado de Chile, finalmente se devuelve en espacios para estudiantes que puedan ir a perfeccionarse y a trabajar finalmente en el CERN. Así que hay una ventana abierta que lo está aprovechando muy bien la Universidad de Santa María y yo espero también que la Católica haga también ese mismo esfuerzo. Y se está trabajando los algoritmos cuánticos. El fenómeno de la computación cuántica, que esperamos avisore sus primeros inicios, sus primeras aplicaciones prácticas dentro de muy poco. Estamos pensando en un horizonte de 7 años. Implica trabajar primero en los algoritmos cuánticos. Esto va a generar una aceleración de todos los procesos que hoy día tenemos para los efectos de, por ejemplo, determinar una materialidad para una determinada labor constructiva, que una labor que hoy día pudiera demorar 20 años, va a disminuir su tiempo de trabajo y de poder generar un deep learning, fundamentalmente por la cantidad de flujos de datos y estructurarlos y tomar decisiones que nos van a permitir lograr sacarlo adelante en cuestiones de días o de semanas. O sea, lo que se nos viene por delante es una aceleración completa de los procesos de conocimiento y esperamos también de aplicación de ese conocimiento también a la ciencia. Y por eso yo quiero destacar el esfuerzo que hace Alexis y que hace Robert, porque sin lugar a dudas hoy día estamos viendo una tecnología aplicada a la citometría de flujo, que como señalábamos anteriormente no solamente fue esencial para los efectos de la investigación del COVID-19, sino que también respecto de la influenza y otras patologías, sino que también tiene la aplicación en la ecología, que es determinante. Entonces, instarlos a aprovechar esta oportunidad, también a estar muy atentos, porque la educación va a requerir cambios dramáticos en lo que viene por delante. Ya no basta con memorizar contenido, se requiere ahora en adelante, pensando básicamente que en los próximos 7 años, 1/3 de los empleos que hoy día conocemos van a verse impactados. En 2050, la mitad de los empleos que hoy día conocemos van a desaparecer, y eso implica efectivamente capacidad adaptativa, criterio de investigación, que es determinante para finalmente formar profesionales para el futuro. Muchas gracias por la oportunidad. Gracias, rectora, que está además de rectora subrogante. Y por supuesto a todos y cada uno de ustedes aprovechen esta instancia y felicitar nuevamente a Alexis y Robert. Alexis, nuestro candidato a premio nacional. Nos vemos.
We thank Senator Tawan for his words. We invite Dr. Hernán Peñalosa, Assistant Professor and Young Investigator of the Millennium Institute on Immunology and Immunotherapy.
So thank you all the authorities for being here today. It's a pleasure. And also I'm seeing a lot of young faces in the attendees. I hope the people at the streaming are also watching us. It's a pleasure for me to co-organizing this with Professor Kalergis of the School of Biological Sciences. When we talk about impactful science, you have to take in picture that it has to go hand by hand with the advanced technologies. And this is the research that we do in our institution, impactful research that is not just reflected in papers, but also in the training of young scientists. And I witnessed that when I was a graduate student, and there was a vision of some faculties that put some efforts in getting advanced technologies for us. And now as a faculty, it's a pleasure for me, but also a responsibility to work alongside with these faculties to basically train young scientists at the highest level. So this is a call for us, for the young faces here, that we can do that here. You don't have to go outside. We do that here, and it's a pleasure for us and also a responsibility. But also, no matter how big the research group is or an institution, you have to work in collaboration. And we took that seriously. So we invited people from a lot of places to this workshop because we are interested in working with them. We invited Bob because we are interested we know how important it is to get this interaction between academics and industry. And we are happy actually to have you here. And so we have a huge responsibility. We are taking this very seriously, just to impact in different areas of our society. We are interested in doing public science, science with public relevance. So we are very, very happy. I'm very happy to have all of you here, and I hope you enjoy this workshop. Thank you very much.
We thank Dr. Peñalosa for his words. Next, we welcome the Vice President of BD Biosciences, Dr. Robert Valderas.
Good morning, everyone. I think before we start, a couple comments. You know, nearly 35 years ago, as I completed a graduate school, I came into a lab, actually a very famous lab. A lab that had Jonas Salk, it had Sabin in it, it had Dixon in it, that came out of the University of Pittsburgh. And I found myself in their lab. And what was interesting was it was only 1 week after the signing of a collaboration between industry and academia. And it was a very big one with Sandoz. And it changed the landscape of an entire institution, which at the time was the largest immunology program outside of the NIH in Bethesda. And I think those days reshaped what my thinking was and the efforts that I did over many years, although 20 years in academics and now another 20 some years in industrial science, gives you a chance to be able to meet scientists like Alexis, to be able to talk about what could be done. And then it is a matter of how you put the platform together, right? Because it takes two things. Now, the reason why I put those kind of words together from that perspective is that collaborations are a very special space. It's a place where you have to work together with a common goal and sometimes divergent goals. It's a place where scientists from different dimensions need to work together in a different way because we're not in the same locale sometimes. It requires a communication that's actually more difficult than sometimes when you're in the same lab. But what it has behind it is that there are technologies in industry that are not products, but they're technologies that we use to be able to build new products. And if we do it right in our collaboration, then we can share those things in a different way. And those are some of the things that can happen here at this institution. It's not about money. It's not about time. It's simply about how you work together to solve a problem. So one of those scientists many years ago that was in my lab was an individual that kept looking and looking and looking and looking for something, and he never found it. And he was he was in a very rich laboratory, and he spent millions of dollars trying to find a suppressor cell. And then he went home. And he had the opportunity to work collaboratively with many pharma, but unfortunately, he still could not find what he was looking for. Now, I will leave this thought to everyone here because About 10 years after he stepped out of my laboratory, I got a telephone call early in the morning. And he said, Bob-san, I found it. So it was the T-Reg. Now, the reason why I share that is because sometimes when we link a collaboration like we do today with some technology that can reshape what we do, it can bring an opportunity to think differently. I flew back to Japan and went into Shimon Sakaguchi's lab. And I asked him how he discovered it. And he pointed to an instrument that was sitting in the corner, which was 20 years old, which had nothing, no modern technology in it. And he said one thing to me that I'll never forget. It's not technology that answers the question. The hard part is asking the right question and working hard every day. So it's a pleasure to be here. And I know there's one thing that links us all. So for everyone in this room, we all share one thing. There's a patient at the end of everything we do. It'll be a great workshop.
We thank Dr. Valderas for his words. To conclude the welcome addresses, please welcome Dr. Alexis Kalergis, Director of the Millennium Institute on Immunology and Immunotherapy.
So thank you very much, Antonia. She's our leading coordinator in this course. So I just wanted to thank you. She does such a great work, and she's an alumni from the university. Of course, authorities, deans, our provost, senator, my friend Sergio Lavandero, president of the Academy of Science. I just wanted to say two things in this short ending of the beginning of the workshop. First is that when with Bob and Hernan decided to do this, we thought, what are we going to do? We're just going to launch the use of this new technology for the community, or we'd like to make it very scientific. And then I think Bob was a bit busy traveling. He travels a lot. And so Hernan and I were talking, do we really want to do this? Because this involves just, it's a lot of work. And I really want to thank Hernan because he put a lot of thinking, time, connections. Many of the people that will be speaking today were connected through Hernan, previous experience in the US. So we finally organized a workshop that covers pretty much every continent. We have speakers from the US, speakers from Europe, from Asia. Actually, it was last week in India, just before the war started with Pakistan. And one of the speakers today, he wanted to be in the symposium. He will be our last speaker. So it's a very comprehensive workshop about science, as Bob mentioned, science that is like cutting edge, something very important that Professor Peñalosa said. So cutting edge science helped with technology that we are very lucky to have here in our university, and also connected with other universities like University of Chile, University Andrés Bellos, and other universities that are part of this collaboration. So thank you so much for being here. Thank you to the audience. It's a rainy day. You have the option to stay home and watch it in YouTube or being here. I'm very grateful that many of you decided to be here, and because it never rains in Santiago, and you're lucky, Bob, your day here, you have some rain. So thank you for the effort, and of course the organizers, students, the technology folks, they're very helpful. And we hope you enjoy the workshop. Thank you so much.
We thank Dr. Kalergis for his words. Now, we invite Dr. Longyang Wu, Director, Core Facility Institute for Immunology in Tsinghua University in Beijing, China.
Good morning, everyone. It's a great honor to be invited to participate in this symposium. I would like to express my sincere gratitude to Professor Alexis Kalligis and the conference organizer for their kind invitation. I come from China. I'm the director at Tsinghua University. I'm the director of the co-facility of the Institute for Immunology. I'm also the senior research associate in the School of Basic Medicine Sciences. Our platform is affiliated with the Tsinghua University Institute of Immunology, and our institute was established in 2014. Now we have 15 PIs in our institute now. We have mainly Professor Chen Dong is our founding director, and the institute now is led by Professor Hai Qi. Through 10 years of development, Tsinghua, our institute was ranked among the top 3 in Asia in the field of immunology in 2023. Here are the 15 research team in our institute, and recently we just recruited a new member, Xiaoyun Chen. She used to work in Washington University School of Medicine in the field of neuroimmunology. Here are the research fields of our PIs, include the innate immunity and inflammatory diseases, and the adaptive immunity, infection immunity, allergic disease, tumor immunology, autoimmune disease, and immune cell development and function. Also, we developed the neuron immunology, a new field. Our professors are highly capable and have published remarkable numbers of scientific research articles over the past decades. Maybe this is because we have a good instrument platform. This is the platform of option architecture of our co-facility. We are the first public instrument service platform to implement the 7+24 hours optional model. We have a platform committee include 4 PIs in our institute for the platform planning and major issues decision. They are also my visors. And I'm the director of this platform for daily operation management and the technical guidance. The external technical services and also new technology development. The daily startup and the maintenance of the instrument is operated by technician. We have 4 technicians now, and we are going to include other 2 technicians. They also help the users to use the instrument and do some hands-on equipment training, and also routing some external technical services. Another part is very important is our student assistant. We maintain about 8 to 10 students in our platform for the startup of the instrument and troubleshooting during the non-working hours. Here are the architecture of our co-facility. We have many instruments now. Originally, we centered around the flow cytometry, and then after 10 years of development, we have nearly 17 many kinds of flow cytometer now, and also expanded to include microscopy and high dimensional multi-omics equipment and other sharing instrument from dozen instruments to over 20 pieces of diverse equipment in our co-facility. Last year, not last year, 2023, we installed 3 BD high-level instruments. So last year, they are the priority equipment to promote. They are the BD Symphony A5 SE analyzer and S8 sorter, and also the Rhapsody single cell sequencing, single cell system. The Symphony A5 SE, we installed the Symphony A5 in 2019 and updated in 2023. It's a 5 laser, 48 color instrument, and a full spectral flow cytometer. It can combine with traditional composition and spectral analysis. So the voltage is preset and easy to use. So students like to use this instrument better than a FOTASA. The highest parameter panel set up on this instrument is 36 colors a panel, T-cell deep type panel. So the advantage of this instrument is self-emitting fluorescence subtraction, and more similar fluorescence can be used in the same panel and easy to set up. During the external services, I found many students couldn't get good results because of their lack of experience in experiment design, sample preparation, or data analysis. So I set up the FACS integrated service system for the students not in the field of immunology. From the panel design, I will communicate with the users what marker do you want and what antibodies in your lab, what kind of antibodies in your lab already have in your lab. And the second, I will design the panel and discuss with the BD company, choose the proper fluorescence for each marker, and then set up the sample preparation. We have a tissue dissociator and Max, Automax Pro from Mitini, and also can do the PBMC centrifugation. Then we will help the users to running the sample query on Symphony A5SE or Discover S8. And finally, we also offer the training for the use of FlowJo to high level data analysis. So Professor Lin Qizhang is our major customers because she is not in the field of immunology. She's not an immunologist, but his subject is should use the techniques in the immunology. So I help the students to design their whole protocol. The second instrument is the BD Discover S8. It's a full spectrum imaging sorter. It has 5 lasers and 78 fluorescence channels, 8 imaging channels, and totally have 86 detector. Its advantage is there are there are 8 imaging channels on the 488 nanometer laser. 3 fluorescence imaging channel FITC-PE P7CY7. Each channel have 15 parameters to analysis. Here are the parameters can be used for each channel. And another advantage is the non-label sort, FIC, SAC, and light loss. So we can use the non-label sort for nuclear sorting and other samples. It's also a high throughput sorter. The sorter rate can reach 10,000 events per second. So in our platform, we use it on the cell cycle, cell contact, phagocytosis, and the colocalization of the proteins or the contact of the cells. Here is the our first user's data. She do the immune cells, phagocytose, the microsphere. She used to use a confocal to scan the cells and count it manually. Very expensive and very costly, and take many times. So when we install this S8, she is the first user. You can see this is the just get out the cells. This is this cells has one microsphere, and two, and three, and many microsphere. Just get it out and export the statistics. She will get the result. Very easy. I will only give this example. There are many other examples, but not in this report. So the flow cytometer, the technology has developed many major protocols, also published on the flow cytometer part A. So they are the very important reference for us. So if you want to set up a new panel, you can refer to the matrix scheme on the literature and do some personal tailor. Nearly all the users is major protocol plus the major tailor. Single cell sequence is next to the cell sorter. So I set up the single cell sequence integrated service system. Also because many clinical doctors don't have the ability to do the sample preparation, and also don't know how to sort the cells. So I will set up from the beginning the experimental design, sample preparation. We use tissue dissociator and density gradient centrifugation to get the sample, or use max to get to enrich the cells. Then sort the cells using area 2 or discovery S8. We have 4 area in our platform and 1 fusion. So it's very convenient for us to do a large experiment. And then to capture the cells or RNA, we have 2 different methods to capture the cells. First is BD Rhapsody. It's a microwave dependent, and another is a China band, Mobile Nova. It's similar with the 10 genomics. It's water in air illusion based instrument. Then sequencing it Illumina or China band BGI. Then we have our own computer servicer to transfer the data. And then data analysis using BD software or our platform. And finally, we can validate the data on our A5SE and other instrument. I will introduce the BD Rhapsody in this report. It's launched on 2023 and in the end of the 2023. So we installed it last year, about January. It's a high throughput multi-omics single cell analysis system. It's really a high throughput instrument. It have 8 channels. Each channel can load 1,000 to 10,000 cells and capture nearly 60,000 cells per channel. So also can use sample tag to save reagent and minimize inter-batch variability. That means sample tag we can label different cell samples can label with sample tag and mix it together. Each samples we only use one reagent and get several samples data. It is also a real multi-omics system because it can combine different multi-omics together in a single experiment, RNA, protein, or VDG, or ATAC. The common is the transcriptomics, the RNA, WTA, and also can make samples together at the sample tag SMK, and also can do the immuno-omics, VDG, TCR, BCR sequence. We promoted recently the AppSeq. In one cell, we can detect the RNA and the surface protein expression level together. We know BD's antibody is famous in the field, so they also use it in the single cell sequencing analysis. So we can get RNA and the proteomics together. Human has more than human and mouse. They both have more than 200 antibodies and also have some fixed panels in the human data analysis. We are going to do this experiment in the field of T or B cells using this AppSeq. And they also have 14 intracellular proteins antibody can be used. All these omics can be combined together. This is an example. You can see the priority of the transcriptomics plus the proteomics. It can divide this cluster very clearly. This paper is a very classic examples for the use of RNA and protein, and also the sample mixture. If you feel interested in this application, you can read it for detail. The author detected HPLCs bone marrow bone marrow and labeled cells with 97 to 197 antibodies. This is AppSeq, and makes some cell samples together and do the sequencing and get many low number of cells in his result. And another product of the BD is the ATAC-seq. We have done several ATAC-seq plus RNA-seq together, the transcriptomics to epigenomics last month, and get some good results now. Because the RNA-seq is very common in our research, so researchers also want to know the epigenetics of this gene. GBD is the only product can combine this together to do a one single nuclear, the RNA and the ATAC. I like the BD Rhapsody because it's full process visualized. We can see the cells loaded into the microvial, and we put the beads, load the beads. You can count one beads with one cell, one well cell in the well. And also you can, when you get all the beads out of the well, you can see it's very clear, no cells left. This is a report on this scanner. We have done more than 100 experiment samples in our platform. So I have some experience in how to get a good result in the salt plus single cell sequence. Only one sentence, high quality single cell suspension is the very important to get the good result. There are some do's and don'ts. First is viability. Some samples are T cells, or some samples are PBMC, the cell suspension. So the T cell storage is very important. Fresh T cell is where is the best choice. But if you don't have enough time to dissociate the immediately, you can storage the T cell in a storage solution. You can use maintaining mass T cell storage solution that can storage the T cell 48 hours, within 48 hours. Well, BD is the new sample preservation buffer can save the viability up to 72 hours. And you also have to find the proper dissociation buffer and steps. This is the cellular show from the BD's protocol. You can see the PBMC can can maintain the viability up to 7%. And it's my data. It's a tissue from the uterus, a human uterus. And the viability is more than 9%, 90%. The second is a short strategy, because the good FACS protocol is very important for the viability of the cells. The live dead dye is must be used. CMAD, Jumbo system, Jumbo, and FAS, etc. And I found salt with the calcium, the drug 7 staining together is very better for the CD47 sorting experiment. You can see we can get out CD47, 45 positive immune cells and get out the dead cells, and also the calcium can label the live cells very, very well. And in my own experiment subject, I studied granulocyte. I'm very lucky to choose the BD Rhapsody because I found the eosinophil is the main subject in my project. If I use other methods, I can't get the eosinophil group. The third is the sample reception liquid. Many students want to get a viable cell, want to the cell to stay in the cells stay in a comfortable liquid. So in this experiment, the students use 10% FBS medium as the reception liquid. So we found very high doublets and large debris from the FBS. You can see in this picture. So we changed the reception liquid into BD FACS stain buffer. It only contained 2% FBS. So we solved this problem very low doublets and no debris in this sample. From the sequence data, we also can see if we have high debris, the waste many sequence rays, but the sequence saturation not very high. But we don't have to seek so many data, but we can get a high saturation, sequencing saturation in a good sample. The fourth is SMK mixed samples. They can save money because when you set up the mixed samples, you must see the FACS data. It's based on the FACS data. You must have enough cells, 100,000 per sample is better. Can sort CD5 positive or negative subsets separately and mix them together and seek the cells separately. This is an example from the mouse skin tissue. The students want to sort the CD45 immune cells and mix 4 samples together and get nearly 10,000 cells in each samples and low undetermined cells, low multiplex cells. But the CD45 negative cells, if we labeled with sample tag, we can see there are many undetermined cells because the sample tag labeled CT45 negative cells is MHC1. It may be disappeared when you dissociated the tissue. So it's not recommend to mix on non-immune cells. And another example is cells very the number of the cells is very low. We can't get enough cells. For example, in this experiment, they thought human TFA cells is very little subject in patients' peripheral. The one sample only get 40,000 cells. So we can stain the sample tag with the FACS antibody together and sort them out and count the cell number, mix them together, save nearly 1 hour, and the viability of the cells is much better. In this experiment, we totally get 57,000 cells, and in each sample we can get more than 10,000 cells in each sample. Another, I will give some user case in next slide. This is using SA to sort cell nuclears for RNA sequencing. We choose. You can see the first gate to get out debris. The red arrow shows the debris. This is the nuclear, and also use radial moment and eccentricity to get out the round regular shape cells, nuclear. And use the light loss H and W to get out the doublets cells. The light loss in S8 sorter is equals to FSC. And the students also use a dye to get out the G2 cell cycle cells, nuclear. You can see the nuclear is very clear and round. We sort out more than 80,000 nuclear and load all the nuclear into the well, and the putative cells finally we get 223,000 nuclear finally. And in this experiment, there at first we don't get the after sorting, we don't get the cells because the user the student user a wrong sample reputation liquid. She wanted the nuclear to comfortable, and so she chose the nuclear storage buffer. We don't know the what is it, but if the cells are sorted into this buffer, it can't be centrifuged down. So at the first time we don't get any nuclear. It's very strange. So the second time we use fast buffer as the reception liquid. So she got a good result. Another user case is RNA and VDG. The students get 63 killed 63 mouse and get the brain in real time. Nearly 5 or 6 postdoc or students postdoc help her to do this experiment and sort with area. Sort of the reported positive cells only get 10,000 cells in 300 microliter. And finally, we get nearly 10,000 cells finally, and the high quality T cells, she saw the T cells is 1,000. So the advantage of the BD system is a large loading volume. We don't have to change the sample buffer. Only use the fast buffer to move part of the supernatant and left 300 microliter and suspend it and load it directly. Another use case is RNA and SMK and the BCR. It's from Linqi Zhang's lab from 8 humans PBMC labeled with sample tag with a FAX antibody together and sought 2 kinds of antigen-specific B cells. Only eight samples only get less than 3,000 cells, and mixed with T cells, equal T cells in 300 microliter sample buffer. And when we load it, but the viability is very, very bad, only less than half. So we only get 2,000 viable cells, and one sample gets less than 2,000. And this sample, the result we transfer finished state is 1,600, and the high quality B cells is more than 500. And the final example is very complex, because the user first sort out YP positive and YP negative cells to do the RNA WTA, and also do the WTA plus the ATAC-seq. She only get 140,000 cells in the YP positive group. And finally, we get enough cells. You can see loaded cells and the putative cells. The capture rate is very high, and the WT and ATAC-seq data has back 50,000 cells. We get 38,000 nuclears finally. And she transferred the YP plus cells, FP positive and negative cells into the recipient mouse. And 3 days later, saw them out to do the RNA-seq again. The YP positive cells only get sorted out 4,000 and 1,500, less than this number. And when we loaded them all into the chamber, and we get more than 3,000 cells finally. She is satisfied, very satisfied with this data. The sequence data analysis is maybe very difficult for many students. Our platform don't have enough technician to do this.
Dr. Wu,
Okay,
sorry to interrupt, but we need to move
only
along with the next speaker.
the last yes. This is the BD Celesmo is a new software, very workable in our platform. The priority requirement for 2025, we will promote this instrument. Integrate. I want to integrate the multi-omics in the field of immunology. Oh, thank you to all my outstanding users and colleagues. Thank you for your attention. Thank you.
Thank you very much, Dr. Wu, for your presentation. Next, we welcome Dr. Pablo González, Associate Professor of the School of Biological Sciences from this university and Millennium Institute on Immunology and Immunotherapy Principal Investigator.
Okay, so first of all, I want to say a special hello to students from the Bio 228C from the biochemistry course and molecular genetics that are today here interested in this technique. So I'll show you some slides about the technique so that you can grasp this from the beginning. So un especial saludo a los del Bio 228C por estar aquí presente y a todos los demás. So also I want to thank, of course, the organizers for inviting me to present today a technique that adapts flow cytometry to the study of metabolism. You will see this is a technique that usually is quite new. It doesn't require too much sophistication. Actually, you'll see you can do it actually with almost any flow cytometer. And it's a bit what Bob said before. Sometimes it's more of coming up with the hard questions, the good ideas, and how to implement them that make important changes. But of course, during the workshop, you'll see other implementations of the amazing capacities of the new cytometers, specifically and mostly in spectral flow cytometry. So as it says there, I'll show you this new approach for analyzing cell metabolism. So as I mentioned, because we have here some students from a very early stage of undergrad careers, I just want to explain very rapidly some aspects of flow cytometry so that they can grasp this from the beginning for the rest of the workshop. So the flow cytometry you saw already is kind of a box. There's many things happening inside the box, and mainly what's happening inside is what you see here. You have a flow cell. That's where cells are put into in a straight line. So you have a sample with different cells. These can be cells. It can also be beads. It can also at the end be events. So you just need a particle, a small particle that can be put into a laminar flow of liquid, and then cells or the events are aligned one by one after the other. And as they pass one after the other, they're hit by one or multiple lasers. And then depending on their characteristics and the fluorophores they may have, these cells or events will fluoresce. And they will fluoresce in different wavelengths. So you can actually detect those wavelengths with different detectors. And you'll have kind of what you see here, a spectrum of fluorescence for that event. And that, of course, you can collect it in a computer and determine what is the characteristics of fluorescence of each event that's passing through this cell. So furthermore, you'll see also there's some flow cytometers
that can sort the cells. This means that once the cell has been analyzed, you can separate the cell into a different tube. And this allows you to then go and sequence what's in that tube, which is what you're interested in, because you have selected those cells to be put into that tube. So what do you see when you collect the data? Well, you mainly see the information in two forms. One is in the form of an histogram. And here I'm showing fluorescence intensity distribution for one analyzed parameter. So it's only one parameter that I'm showing here. And what you see is intensity of fluorescence versus number of events. And each dot here is one event, which in this case can be a cell. And you'll see that one cell with this fluorescence will accumulate with others that also have the same fluorescence, and you'll see a population. You'll see a global population of cells. And this will have as many cell populations a Gaussian distribution. So you see a Gaussian distribution of cells with that range of fluorescence. And you can distinguish positive cells for that fluorescence from negative cells from that fluorescence, which are here, because they separate based on their fluorescence intensity. Now, because this is the most simple example of one parameter, you can start looking at two parameters. And in these cases, what you see is this kind of graph, where you have quadrants that will show you the intensity in parameter number 1 and the intensity on parameter number 2. And you'll start to see cells that gather in different populations in different regions of this graph. And you'll see cells that are both positive for parameter 1 and parameter 2, cells that are double negative for parameter 1 and parameter 2, and then cells that are positive for one parameter or for the other. Now, this is also a very simple case because it's two parameters, so we can see it in 2 dimensions. But nowadays, with spectral flow cytometry, you actually have up to 80 colors, 80 parameters that you can analyze. And of course, we don't have enough dimensions in space to represent all these 80 parameters. So nowadays, what you'll start to find and start to see is this kind of analysis where you have distribution of populations with different markers, and you'll see that they gather together based on their characteristics. But each region will actually be a collection of cells with multiple parameters that make them fall together in that group. So it's becoming each time more complex, but also the amount of information we can get from this type of analysis, it's amazing. It's a complete transformation. When I started my thesis, I could see 4 colors, 4 parameters. Now we can see 80. So the depth of knowledge of information we get is just amazing. So flow cytometry, I may dare to say, started in immunology. Very early on in immunology, we could, with flow cytometry, analyze the different cell populations. So the main application, or one of the most common applications, was actually fundamental research. But of course, there was a niche, a space for this technique in diagnosis. So actually, when you go and analyze your blood, when you get a blood sample at the hospital, and you actually are doing diagnosis, you're looking at the cell populations in your blood based on flow cytometry. And of course, also this has found applications in follow-up of treatments. Nowadays, you can follow up cancer treatment by analyzing your blood and seeing how you're responding to treatment. Now, although flow cytometry started in immunology, then it extended to other disciplines, to biomedicine, then overall to biology, to life sciences. You saw the dean of our faculty that she mentioned. You can do flow cytometry today in ecology for analyzing samples in the environment. This has also extended to food safety, environmental monitoring, as she mentioned, and also even nanotechnology. We're talking about analyzing things that are very, very small at the cell level, micron big. Now, within the field of biological sciences, also, there have been expansions on the applications of flow cytometry. So for example, we can analyze cell signaling events. We can, of course, analyze the viability of cells, protein expression. Those who work with microbes, we use this very frequently for analyzing infection. You can study cell cycle of cells. You can study fluxes of calcium within the cell, soluble molecules using solid platforms as platforms for binding molecules of interest. You can analyze reactive oxygen species, phagocytosis, and what I'm going to show you today, metabolism. So metabolism is also one of the applications that now you can analyze using flow cytometry. So the platform is incredibly diverse in applications. So one important concept I think I would like to share with you is that nowadays metabolism is way beyond cellular energy balance. So you'll see, and we'll see in the course of biochemistry, that metabolism is mainly energy balance, but this is an old vision of metabolism. Nowadays, metabolism is going beyond this classical approach and is involved, for example, in neurodegenerative diseases. So we see this more frequently, each day more frequent, that you have papers, reports, reviews that are showing that metabolism plays a significant role in the shaping or the function of cognitive functions in our brain. You even have these new concepts, such as type 3 diabetes, which are based on metabolism alterations and their impact on neuron development. You also have metabolism impacting stem cell fate. So there's also a huge area of research that shows how metabolism, how the choosing glycolysis versus oxphos can, phosphorylation oxidation can define what a cell is going to become. You also have, of course, the impact or the role of metabolism in cancer. Actually, there's a very known effect called Warburg effect that's seen in cancer cells. And this, of course, was the initial step in analyzing the impact of metabolism in cancer cells. You also see metabolism involved in autoimmune diseases. And of course, you also see them now involved in infectious diseases. And actually, we've been very interested in this field. Actually, we have a few papers on this, and we actually are looking now at how viruses, and particularly herpes simplex virus, can modulate the metabolism of immune cells to evade the immune response. And this is what's being called immunometabolism. So just want to show you briefly why we're focusing on these cells, on dendritic cells. So dendritic cells are immune cells that can control the fate of T cells. They can put an imprint on those cells. And T cells are important because they can help other immune cells. So for example, this is a T helper cell that's actually helping a B cell. And it's helping the B cell to define what kind of antibody it will produce. So the role of the dendritic cell is very important because it's at the beginning of this initiation and regulation of the immune response. What's been reported recently is that dendritic cells, depending on what they eat, they can have different profiles, different phenotypes. And those phenotypes, depending on what they eat and what their metabolism is, will define the outcome of the T cells with which they interact. So depending on whether they're doing glycolysis, oxphos, or lipid metabolism, they can shift then the immune response toward different types of response that either resolve cancer and infectious diseases, either resolves parasitic infections, or either resolves extracellular bacteria infections. It's also involved in regulating tolerance to our self-antigens. Now, so as I mentioned before, we've been interested in going into this field lately. We have this very recently published paper from my lab, from Monica Farias, where we saw that dendritic cells infected with herpes simplex virus have a significant shift in their metabolic lipid-related genes. So you can see here that we have many genes within an array of lipid metabolism genes that are either upregulated or downregulated. So there's a complex alteration in terms of how these cells respond metabolically to infection with the virus. You can see also in this type of analysis, where you have now in transcriptomics, hundreds of genes, and you can see their overall behavior and how they impact overall processes. So for example, here we have adipogenesis. It's been affected because many genes are either upregulated or downregulated, and we know that when that happens, that means that that process is being altered. So we see that there's significant alterations in lipid metabolism. We even see the formation of lipid droplets. So what you see here in green is the accumulation of drops with lipids inside, which is not normal in these cells. These cells are healthy. They don't have lipid droplets. When they're infected, they start to accumulate lipids and become fatty dendritic cells. And when we do lipidomics and look at the lipids that are being synthesized and produced in the cells, we start seeing families of lipids that are also telling us what routes of metabolism are being turned on and which ones are being turned off. So this is quite complex. We will certainly need in the future AI to deconvolute, to understand what's happening in a more simple way, because many things are happening. But in the short term, we're also very interested in studying how lipid metabolism relates to carbohydrate metabolism in the cells. So we started to analyze carbohydrate metabolism in dendritic cells infected with this virus. And that's part of the thesis of Francisco Romero, who's right here. So for this, traditionally, we have used this technique called seahorse, which is this equipment that's quite scarce. It's very expensive, and it's quite scarce in Chile. Actually, I know two of them only. One of them is at the laboratory of Álvaro Elorza in Universidad Andrés Bello, and the other one is Universidad Aysén, way in the south of Chile. So it's a very rare equipment, and also the modules to analyze are very expensive. So obviously we started doing some of work with this approach because we had the chance of Dr. Elorza collaborating with us, and we can see that dendritic cells, when infected with the virus, they have alterations in their carbohydrate metabolism. So we analyze what's called ECAR, which is extracellular acidification rate, which relates to glycolysis. So production of protons into the media is a readout of glycolysis. And we can see here that infection produces some alterations of these parameters. Now, this is very broad, and of course, we would like to see this at the single cell level, or specifically over cells that have certain phenotypes. And looking for that approach, we found this new methodology, relatively new methodology, that allows analyzing metabolism by flow cytometry. And this is the technique that's called CENIT, from a group established in France that has established a flow cytometry-based method for functionally profile energy. I have here, don't see it whole there, to functionally profile energy metabolism with single cell resolution. And this approach is quite interesting because, as the title says, it's a single cell level analysis. So we're not analyzing a population, we're analyzing cell by cell how that cell is, metabolism is behaving, and based on the profiling of translation inhibition. And I'll immediately explain what that means. So there's this finding very old, more than 30 years old, that has established that in the cell, most of the energy, most of the ATP is used in translation. So up to 50% of the ATP in cells is used on translation. So this important consumption in that process gives us the opportunity to use translation as a readout of ATP levels in the cell, because they're so tightly related and it's so expensive to produce proteins that reading protein translation, I can translate that into ATP availability. So with a technique that allows me to assess translation, I could read out ATP. And this is exactly what this technique does. So how does it do it? So it's quite easy, actually. You use an antibiotic called puromycin, which binds to nascent peptides that are being synthesized in ribosomes. So you can see here puromycin within the ribosome, a ribozyme is being incorporated in the last amino acid. And you can see that translation is terminated early on with the peptide having puromycin bound to it. So now you can go with an antibody against puromycin and detect the amount of peptides that have puromycin on its end. And this is a readout of translation. The more puromycin bound to peptides means that more translation is occurring. The less puromycin you detect bound to peptides, less translation is occurring. And if there's a relationship, direct relationship between ATP and translation, then with this method, I could read out ATP. And this is exactly what you see. So you look at the bottom here, when you inhibit glycolysis and OXPHOS, you have a steep drop in ATP availability in the cell. You're cutting metabolism related to glucose. And at the same time, if you're inhibiting those processes, you can see that the decrease in protein synthesis is very similar as how it behaves in the drop of ATP. And if actually you do a correlation between ATP drop and translation drop, you almost have a linear relationship. So again, that means if I can measure protein translation, I can transform that readout into ATP presence in the cell. So now I have a method for detecting ATP or metabolism in the cell based on detection of translated proteins. So this is very interesting because we do have different inhibitors that can block different processes of metabolism in the cell. Mainly here, we have aerobic glycolysis, glycolysis with OXPHOS in the mitochondria, and also you can have in the mitochondria fatty acid oxidation or amino acid oxidation. And we have different inhibitors, for example, 2-deoxyglucose, that can inhibit the input or income or incorporation of glucose into the cell. And those, by using this, you can block glycolysis. And at the same time, also block OXPHOS. And then if you use oligomycin, which blocks ATP synthase in the mitochondria, you can block OXPHOS and also block mitochondria-mediated fatty acid oxidation and amino acid oxidation. And if you use both, you actually kill the cell because you give it no option to metabolize anything because you're cutting both the carbohydrate metabolism and also fatty acid and amino acid metabolism. So with these two inhibitors or three combinations, you can now dissect what kind of metabolism the cell is using. So this is what you see here. You have a population of cells, a mixed population. You have population 1, population 2, and using these different combinations, control, do the 2-deoxyglucose or oligomycin or both, you can measure the levels of protein translated, and based on how they behave, the levels you get, you can translate this into information whether the cell is using glycolysis or mainly using mitochondrial metabolism. So this is what you see here. So this is a histogram. You have the control cells on the right. You have the cells that are completely shut of any source of energy on the left, which is when this here. So you can do, and you can have the one with glucose incorporation blocked. And based on the levels of fluorescence that you get for these three conditions, you can determine glucose dependence of the cell. And here you can see, for example, this cell, this particular group, is depending 91% on glucose for its metabolism. And you can actually do the rest. You can do the subtraction, 100 less 91%. So the rest of the metabolism is due to fatty acid oxidation or amino acid oxidation. So just with that technique, you can immediately know what is the profile, the metabolic profile of that group of cells. The same thing you can do here with oligomycin. Again, you have a control condition. What we're measuring, remember, is translation by detecting puromycin in the cell bound to a peptide. So you have control levels, which is translation in normal conditions. You have translation that's completely shut when you block the whole metabolic processes with both inhibitors. And when you block with the oligomycin, what you're blocking particularly is OXPHOS. So again, now based on a mathematical calculation, you can determine mitochondrial dependence and based on the subtraction, glycolysis capacity. So again, you have a profile, metabolic profile based simply on a few parameters that are measured. Of course, those that use seahorse will want to know how well does this relate to the other technique. Well, these authors also did it, and you can see you have a very nice correlation between seahorse and this new technique. And the great advantage, as I mentioned, is that this you can do with practically any flow cytometer, while seahorses are quite scarce. And here, of course, you have comparisons between both techniques, and you can see that for this flow cytometry approach, you do not need to purify cells. You can do at the same time a phenotypic analysis of the cells, which you cannot do with seahorse. You can cell sort the cells after the analysis. You can keep those that you want to further analyze. And you can do this much more rapidly than with other techniques. And you need very few cells. And you can do it with any flow cytometer. So it's very, very smart approach that I would highly recommend. So just as a conclusion, metabolism is as I mentioned early on in the presentation, is being found to have profound effects over numerous diseases. It's impressive how diseases that were thought to not have any relationship with metabolism now have a relationship, so we can maybe target those diseases through that approach. There's an important need for analyzing cellular metabolism at the single cell level, and this is particularly important for cell populations that are complex. For example, those extracted from in vivo settings, from an animal, where you have a mix of different cells, and you just want to know what is the metabolism phenotype of a particular cell type in that mixed population. So it's very useful because you cannot do that with seahorse. Of course, this novel approach or practical approach for determining metabolism preference in different cells upon different treatments will allow us to better understand what is the role of those different metabolism profiles in these cells in the context of health and disease. And I also am sure, and I think we should expect, that there will be future AI-designed approaches based on inhibition of some other processes that upon the convolution, simple or complex, assisted by bioinformatics or not, will allow us to read out more specific preferences in metabolism in these different cells. Just a matter of time, how we identify those routes and how we deconvolute that information. And then also I want to share with you, just because of a matter of time, I don't want to extend too much, that there are also other very interesting approaches that complement these metabolism analysis that are available today. For example, analyzing the state of reduction of NADH by flow cytometry. We were impressed with Francisco the other day that this can also be done by flow cytometry, something that people did not think about it before. It's a very simple question. Almost any equipment can do it, but nobody had asked themselves this hard question or thought about it enough until someone did and found that you can actually resolve this using flow cytometry. And this is a very recent paper from, as you can see, from just last year. So I just want to highlight the participation of the data of Francisco Romero, who is here today in part of his PhD thesis. Is actually studying carbohydrate metabolism in dendritic cells infected with virus. And we will be implementing very soon, we already started in the lab, this approach based on flow cytometry. So we'll be also happy to share with any of you our experience, whether how this works or whether it provides the information we're looking for. Thank you very much.
Okay, so we have time for one or two questions for the question.
Good question. I haven't seen it, but I would guess it should be applicable. So actually, puromycin is an antibiotic that works in eukaryotic and prokaryotic cells.
Oh, that's
so actually you could extend it to bacteria. They do not mention it, but certainly would be a feasible approach, I think.
great. Thank you.
Thank you, Pablo, for the great talk. So just advertising, we do have a seahorse, an upgraded actually seahorse at the Faculty of Medicine. You're welcome to use.
We'll be asking for it. Good to know.
Yeah. I was amazed about the, how would you study metabolism in the different cells and what you show about the dendritic cells. So do you think there is a link between the dendritic cells being able to prime at Th1, Th17, and analyses that also we know that they actually they have a milieu of cytokines that may direct the capacity of the dendritic cells to prime different ways T cells.
Certainly. So actually, I think something that has not been analyzed that much is how cytokines could impact the metabolism of a cell detecting those cytokines. And that may define that different combinations of cytokines could also provide to these types of cells, innate cells, as ILCs, different phenotypes based on their metabolism, not only on just the signaling pathways and transcription factors. So metabolism, at the end of the day, is much more involved in the fate of the cell as we thought before. Before it was just metabolism is cellular energy, just making sure there's always ATP. This goes way further beyond that. And I think we're starting to discover this, and it will be relevant for the future for new therapies and treatments and prevention. Thank you. Thank you.
Thank you very much.
Okay, welcome. Thank you.
So to close this first session, we now welcome Dr. Ignacia Negon, who will be joining us online. FOSI's President and Director of Research in INSERM, Emerit in Nantes, France.
Okay. Hello. Do you see my slide? Do you hear me?
Yes, we do. We're
Okay.
not seeing in presentation mode.
Okay. Let me put in presentation mode. What about now?
We now see it.
Okay, great. Good morning. Buenos días. Bonjour. Happy to present my results. I'm very grateful for the invitation. So the topic of my talk will be CD8 T-Rex. This work has been done in this INSERM center in Nantes, France. And this is the outline of my talk. I will give some background on CD8 T-Rex, some results on rat CD8 T-Rex and then human CD8 T-Rex. I will describe a clinical trial that have begun with patients-derived CD8 T-Rex and some results with genetically modified CD8 T-Rex. And these are my potential conflict of interest. So why T-Rex cell therapy? Well, when there is let me see if I can turn on my my pointer. Yeah. When there are autoimmune diseases or allograft rejection, there is tissue destruction by common effector mechanisms. And basically, the working hypothesis is that the T-conventional cells illuminate over the Treg, which mediate the tolerogenic mechanisms. And immunosuppressive drugs are effective in part to control these destructive processes, but also this comes with a price because there is first inhibition of both T-conventionals and Tregs. So this hampers the possibility of establishing long-term tolerance. And there's side effects as cancer and infections, and even off effects like chronic dysfunction for kidney grafts, for example. But the possibility of using Tregs is that there will be an equilibrium establishing immune homeostasis in the case of autoimmune diseases and allograft tolerance in the case of organ transplantation. And although there are more of 60 clinical trials using CD4 Tregs in different settings, solid organ transplantation, GVHD, autoimmune diseases, etc., there are no clinical trials until now. This would be the first one. Despite that the suppressive action of CD8 Tregs have been demonstrated in many pathological situations, and there are functional similarities also between CD4 and CD8 Tregs in terms of mechanisms. But CD8, for example, recognize MHC class 1, which is presented by all grafted cells or all tissues or all cells of any tissue, whereas CD4 Tregs will be activated through MHC class 2 antigens, which are present mainly in APCs. And there have been also publications that show that there is better suppression of memory cells when CD4 T-conventionals by CD8 Tregs versus CD4 Tregs. So in a certain historical perspective, CD8 Tregs were the first suppressor cells that were described by Gershon Group in 1972, but in 1988 they become discredited mainly because of the absence of markers. We didn't have at the time the technologies that we had later on to define them. And the first avatar of resurrection of suppressor T cells were the CD4 Tregs described by Sakaguchi. group. And there is a second avatar in terms of CD8 Treg perspective, which is the description of the CD8 Tregs field established by Cantor's group at that time in the mouse. During these years, there have been different subsets of CD8 Tregs in a certain way, similar to CD4 Tregs, in which there are FOXP3 positive and FOXP3 negative CD4 Tregs. With CD8 Tregs is the same thing. This Cantor's group mouse CD8 Tregs were FOXP3 negative. And very recently, Mark Davis' group also described the same type of cells, CD8 Tregs FOXP3 negative in humans. But there also have been CD8 positive FOXP3 positive Tregs and different markers. The problem is here that there have not been really a systematic comparison of all markers in different species, for example. There have been different markers in different species. We have used these markers, CD45RC low negative, and described this population primarily based on this, but with other markers also. I just wanted to show you this paper, which is interesting, I think, because it shows that there is continuous research on CD8 Tregs, and this paper described the CD8 FOXP3 positive Tregs, and interestingly, much more present in tissues, for example, intestine, as compared to lymphoid tissues. In our case, we began using the CD45RC markers to enrich for CD8 Tregs, and in particular, the isoform RC. So CD45RC, and this is different from RA or RB or RO, is expressed in naive Th1 precursors and in effector T cells, but not by Tregs. So Tregs are enriched in this fraction of CD45RC low negative cells. And this was described early by the Mason groups for CD4 Tregs, and then in CD8 Tregs by Abdel Saudi's group and our own group. And again, the CD8 Tregs were in the low negative fraction, like schematized here. And using this marker, we have analyzed in other groups, not only in rodents, but also in human, the impact of this. And for example, these are examples in kidney transplantation, where other groups and our group showed there is a correlation between the expression of CD45RC on T cells and rejection or not of kidney allografts. And this correlates also with the fact that if we use a CD45RC depleting antibody that will kill the high cells and not the low or negative ones where the Tregs are, we can induce graft survival tolerance. And in this example, we used a human skin rejection model in vivo in immune humanized NSG mice, in which we transplanted skin from a healthy volunteer 1, and 30 days later, when the skin was healed, not rejected, because these NSG mice are immunodeficient, we injected human PBMCs from another donor, and these PBMCs would reject the skin of the first donor during time. And we treated or not the recipients with an anti-CD45RC antibody, human one, depleting antibody, and with rapamycin at a suboptimal dose. And the survival of the skin grafts is depicted here. Of course, there is rejection without treatment, and with only rapamycin treatment, there's no significant prolongation, but with the treatment or with the antibody alone, and even more with combination antibody and rapamycin, there is protection of the graft. And similar results were obtained in solid organ transplant models, in acute GVHD, APSET, and autoimmune disease, rheumatoid arthritis and colitis models by treating with the anti-CD45RC. And we showed that in some of these models that tolerance after 30 days of treatment, for example here, was mediated by CD8 and CD4 Tregs that are induced afterwards. And we initiated a clinical trial with this strategy, a anti-human CD45RC depleting antibody by this startup that we create. Our interest on CD8 Tregs came from results some years ago now. Cardiac transplant model completely mismatched between two rat strains that were treated with a molecule which is CD40Ig, soluble CD40 that will block CD40 ligand, CD40 interactions. And you can see that in this case, almost all of the grafts go to indefinite survival, more than 120 days, whereas the control or non-treated the grafts were rejected. And we asked the question, which cells were mediating this effect? And to analyze this at day 120 after transplantation, tolerant recipients treated with CD40Ig, we harvested the spleen and transferred different cell populations into animals that were transplanted with the same the heart from the same strain as here, and that were only slightly irradiated, no further treatment. And we transfer CD45RC low, negative, or high CD8 T cells using this kind of cell sorting parameters. And you can see here the survival of the grafts was obtained when we used CD8, CD40FRC low or negative T cells from animals treated with CD40Ig, or when we used also these kind of cells coming from animals that were previously adoptively transferred with cells from the spleen of tolerant animals. And we did not obtain tolerance when we transfer CD45 high T cells from CD40Ig treated animals, and not even when we use cells CD45RC low negative coming from naive animals. So showing that these cells coming from the tolerant animals were more suppressive than the naive ones that were capable of protecting the graft. Excuse me. And this, I should point out that the only cells that were capable of transferring tolerance in this model were total splenocytes, total T cells, and CD8, CD45RC low negative cells. The CD40 regs were not capable of doing neither B cells or myeloid cells. Over the years, we have analyzed different mechanisms of CD8 regs by which they suppress or modify other cells, for example, IL-34, interferon gamma, fibroglobin 2, TGF-beta, etc. But I would like to refer to some recent results. And here we generated a genetically modified rat, a transgenic rat, using the CRISPR technology. We knock in EGFP in the 3' end of the FOXP3 gene in a way that the FOXP3 gene would not be affected. It's expressed normally, but the GFP is now under the transcriptional control of the FOXP3 mode. And in these animals, we observed that if we analyzed cells and CD8 cells, positive or negative cells, we observed there is a population of GFP positive cells, which in this case and in general is between 10 to 20% of the total FOXP3 positive cells, and which correlates with the percentage of cells detected using anti-FOXP3 antibodies. And the expression of GFP is really limited to T cells, because when we gate on TCR negative cells, all the cells are negative. And this is a lymph node, but the same is true for blood and other lymphoid organs. So we went on analyzing the phenotype of the CD8 FOXP3 Tregs in these animals compared to the CD4 FOXP3. We used CD25 and CD27 to begin with, and we observed, sorry, that the FOXP3 positive cells were present the CD27 negative cells and CD25 high, whereas there were no GFP positive in the CD25 negative cells. The same was true for the CD4 Tregs. And we correlated this with the CD45RC marker, and we observed that most of the GFP positive cells were present in the low or negative fraction of CD80 cells and in the negative fraction for the CD4 Tregs. We also did RNA-seq analysis of both populations of cells, CD8 positive, GFP negative versus the GFP positive ones. And here we focused our attention on the high expression of cell membrane markers in one population or in the other one as a way to identify new cell membrane markers for CD8 Tregs. And in this way, we could, we confirmed in this analysis that most of the GFP positive cells are in the CD25 high fraction of cells. But for example, for CD314, this is NKG2D, most of the GFP positive cells are in the negative fraction. And the same is true for CXCR3. Most of the GFP positive are in the negative fraction. Few cells are in the positive one. And this is also the case for CXR6. We also analyze the combination markers. For example, here I'm showing you the combination of CD25 where 6CR3 low negative. So if we only use the negative cells for CXR3, we don't get good enrichment of the GFP positive cells. But if we analyze in this cell population the CD25 high, we enrich the cells very much. Importantly, also, we can see a significant difference in between the enrichment that we obtain only with CD25 if we combine with CXCR3. And these cell populations, these ones are suppressive, confirming the suppressive capacity of these cells. So CD8 Tregs, FOXP3 positive Tregs in the rat are more abundant if we use CD25 high and CXR3 negative, similar with the use of CD314 or CXR6. And we are now analyzing in human CD8 FOXP3 the expression of these new markers. I can tell you that with CD314 is different, and there are some similarities with the chemokine receptors. And we like to switch to human CD8 Tregs. So just to show you the kind of suppressive analysis that we do, we use allogenic APCs, for example, from donors 2, 3, or 4. And then we use CD8 Tregs or CD4 Tregs from donor 1. And we analyze the suppression of proliferation of effector CD4 T cells, for example, or CD8 T cells. And we can clearly show that the suppressive capacity is in the low negative cell fraction and not in the high. And if we compare CD8 to CD4 Tregs, they have similar suppressive capacity. And using single-cell RNA-seq of CD45RC cells, we can clearly identify the high conventional ones from the low negative Tregs and also from mite cells, which are present in the low negative CD45RC fractional cells. When we do the methylation of the FOXP3 locus of FOXP3 positive CD8s or CD4s, we observe here in blue are the profile of the CD4 Tregs, very much unmethylated, and in black of the CD8 FOXP3 positive, and the difference is not statistic. And all here, which are statistically different, are the CD45RC high, as well as the CD4 or CD8 FOXP3 negative ones. And although they have similar methylation in resting Tregs, there is a difference of expression in 253 genes. But when the Tregs are stimulated, there are no differences in expression of genes. So we generated a method that allows us to amplify the cells to several hundred times using mainly polyclonal method. We compare to allogenic APCs, but the one that we use is using anti-CD3, anti-CD28, and low doses of IL-2, IL-15, and rapamycin. And the Treg isolation begins with isolating the CD8, CD45 RC low cells. And then after 14 or 20 days, 8 days of expansion, we can test them in vitro, in vivo, and we use in vivo this model that I explained before of human skin, allogenic human skin, or also a acute GVHD model. We developed a GMP-compatible GMP protocol using the Clinimax system, and then a FACS Taito cell sorter also from Milteney to isolate the CD56 negative cells, CD8 positive, and CD45 RC low negative cells. And we did validation runs in our university hospital GMP facility, and the runs were okay. And then we moved to this really GMP protocol that I will not describe in detail, but basically it's these low doses of IL-2, IL-15, and rapamycin, and anti-CD3 and anti-CD28. And we compared the cells obtained with this protocol from healthy volunteers in gray to patients with chronic graft failure, and the reserve capacity was the same, and also the yields. And the cells were resistant to immunosuppressants that are used in kidney transplantation, and they did not cause any toxic effect in NSG mice in terms of behavior or growth, which is not the case if one injects allogenic effector cells. So we generated this clinical trial that has been accepted, and one patient out of 9 included, in which we are an autologous therapy, first in human, open clinical trial, prospective, in which we will graft end-stage renal failure patients with a kidney from a living donor. And here we are substituting the induction treatment, which in transplantation is generally ATG or anti-CD25 antibodies. And we will give to the patients in a 3 design these different doses of cells, 1 for 8 million kilo, with classical maintenance, immunosuppressive, and tapering, and in-depth immunomonitoring with a follow-up of 13 months, and kidney biopsies, graft biopsies at 1 and 3 months. So the primary objective, of course, is safety, and the primary endpoint, toxicity, grade 3 or at M3, and the secondary objectives are long-term safety, immune reactivity, and efficacy criteria depending on the doses of immunosuppressors that the patients will receive. And of course, we will try to get as much information that we can using high-dimensional cytometry, single-cell RNA-seq, and spatial transcriptomics. I will then now move to the last part of my talk with results of genetically modified CD8 Tregs. Basically, the use of CAR Tregs and of allogeneic off-the-shelf cells. And I want to point out this recent review, very good one, on the clinical development of allogeneic CAR alpha-beta cells, which describes most of the clinical trials, advances, technical tips, etc. We published this some years ago, 2019, while time passes by fast, using CD8 Tregs with a R that recognizes HLA-A2, and we use this in the context of skin transplantation and GVHD. So the idea is that we are grafting skin from an HLA-A2 donor, and we use PBMCs from an HLA-2 negative volunteer, and this CD8 Tregs and lentivirally transduced to have express them a CAR, chimeric antigen receptor, directed against A2. So the Tregs, most of the Tregs will get activated against the donor antigen, whereas when we're using the natural Tregs, it's only a small fraction that recognizes the alloantigen. And we did get protection in this model of human skin rejection. And also in a model of GVHD, in which the model is we inject PBMCs from a human volunteer, HLA-A2, and then the CD8 Tregs come from another individual, A2 negative, expressing the CAR, and we obtain also protection. Concerning the off-the-shelf allogeneic cellular immunotherapies, until now, most of the work has been using autologous therapy, both in any T-cell therapy protocol, including CAR T-cells in cancer, but also for Tregs. And there are several problems linked to this. Sometimes there are not enough Tregs due to treatment, for example, in this case. Sometimes there are potential intrinsic Treg defects, genetic ones. It's an expensive and slow to produce protocol, and there is risk of failure of production. Whereas allogeneic therapy would be ready to use, faster, cheaper, and normal Tregs. One would take cells from a normal donor, amplify them, freeze them, and they're ready to be used in allogeneic therapy. But of course, this is linked to problems, mainly the fact that if they are allogeneic, they might be immunogenic. And we analyze this by incubating conventional T cells, CD4 or CD8, labeled with CFC-E, with expanded CD8 Tregs or APCs as positive control, one or the other one, and we look then to by cytometry to CFSC dilution for proliferation. Interestingly, when we see a different ratio of responder to simulators, so 4:1, this is a low ratio of stimulator, let's say Tregs in this case, in blue versus the APCs, we see that there is an induction of proliferation. And then this proliferation, when we increase the amount of Tregs, decreases probably because the Tregs themselves are inhibiting the CD4 Tregs, whereas with APCs, this continues to increase. So clearly, the CD8 Tregs, which express class 1 antigens, of course, stimulate CD8 T cells, are allogeneic. What happens with class 2 antigens? They also express class 2 antigens, these cells, and a similar effect, although less marked. At low ratios of Tregs, there is some activation and then decreases, whereas with increases quantity of APCs, this continues to increase. So the allogeneic Tregs are immunogenic at a certain level. So we aim to eliminate the expression of class 1 and class 2 by using CRISPR-Cas9 technology, electroporating RNA complexes, and using the protocol that I explained before. And in the absence of knockout, all cells express class 1 and class 2, and after using simultaneously guided RNAs for beta-2 microglobulin, which, as you know, is the chain that goes with all the classical class 1 and non-classical class 1 MHC antigens, and C2TA for class 2, we have that most of the cells are class 1 negative. Some of them are also class 2 negative. There are still cells that are positive for both. So we isolated the class HLA negative cells, and we compared to the HLA positive cells, and both behaved similarly in terms of suppression of CD4 and CD8 cell responses and proliferated comparably. So the idea was then to to compare these cells that were knocked out for class 1 and class 2 to the ones that have the HLA antigens, and in the same kind of, you can see in red that we have abolished the immunogenicity linked to the allogenic CD8 Tregs. So this works, but this comes with the problem of also making them, the cells which are knocked out for HLA1, susceptible to lysis by NK cells. See, this is an NK cell assay, and you can see that the beta-2 microglobulin knockout cells are sensitive much more than the wild-type cells. So we used a strategy that has been previously published by other groups, in which we eliminated beta-2 microglobulin, but then we bring an HLA-E beta-2 microglobulin chain construct with lentiviral vectors, aiming to have HLA-E interact with NKG2A and NK cells, which is a NKG2A a suppressive receptor. And the result was that we did reduce the cytotoxicity of NK cells, but it was not abolished. So there are potential solutions to these that other groups have used, for example, knocking out only HLA-A and B and not C, which is less polymorphic, or only doing downregulation and not knockout. There are other groups, very recent nice paper in Nature using NEF from HIV, and also, for example, depletion of NK cells in recipients. That would be another possibility. This is my last slide, I think, and it's just to highlight the fact that there have been tremendous advances on T-cell therapy in autoimmune diseases using CAR T cells directed against T-cell antigens, which was pioneered by the German group of Georg Schett, in which using CAR CD19 effector T cells, several autoimmune diseases have a spectacular improvement, particularly lupus. And very recently also, this was all with autologous cells from the patient. But now recently, this paper describing the use of allogeneic cells in which class 1 was deleted, etc., and expressing vector T cells, CD19. So allogeneic from normal donors derived, and with very spectacular results in was only 3 patients, but in severe conditions. So these are my acknowledgements, particularly to Severine Desi and Sonia Sal for the human CD8 Treg, and Severine Menoret for the rat CD8 Tregs. And thank you for your attention.
Thank you very much, Dr. Anegon. We have time for 1 or 2 quick questions if anybody in the audience has one. No? Okay.
Hi, Ignacio. This is Bob.
Hi, Bob.
So I have a very nice presentation, and I think it's very interesting to finally see rat Tregs come out because of the impact it could have to human disease testing and that. But I have a question. In the mouse and in the human, CD39 and CD15S, that carbohydrate receptor, have been used to kind of put a foundation on stability versus lack of stability of the Treg. So if they were 39 positive and 15S positive, they're very stable Tregs. But the lacking of the 15S puts it in that non-stable side. So, but what's very interesting is that 15S hasn't really been described at all in the rat Tregs. Is there a reason?
Well, I don't know, Bob. Frankly, I'm not sure that CD15 is available in the rat system. And frankly, I think we tested in the human CD8 Tregs and we didn't find it expressed. And for CD39, I'm sure there is no available antibody in the rat system. This is a short, problem in the rat system. So, but I think it's interesting that what you point out, and maybe we could look at transcript levels, maybe.
Okay. And if there's not another question, I have one more. So when you look at the diversity of the immune, let's say, repertoire of the receptor repertoire on cells, it is vastly different between people, right? So when you do your expansions in Treg for an allo versus auto, the reflective, kind of receptor expression that you have on allos is in many ways a really different match. There are so many different types of receptors on those cells. How do you explain that you would want that over auto other than saving money?
Well, I think it's an important and difficult question. I'm not sure how to answer this, Bob. Of course, it's important to bear this in mind. But at the same time, I think the allogeneic therapy comes with the possibility of genetically modifying. So I really think that the future of Treg therapy, is on first off-the-shelf allogeneic, and then using CARs whenever this is possible. And the reality and life will tell whether this is really better than the natural Tregs, but the in vivo data using a humanized model, it's not real life, really shows this. And now there is a clinical trial ongoing by Quell, this company in UK, using CD4 CAR Tregs anti-HLA-A2 in liver transplantation, and we'll see.
I think that's it.
Okay. Thank you very much.
Thank you. Thank you very much, Dr. Anagon. So we'll now... Okay, so we now invite you to a little break. We will have 10-minute break. We have a coffee break outside this room, and we'll see you back in 10 minutes. Nos vemos en 10 minutos. Hello, welcome back. Okay, so to start session 2, we welcome Dr. Robert Valderas, I'm sorry, Vice President of BD Biological Sciences from San Jose, United States of America.
Okay. So once again, good morning, and this is a very special part of this day because it represents an instrument that is extremely different than any other instrument that's ever been built, in our field. So when we look at biology, it's not only about an instrument. It's about all the tools that we use, right, to accomplish or to answer a question. And there's a few things that we know. That all of the colors that we use in our flow cytometers are not good colors. There's also something else that's important. There are no biological standards in our field. So just think about those two things. No standards, and many of the colors are not very good. So through the history of this field, looking at different dimensions and resolutions and different things have always been challenges. And this timeline reflects many, many things that have been done over time. But recently, in the last few, there's been a few areas where a lot of attention has been brought together to address not just bringing in a new instrument, but utilizing the spectral technologies, not just for biology, but to be able to help us understand more about the dyes themselves or the fluorochromes. So this story about, yeah, it's better if it's dark. This story about what we do with dimensional biology had its roots because in flow cytometry, we were stuck at 18 colors for many years. No more colors came through, and the sophistication of the tools and the instruments, they just didn't advance. But Gary Nolan and I wrote a paper in 2011 with a technology called CyTOF. It was a technology that we worked really hard on to be able to use lanthanides rather than colors. And we advanced the number of lanthanides up to 34. So we went from 18 levels of detection to 34 with lanthanides. But it didn't take long. A couple years later, we developed a flow cytometer in this high parameter conventional space when the spectral wasn't there yet. And then soon after that, we moved into the fields where we looked at RNA-seq, and then this new spectral instruments with imaging came out. But in order to get a better biology, we need to do a couple extra things too. One was advancements in the technology of flow, but the other was in the advancements of dyes. And this chart and this figure represents something very interesting. It's something that tracks the number and the number of the dyes that have been developed over time. But what it doesn't say is which of those dyes are good and which of those dyes aren't. And that's a challenge for us all, because companies like BD and Thermo and BioLegend and many of these, we make lots of reagents, but it doesn't mean that they're all really good reagents, because you need to match the color to be able to detect the appropriate biology. And we're going to take a look at that right here. So for everyone that's here, this is the way that we look at memory markers for T cells, right? Our effector memory cells, our TMRAs, our central memories. And the only plot that is correct here is this one right here. Okay. So you can see that it required us to have very bright dyes. But if we used medium bright dyes or dim dyes, you can see what happens to the resolution of the biology of memory cells. So you can see that these reagents are available for everybody to purchase, but then they don't tell you the correct story. So the quality of the color matched to the biology is absolutely a necessity in terms of what we do every day. So you can take a look at it like this. If we were looking at a primary marker like a CD4, right, that has a complete negative and a positive, then if we had a bright dye or a dim dye, if we look down below, we can see that separation without trouble. But if we go to something like a secondary marker like CD45RA or RO, you can see that with the bright dyes, we pull out a population right in here. But we lose that if we go over to the dim dye. So it either goes into the positive or it goes into the negative channel. So again, just a watch out when we're working in flow that colors really matter to the appropriate biology. So because of that, there was a whole bunch of work in BD to change something. And for everyone in here who's run a flow cytometer, when PE or a tandem is used on a yellow-green laser, it spills into a blue laser 44%, and then you take care of it with compensation, right? So this is a real problem in our field. So the chemist in BD began to develop some new reagents that had a really separate blue and a yellow-green laser line, so that something on the yellow-green laser doesn't spill into the blue, and a color on the blue line doesn't spill into the yellow-green side. And they built 14 different dyes to tell this story. So this is a friend of everyone. This is the emission profile, let's say for PE size 7. It has one major peak in the yellow-green side and one smaller peak in the blue. Now, if we would put that together with a brand new dye called RB780 that's in the same space but on a blue laser line, you can see what happens, that the PE size 7 spills into the RB780. And that's a real problem to see that type of spread. So to show you and demonstrate the quality of new colors that are being devised, we took PE size 7 out. Now you can see that the contribution right here, this spread is due to this little peak right here, okay? Now look, if we build a completely new PE size 7, we take that peak almost completely out and then look over here at the spread as compared to this spread. So building new colors really matters in our field. Now there's other things that matter in our field, and that's things like the impact of a buffer to a color. So if you were studying phosphorylation, we use methanol buffers to do the perm step following a fixation. So you can see here that if we use PE, or if we use PE CF594, and if we treat those with no methanol, we're able to get a very nice signal. But what happens when we use methanol, we lose the signal completely, right? So this shows you an instability of colors based upon a buffer. So if we move this forward then, these real blue and real yellow colors, if we look here for PD1, we don't lose any color here, and we don't lose it over here for PD1 on the RB613 line. So you can see where advancements in color to make sure that colors are not impacted by different types of buffers is also an important attribute of designing colors for the future. And if we'd like to do it a little bit more, you can take a look here where we have no fix in perm. So this would be no methanol that's in here. So we can see that in both cases with RY586 and PE, everything looks good. But then if we use the fix and perm with methanol in it, we lose that whole population right here as compared with that new real yellow 586 dye. And then likewise, we do the same thing by looking at a different one, the RY 586 on PD1 here versus the PE. So the lessons here again are chemists are developing new colors, and these new colors are better and better all the time. Now, another thing that everybody should pay attention to is photostability of your colors. So leaving your dyes out on the table where light can look at them. So if we look at like activated PBMCs for 3 days with PHA, and then we stain those with CD4 or with CD19 with these two colors, you can see that everything looks really good on the line. But what happens if we go to a Starbright reagent from another organization, you can see the impact of the Starbright, where this is standard, and this is where it would be without light, and you can see that we lose the CD25 completely. So colors that are very stable, such as PEC Cy5, you can see again the stability is there, just like the RB670 dye. And we can repeat that over by looking at completely other ones with the RB670. You can see this on the CD25, and we get a complete compression in here. And then if we go back over here to the PE Cy5, everything looks good. So one of the other things to always look at when you're doing your flow is, are the colors photostable? And then we can move to say, like, what are the best dyes? So what we've done is this slide compressed a little bit, but if you look here, dyes that have a single peak when they're hit with a laser line across all the different lasers are really good dyes. But if we look at this one right here, that when this color is hit with all 5 lasers, it creates a peak in every single laser in every detector on at least in one detector on every single laser line. That's an example of Percy P-Sci 5.5. Okay, it's also a reminder it's not a very good dye. Now we bring that together with what we do with color. So very dim dyes to very bright dyes. And when we take all those together and bring them together in a chart, this chart is something everyone should take a picture of, because all the colors that are in the yellow and on the green are the best dyes to use in flow cytometry. So if you have a 4, an 8, a 10, a 12, a 20, and maybe a 25 color, there are 25 colors on that side that you should use over using anything in the 3rd and the 4th columns. And if you just take those colors and make panels, imagine making panels in 5 or 10 minutes, because we know which dyes are the best. So what this says is brightness is on the x-axis here, is on the axis, and then this spillover is over here. So all of these dyes spill over a lot in these 2 columns. These dyes in these 2 columns don't spill over at all. And then these are really bright up here, and then these are less bright down on the bottom. So if we just take those and match them up very quickly, we can make panels that look like that. So each one of these panels uses only the dyes on the yellow and on the green side. So you can see how good they look and it just takes 5 minutes to make those if you use that chart, okay? And if you're on a box like the S8. So I thought I'd let everybody know what's coming in the future. So I'm sure individuals in here use the violet dyes and the UVs, the brilliant UVs and the brilliant violets, right? These are dyes that lots of people use, and they have a problem. The problem is they have dye-dye interactions between them, right? And you use a brilliant staining buffer inside if you've done. So Here's a great example of the new dyes. Here's a brand new real blue, the 800 dye. So you can see how good that dye looks. And here's another one on a real yellow. So these are two dyes that are way out here in the 8 and the 600 range. But let me show you something special. The chemist in BD are making all of the violet dyes from the Sirigen line, and they're changing all of them into this new type of dye. So this is the first one, which is called Real Violet 820, and all of the red dyes too are being transformed from the APCs and the Alexa floors into a brand new technology. So these are the kind of dyes that can be used through the collaboration that we work with here at the university. Early access to these dyes through the BD team will make these things available so that you can try them and use them. But in order to achieve better resolution, this is what we're after, right? If we look at this figure here, this 2x2, and compare that 2x2 with the one that's right here, you can see that the panel worked, but the biology is compromised. So learning about which dyes are the right ones to use and which ones are the ones that are not the best ones to use was in that chart. So if you didn't take the picture, you better get it from someone else. Okay? So let's just talk about something else here in the next 15 minutes, because this is where life is really important. So Alexis and team made a decision to bring a technology in here, and they worked very hard with our BD team here in Chile. And there's very few of these boxes in the world. And they are starting just to come out into the many institutions around the world, but you're lucky you have one here. So let's take a look at something for everyone in here. Let's assume something. Let's look at the bottom two here. Let's assume that this one right here in the center, this one, is a diffuse staining on a surface of a cell. So just very diffuse, right? Well, some receptors are not diffusely. Some of them are punctated like this. But let's assume there are 10,000 receptors for each one of them. Or let's just say you got 50,000 photoelectrons that have been seen, right? So they each would package and each would report out 50,000 photoelectrons. How about up here, if we looked inside the cell where we have one place where all the dye is, or maybe it's in 2 or 3 places, and let's assume they were all 50,000 photoelectrons. So if you did, in a conventional flow cytometer for the last 50 years, this is what happens. They look just like a histogram. So you can't discern the difference between the biology of those receptors, because all we do is count photons and photoelectrons and then put them in a histogram, a dot plot, or a contour map. What if you could look inside that cell and see that different biology? Just think about that for a second. Okay? So this instrument allows you to see cell-cell interactions. So here's another thing to think about. Anyone in here use DIVA on a BD instrument? Has anybody used DIVA? Probably a couple, right? Do you know what DIVA stands for? In the year 1997, an instrument was made moving from analog into digital. And we built a software called DIVA that stood for Digital Vantage. Okay? So what it gave us is a chance to do doublet discrimination. So what do? We find those doublets and we throw them away. Right? That's what we do, so that we go back to the singlets. Well, in our blood, we see things like this. There's synaptic cells that are there. There's also coincidental doublets, and I'm going to show you how how to look at those, in your work. Or imagine you have a viral infection. When a T cell is activated, it changes shape. And when it changes shape, it turns into a T cell from a round 7-micron round cell. It can turn into almost a droplet. And up at the top of the droplet where the pseudopodia is, it's where homing occurs in the cell. So your chemokine receptors are expressed there. Okay? So imagine having a flow cytometer that could find that in a sample. I'm going to show you a little bit more about what that is in just a few minutes. So let's just look at this instrument for a moment. This instrument has 5 great big lasers. Actually, they're 5 very small lasers. We don't have to use the big ones anymore. It splits the blue light in half, and half of it goes over to 3 imaging parameters, and the rest goes to flow. Okay, so it has PMTs, the old technology for image, and has APDs, or avalanche photodiodes, on the other side for all the flow cytometry. And this is what happens on the imaging side. The cell, the light goes through 104 beamlets, and then we vibrate a frequency over every one of those. And as the cell goes through, it creates a waveform. That waveform then, through a Fourier transformation, is turned into actual data that we can pixelate, and it's compressed from a Z-stack into the image of a cell. That's how that technology works, and it works at 3,000 to, let's say, 10,000 events per second. So here it is one more time. A cell goes through these 104, is characterized by all these beamlets. Each one of those little round circles has a unique frequency. That frequency then is collected, and it creates a waveform. And that waveform then, through a transforming algorithm, then turns it into a 2-dimensional electronic signal. So it's not optical. It's electrical. And then it's pixelated to be able to see it. And within that, we have many different ways that we can measure things in cells. So let's just say we look at the one that is maybe let's say we can look at eccentricity. So if we look inside the cell, and if something was in the nucleus, if we were looking at a nuclear stain, the nucleus might be nice and round. So we make 2 measurements across that round cell here in eccentricity, right here. We make a long axis and a short. And if it's a round circle, the ratio of those 2 would be 1. But as DNA, let's say you would go through cell division, and as you go through cell division, we get an elongation of the nucleus on 2 sides, right? So now the long axis has a large number versus a small axis, and then that's called high eccentricity. That's the way that the instrument measures at 3,000 to 10,000 events per second and gives you those measurements. It also gives us measurements here of diffusivity. So if it's very diffuse, or if it's very, very located in one region of the cell. So it uses those measurements there too. And then those are turned into all of these different kinds of images that we can track in a wall, in a cell wall, to be able to just pull all of that information out. So when we use doublet discrimination, 2 things happen. One, we lose cells in the doublet gate that are coincidental. So 2 cells that go through that are so close that the instrument can't determine if it's a singlet or a doublet, but it has a different shape. So because of it, it throws it out. The other one is that it's a real doublet. And if it's a real doublet here, you see that little yellow in the middle there? That's called coincidence. When 2 colors come together, they actually change a color. And this instrument can see that and measure it at 3,000 to 10,000 events a second. It can do all the measurements simultaneously in order to do that work. And I'm going to show you exactly how we can measure that. And this is an easy one. This is just translocation, right? So we have a protein out here in the cytoplasm. We have a color on our nucleus. We increase the concentration of TNF alpha and the protein starts to move into the nucleus. And when it gets all there, it turns it completely a different color called correlation. And then we know if the protein went through, the nuclear membrane. But in real flow, this is what we see down here. So the world's different now because we can actually see the image of specific cells. And then if you wanted to see something really big out of this instrument, this was the first 50 color paper that Florian Mayer did about a year ago. And in a week and a half or 2 weeks, when we go to Cyto in Denver, Colorado, Florian is going to show the first 60 color ever experiment on a big flow cytometer. So let's just, in the closing minutes, bring spectral flow together. So I shared this, right? It's just an image of a T cell and a monocyte together. But if we look at this 2x2 here, it looks a little different, right? So if we stain our cells over here with a T cell marker, and we stain over here with a monocyte marker, CD14, then what we see here is here's our T cells, here's our monocytes, here's our non-monocyte, there are probably B cells in here. But what is that? What's that double positive? Remember, there's no doublet discrimination here. So that in that box could be two cells coincident close together. It could be two cells stuck together, or it could be some magical cell like a CD4, like a CD3, CD14 that probably doesn't exist, right? But it could be there, right? So let's just take a moment on an experiment. I made a 20 color panel that allows me to be able to look at T cell subsets, B cell subsets, as well as monocytes, just so I can kind of look at what's in each lineage. All right, and I'm going to investigate a monocyte T cell interaction, and I'll look at a B cell T cell in the same. So let's say we stain the cells with that 20 color panel. All right. Then instead of doublet discrimination, we use two imaging parameters. One of them is called radial moment, and the other is called eccentricity. And when we do that, the cells in this area right here are predominantly singlets. And when you see this horn, that horn is predominantly doublets, but not pure, right? Because those populations move against each other. All right, so let's just take this CD14, CD3, the monocyte T cell, from this doublet gate right here. And when we do, we see the T cells, we see monocytes, we see B cells in here and DCs, and then there's that doublet. So if we take this gate and we use two other imaging parameters, one of them called delta center of mass and another one called radial moment or light loss, it's just then what we see is a wing, okay? That wing always shows up. So if we look at the cells up here at the top of the wing, you can see over here, they're coincidental. So they're apart from each other. If we look in the green gate here, those cells are stuck together. Now we could use an antibody to a synapse to be able to verify that and make sure, but the instrument pulls them out as two cells stuck together. All right. So let's go down here to our B cells because we stained our B cell markers were in there too. And here's that doublet of a T cell, B cell. We go through the same exercise. Here's the wing. All of these cells, it's easier to see over here because those are all coincidental doublets. And then everything in the red gate here sits here. And then you can see in some of those areas, like right there, the coincident change of the two colors of both of them coming together. So we know they're stuck together. We know they do. And the instrument reads that as an actual doublet. So let's just say you see some of those observations, because for 50 years in flow, we've thrown them out, right? We've never looked in those gates. So let's just take a moment. We stain those using that 20 color panel, and this is all of the gating strategy that was used that could find their T's and the B's and the monocytes, right? So imagine this question. Imagine asking this: is there any bias to which T cells and B cells bind together? Because T's, right? There's many different subsets of B's and T's, right? And there's different subsets of monocytes and T's too. So is there any bias? Are all people the same? Are they different in disease? Do they represent something that we've never really explored in our field before? I would say right now, no one knows unless they had seen this data and did the experiments. But I just took a look at one sample. And what I see is that between the T's and the B's, there were predominantly CD4s and not 8s. They were an IgM. If I look over here at the T cells and monocytes, there's multiple different types of interactions that occur, but they're very different. So the question you could ask using an instrument like this would be, are there differences in diseases? Are there differences in genders? Are there differences in age? All the kinds of questions that we look at functionally in many, many different types of things, but this is what you can do with that instrument that's sitting, in the institution here. It's a tool that allows you to think about biology in a different way. Just imagine there's only a few of them in the world, right? And one's here. So in closing, what I'm going to do is share this HIV story that I shared with you in that one slide. So there's a hospital research institute in Mexico City called CNA. There's a scientist there by the name of Gonzalo Soldado. And to share with you what the problem of HIV in Mexico City is, at that clinic, they do 1,000 CD4 counts a day, 7 days a week, 365 days a year. And it's the second largest HIV clinic, right? So it's a big problem. So I took the instrument down just to be able to let the team work with it. And I asked them, do you have any ideas about what you could do? So we know this, right, for all of us in terms of how viral replication occurs and how it goes through and then it comes out budding and exacerbates the big problems. If we took an EM picture of an activated T cell, this is that area up here in this distal neuropod that's here of where homing occurs. That's what EM looks like, okay? So if we did an experiment inside. The experiment we did is to compare the difference between a normal individual that's non-HIV, infected and an HIV infected individual. So not on drug, just came right through the clinic. So we took the uninfected cells and we kept a non-activated form of them. So we didn't do anything to them. We just took the blood. And then on the other side, to control it so that we had activation of the normal cells, we used PHA and IL-2 for those. So you can see, here's the normal cells activated, and then here is the HIVs. And we did this on multiple samples. So when you see this, you can see that they're certainly different when we use imaging parameters to look at those specific cells. This horn tells us a lot of things. So if you look inside that horn, that's what those cells are. Now, to verify that, the yellow is CD4. the brightest that you could ever met, because there's a down regulation of CD4 also when we have activation occurs. Those cells were sorted on the instrument that you have here. And then to verify that it was HIV, we stained with an antibody to P24, and that's what the green represents. It's quite interesting then, if we took many of those cells and looked at them, they don't all look like that shape, and there's a reason. When the cells go through, it only looks in 2 dimensions. So if the cell goes through like this, then it looks like that shape. If the cell goes through in that dimension, it will look more rounded. So this is the one thing different about this instrument, that if you sort your cells and then you do a post-sort check, the cells might look different because they go through the instrument in a different configuration. But you can see here that not all cells have the same amount of infection that's there. The protein changes over time. So I think what I'd like to do from this one perspective is to say thank you to everyone in here. There's a lot of individuals here in our business and all over the world that have contributed to this, that have worked hard to be able to bring the technology here and to make sure that the tool is here for everybody to use. There's a lot of training that has to be done on this over time. It's a different type of instrument to use. It's not just a flow cytometer, but it's an electronic imager. And the individuals that we have in our business as part of this collaboration, myself and others, will work with our team that's in the back room with Matthias and our team here to be able to really make sure that this instrument can be done and used at its highest capacity, to teach everyone how to use this and to share all of our knowledge to be able to make this available for everyone else. So for that, I'll stop here at that and see if there's any questions from anyone. But you're a very fortunate, let's say, team of individuals to have technology like this. Thank you very much.
I'm gonna turn the light on, so it might be a little bit comfortable. Do we have any?
So thank you very much, Bob. Very insightful talk. I love that you did a wide array of different thematics. So I don't want to sound spoiled, but now that you see that this shape that comes out because of this capacity of this equipment shows that we may have distinct shapes for cells in vivo. Could it be at some point, or are you already envisioning to have XYZ analysis of the shapes, or is it too early to determine whether such parameter is so important as to develop it in equipment?
Yeah, it's a really good question. The technology that was placed in here actually came from Jabali's laboratory at UCLA. It had been brought out by a scientist named Eric Diebold into a small little company. And myself and another scientist and a couple other ones saw it very early on, about 8 years ago. And about 6 months later, we purchased it because we thought we could work with it. And there was something about the technology that looked easy to us, right? That it was going to be just a drop-in into a flow cytometer. But it didn't. We thought it would take us 6 months to a year to do it. Turned out it took us 8 years to do it. So it wasn't easy. To answer your question about getting to the third dimension, where let's just say it's an optical image, I think the one part that holds us back is this, okay? So the size of those little files there are about 2 gigs each, right? And we keep them stacked in a Z stack so that we don't hold the images of all those cells that go through, right? Or it would just be a mess. So you can reconfigure and bring them back very, very quickly. But I think if you had 3-dimensional at that speed, we probably couldn't do it at 3,000 to 10,000 events a second. It'd probably be more like what an Amnis is. So the Amnis Technologies is very similar, but does optical imaging, but it's 300 cells a second. versus 3,000 to 10,000. So I think you have to give things up in order to get to that resolution. But if you take these images and go then to a sorted cell into a confocal scope, oh my gosh, you see the most beautiful cells ever. So it does work. It's just, you know, the magnification is 18x versus 60x.
Thank you very much.
Yeah.
So we have time for one more question.
Thank you, Bob, for your talk. It was a very nice one. I really enjoyed it. One experiment that I did once was to evaluate complement binding to bacteria, and we evaluated that by flow cytometry. So I was thinking at your image here, when complement binds to bacteria, bacterial shape changes, and you can see whether the bacteria is happy or is suffering when the complement binds. What's the level of resolution that these images can give us information? Do you think we could see through this image a bacteria suffering with complement binding or something like that? Because it's very small compared to a T cell.
So on my computer, I have that. I have macrophages taking up bacteria, right? So the one thing we need to do is have the color on the bacteria, right? So if you integrate a color into the bacteria, the resolution is a micron. Okay, at a micron, you can look in the cell and see 1, 2, 10, 20, and you can actually sort those out based upon those numbers. So I'm not sure what the change of what it would look like from being happy to unhappy, right? Or whatever, but you can certainly pick bacteria out of a cell that has, you know, optimized or you know, brought the bacteria inside a cell. You can see that.
Thank you. And then the other question I have is, do you have any limitation about the dyes you are using to your images? Because so you show a T cells with different colors, right?
Yes.
So is there any limitation or any strategy that we could pick our dyes to set an image that might look better or worse depending on how we plan the experiment?
Yeah, the colors on the imaging side are only on the blue line, or any color that may be used that spill into the blue line, right? Because we haven't corrected the compensation yet on there. So we do not have a second laser line that can do this work yet, right? Only a single laser line. But it's very, very clear that those new colors that are off the blue and the yellow-green line, those are so bright and so good that they are just the cleanest dyes ever in each of the imaging, you know, channels, 1, 2, or 3 for PE size 7, per CP. So those are those 3 channels that are in that space. I think, you know, over time it'll increase a laser line, probably the violet or the yellow green may be the 2 that go out there, because a lot of people, you know, need M cherry and things like that to be able to see on it. But yeah, I think all those changes will come over time. But it's a complicated system to make work, and there's a analytical instrument connected to this. The analytical instrument does a little bit different because we can shut off the image and we can run everything at 10, 20, 30, 40,000 events a second. On the sorter, you have to do everything at around, you know, 3,000 to 10,000. Now, and the last thing I'll leave you with, for anyone that's in here that works with small samples, right? Like a pediatric sample where you don't have very much blood, or maybe a FNA, a fine needle aspirate of a tumor where you might have 5 to 10,000 cells. Well, in every sorter in the world, there is something called an electronic abort. Okay, so if you ever taken your cells down to a sorter operator, and then you take 1 million in, and then they separate positive and negative, and then you have 600,000 left, that's usually, you lose those because of abort rates, electronic aborts. The instrument kicks them out because of pressure, speed, many things can contribute to it. This instrument has 0% electronic aborts. So if you run it at the right speed and you put 5,000 cells in, you get 5,000 events back. So for working with pediatrics or working with needle aspirates where there's very, very few cells, this instrument is just an amazing thing. So I think, again, you can be spoiled and enjoy this instrument, because I think it will open up avenues and it'll open up different ways of looking at the kind of science that you guys work with all the time. And my team is going to work with our BD Chile team to be able to help you do that here.
Thank you,
Thank you.
Dr. Valderas.
Maybe it's.
Okay, so to close the second session, we welcome Dr. Katina Schilin. I don't know how to pronounce your, sorry. Chinnerling. Thank you. Sorry, I got lost. Yes, assistant professor of the Faculty of Life Sciences from Universidad Andrés Bello. Sí, supongo.
Good afternoon. Thank you to the organizers for the invitation. It's a great pleasure to be here and to present our recent data. After this complex and really amazing technologies, I will tell you a very simple story with simple flow cytometry, but I hope it's still interesting to you. Well, in our lab, we are interested in the role of microbiota in the pathogenesis of rheumatoid arthritis. As most of you will know, it is proposed that it has been proposed that systemic inflammatory diseases have their origin in mucosas, and that the microbiota has a great participation in this process. So I don't want to bore you with a lot of mechanism, just to remind that there are many interactions between microbiota and the immune system, the epithelium in the gut mucosa. So we have microbia associated patterns which can activate immune cells. We have metabolites which can exert immunoregulation. And we also have the answer of the host cells, which can produce antimicrobial peptide, secretory IgA. So we have this interaction between microbiota and the host immune system. So we are particularly interested in automembrane vesicles. So just brief introduction. These vesicles are secreted from gram-negative bacteria during normal growth. And these vesicles contain, they're like a mini bacterium. Yeah, they contain parts of the outer membrane, obviously outer membrane proteins, but also periplasmic proteins, DNA, RNA, LPS, peptidoglycan, toxins, enzymes. And their function basically, or for a long time, has been seen for the interaction with other bacteria. Yeah, the microbial defense, horizontal gene transfer, acquisition of nutrients, formation of biofilm. But for us, it's interesting, this interaction with the host immune system. So these vesicles also have been shown to deliver virulence factors or toxins to host cells, and they can also modulate immune responses. That's what we are interested in. I just want to show you some examples because we are interested in this only, because it has been shown they can trigger or potentially trigger systemic diseases. In this manuscript, the researchers show that if they orally give automembrane vesicles of Helicobacter pylori, these vesicles can travel to the brain and can induce neurodegradation. So there's also another paper which shows that automembrane vesicles from Bacteroides theta iota omicron can travel to the liver, and it's also they might also cause liver disease. And what we are most interested in, and it was a paper from 2023, the authors show that Fusobacterium nucleatum derived automembrane vesicles, which are orally or administered by oral gavage, can travel to the joints of the mice and also aggravate collagen-induced arthritis. And if you compare to vesicles which are derived from, sorry, I don't know how it works, but yeah, if you compare to vesicles from E. coli, you can't observe the same effect. So what we can see in these, we have a lot of evidence that automembrane vesicles can translocate from the intestinal lumen to distant tissues, and they can contribute to systemic diseases. We are particularly interested in Segatella cobri, which is a pathobiont which is associated with rheumatoid arthritis. So formerly, this bacterium was known as Prevotella cobri, and it's a strictly anaerobic bacterium. And normally it's really abundant in the gut microbiota and related to a fiber-rich diet. And this is interesting because we consider it as pathobiont, but still it is related to a considered healthy diet. But there was a paper in 2013 which shows that this bacterium, or the genus of this bacterium, was overabundant in patients with new onset rheumatoid arthritis. And the same patients showed a reduction in bacteroidecy. And then authors tried to confirm this effect of the confirmed the arthritogenic effect of this bacterium in mouse models, though they used germ-free mice and also models of collagen-induced arthritis. And they can show that, or they showed that if they administered this bacterium orally, it aggravates the severity of the disease. And also if they use this bacterial strain isolated from rheumatoid arthritis patients, this strain is more arthritogenic than strains isolated from healthy donors. So to come back to the automembrane vesicles, we ask if these automembrane vesicles from this bacterium, which have not been described before, might promote the activation of inflammatory immune responses and also contribute to the pathogenesis of rheumatoid arthritis. So this is a very broad and complex question. So we tried to put it down to like three objective or three questions which are more simple to answer in short time. So we first wanted to know if these automembrane vesicles can be internalized by host cells such as epithelial cells or innate immune cells, and also if these vesicles are able to activate the innate immune cells and can promote a pro-inflammatory cytokine profile, which is also associated with rheumatoid arthritis. So first of all, we isolated vesicles by ultracentrifugation, filtration, and we got heterogeneous vesicles of size between 47 and more or less 200 nanometer. And we saw that they are spherical by transmission electron microscopy. And we also confirmed that they have a characteristical protein pattern by STF page. And now come back to the flow cytometry, which is the scope of the conference today. We can also, we are also able to stain these vesicles. Yeah, we have a nice cytometer, which is Fax Symphony A1, which permits or which has small particle detector. So we can detect particles until 90 nanometer or a bit less in size. So we can also differentiate between particles which are stained with a lipophilic dye, which is DIO, or with a dye which stains proteins, which is CFSC. And we can see that all the particles can be stained, and we can also separate from the unstained particles. Now, this is important for the following experiments. So first of all, we wanted to know if these outer membrane vesicles can be internalized by epithelial cells. So we used CACO2 cell line, which is colon adenocarcinoma cell line, and we stimulated the cells with labeled outer membrane vesicles. And then we track the internalization by flow cytometry using the small particle detector. And as you can see, this internalization has a really slow kinetics. So after 4 or 6 hours, we can detect some internalization, and after 12 hours, we first can detect an internalization which is more significant. So we can choose this, or we choose this time point to confirm by confocal microscopy, and we can see that really the outer membrane vesicles are internalized by these CACO2 cells. We can see it inside the cells, which are stained in red, the membrane, and the nuclei in blue. We also confirmed this internalization of these vesicles in antigen-presenting cells, which are our cells of interest. And we confirmed this in THP1-derived macrophages. Yeah, THP1 is a monocytic cell line. You can differentiate into macrophages by adding PMA. And we also confirmed it in macrophages and dendritic cells derived from peripheral blood monocytes. So as you can see in the pictures, the vesicles are inside the cells. Then we ask again if how long does it take for the internalization of these vesicles in macrophages? And we used again the THP1-derived macrophages, and we exposed them to the vesicles for different time points. And we could see after 3 hours, we get nice internalization. But then we were not sure if really all vesicles are inside, because by flow cytometry, we can just detect the signal which is associated to the cell, but we don't know if it's really inside. It's not like confocal microscopy, because we don't have like this newest technology. So we use the trypan blue staining, which quenches all the fluorescent signal which is outside the cell associated to the membranes. So we can just subtract all the signal which is outside or associated with membranes and not inside the macrophages. And we see there's a reduction in the signal, so there's some vesicles associated to the membrane. And now we can get this graph where the solid line is the real internalization, the outer membrane vesicles which are inside the cell, and we have the dotted line with the vesicles which are the total vesicles which are detected associated with the cells. Then we wanted to know what is the mechanism of internalization in the macrophages. So we used inhibitors. We used cytokalasin D to block micropinocytosis and sucrose to block clathrin-mediated endocytosis. And as you can see here in the graph, the cytokalasin D really blocked internalization of these vesicles, while sucrose only had a slow or a low or slight effect. So we can conclude that the outer membrane vesicles of Segatella cobri are internalized mainly by macropinocytosis and to a lesser extent also to clathrin-mediated mechanism. So we had also a very motivated undergraduate student, Alison, who asked if these vesicles can activate macrophages. So she did a lot of culture of human macrophages derived from peripheral blood monocytes. And she cultured the cells in the presence of MCSF for 8 days. And then used this flow cytometry panel to characterize M1 and M2 related markers. So what she observed is that markers related to M1 profile, like co-stimulatory molecules, are upregulated when the macrophages are stimulated with outer membrane vesicles derived from Segatella cobri, but not when they just stimulated with the bacterium, the whole bacterium. Yeah. So this is really interesting that the whole bacterium does not really stimulate the cells, but the outer membrane vesicles do. And she also observed slight reduction of M2-related markers, which was significant for CD163. So we also wanted to confirm the cytokine secretion profile of these macrophages in response to these vesicles. And Alison observed, but this is not cytometry, this is ELISA, sorry. But she observed that also the outer membrane vesicles induce the production of pro-inflammatory cytokines like TNF-alpha, but also IL-10, which is normal because it's like a counter-regulatory mechanism. When cells are activated, they start producing also IL-10. So we can conclude from this that the macrophages stimulated with outer membrane vesicles adopt the M1-like profile, but not if they are stimulated with the bacterium. Then we wanted to know, okay, we have this with naive macrophages, but what happens if we use macrophages which are already polarized towards M1 or M2? So Alison generated M1 macrophages by culture with GM-CSF, and then she activated for the last 2 days with LPS and interferon gamma. And she could also observe a further increase of M1-related markers and also of pro-inflammatory cytokines, which are also related to the induction of Th17 phenotype in T cells, like IL-23, IL-6. So we can conclude that these outer membrane vesicles further strengthen the M1 phenotype and further increase the activation and cytokine production. So what happens to M2 macrophages? So we can generate M2 macrophages by stimulation with IL-4. And these macrophages normally are more anti-inflammatory. But if we stimulate them with the outer membrane vesicle of Segatella cobri, we can also get an increase of M1-related markers and also of at least one pro-inflammatory cytokine, which is IL-6. So we conclude that these outer membrane vesicles are able to repolarize M2 macrophages towards a more M1 profile. So this was the data for macrophages, but we have more recent data regarding to dendritic cells. So Sabrina, in her master thesis, she is interested in what is happening with dendritic cells. So she is generating dendritic cells from peripheral blood monocytes in the presence of GM-CSF and IL-4 for 4 days, and then she just selects, she uses this flow cytometry panel, and she selects like the CD11C, CD1C positive cells, which are CD14 negative, and then she observed that there is also a slight increase of co-stimulatory molecules when these dendritic cells are stimulated with outer membrane vesicles. This is quite preliminary data. I know it's just N3, so maybe we are not reaching significant level, but we can say that at least the maturation marker CD83 is significantly increased if we stimulate with outer membrane vesicles with a higher concentration. To be a bit closer to what is the situation, maybe also in vivo, which is difficult because we work with human samples. We tried to do a whole blood stimulation. So we used whole blood samples, stimulated for 6 hours with the outer membrane vesicles or the bacterium or LPS, like a positive control. And then we can just get on dendritic cells using a lineage HLA-DR panel, and then we can separate monocytes from dendritic cells. And we can see that after this stimulation with the outer membrane vesicles, we get an increase in maturation and co-stimulatory markers. So this is the graph. So we also can observe this not only for CD86 and CD83, but also for CCL7 and HLA-DR. And again, this effect is not observed when we use a bacterium. And we can also, in this whole blood sample, we can also detect cytokines. So we just have to add peripheral DNA for the last 4 hours of stimulation, and then we can nicely see production of IL-12. And we also can observe that if we stimulate with the vesicles, we get an increase in IL-12 production. And we can also get this increase for TNF-alpha, IL-6. And again, we only see it when we stimulate with vesicles, but not with the bacterium. So we propose as our model that Segatella cobri is producing outer membrane vesicles. These vesicles stimulate innate immune cells like macrophages and dendritic cells. And these activation, increase of pro-inflammatory cytokines might also have an impact in vivo. So might also activate Th1, Th17 T cells. And these cells can translocate to the joint. And maybe also the vesicles, which is according to the literature, it's really probable that the vesicles are also able to translocate to the joint, and they induce inflammation. So we have a lot of open questions and some things we really want to do. Not only we have to characterize the vesicles, because we did a proteomics analysis, but many of the proteins are not described yet. So we have to just go a bit deeper in which protein or which lipid can have a functional consequence. Probably LPS, but it's not only LPS. And we also want to do characterization of the response to these vesicles by other cells like epithelial cells, neutrophils, synoviocytes, and osteoclasts. And if it's possible, obviously would be nice to do it with a multi-parametric approach. So we also have clinical samples like feces and novel fluid saliva, and we also want to characterize vesicles, so eukaryotic, but also bacterial vesicles. So this would be also challenge for the future. And of course, we are trying to establish mouse model to check if these vesicles have also an impact in vivo on the development of arthritis. So I want to thank my group. So in the picture, well, always someone is missing, but we took it recently. So I thank all the students who worked on this project, also my collaborators from Universidad Andrés Bello, from Catholic University, Universidad de los Andes, Charité Berlin, and also, of course, I need for financial support. Thank you.
Thank you very much, Dr. Schienerling. I hope I said it correctly this time. Do anybody has any questions?
Thank you very much, Katina, for the talk. Quick question. Have you looked at the targeting of the proteins or peptides that are on the vesicles to MAC class 1, class 2, CD1 in terms of presentation or to T cells or Tregs?
No, we haven't done yet, but it's something we want to do. Yeah, we are thinking about proteomics approach just to see which proteins from the vesicles can be presented and also activate T cells. There's one protein which has been described for Segatella cobri, and patients, rheumatoid arthritis patients respond to this protein, and we also find this, we found this protein in the vesicles when we did the proteomics research. But we have to confirm it, and we also have to perform like co-culture experiments with T cells. So this is things we want to do in the future. Thank you.
So we have one more question.
Thank you very much. So I want to ask whether you think the similar results may be observed when these vesicles are opsonized, or you have a projection of studying what happens when they're covered with antibodies and maybe extract from sera, or because you still have TLRs maybe that may be exposed, so you may have similar effects, or also depending on how they're processed through FC receptors, you may get similar or different responses.
Thank you. That's a really interesting question. No, we did not check yet, but it's something we discussed in our group also, because the vesicles are like mini bacteria, but some kind of more immunogenic. And but I think they can be also target of antibodies, of complement maybe. So yeah, it's worth to check it, but we haven't done it yet. Thank you.
I see somebody has one more question. So quickly, please.
Thank you for your presentation. It was very interesting. I was wondering, could be OMBs derived from different bacteria like Lactobacillus present a contrary effect, like inducing a more anti-inflammatory phenotype, or has someone reported something like that?
Yes. This is very important. I forgot to mention it, because why we have to study these vesicles, because they are so different from one bacteria to another. Even between the same bacterial strain, if you culture it under different conditions, or if it's derived from a patient versus a healthy person, it can be quite different in its potential to activate immune responses or induce immunoregulation. So it's very important to define the conditions where you obtain the vesicles and how you perform the experiments. So yeah, this is indeed a really big question. And you have, for instance, the vesicles from Bacteroides theta iota omicron are thought to be more immunoregulatory, but there are also papers showing that it's related to sepsis. So they can really have contradictory effects.
Thank you very much. Okay, so we will now take a break and we'll meet back here at 1:30. Thank you. Ah, perdón. Ahora vamos a un break y nos volvemos a encontrar acá a la 1:30. Eso. Okay, welcome back. Sí. Okay, so to start the next session, we welcome Dr. Eva Acosta, full professor of the Faculty of Chemical Sciences in the National University of Córdoba, Argentina.
So good afternoon, everyone. Thank you. I'm really appreciate to participate in this wonderful workshop, and I would like to thank to Dr. Kalergis for the invitation. So in my talk today, it will be divided in two parts. In the first part, I will share with you our experience in building a centralized flow cytometry facility. And in the last part, I will show you some results of my own group in which we use this facility for the analysis of regulatory T cells in infectious models. So I would like to start by telling you that I work at the CBC, which is the acronym for the Spanish name of our institute, Centro de Investigaciones en Bioquímica Clínica e Inmunología, which is a multidisciplinary research institute that is located in Córdoba at the center of Argentina. It has more or less 30 research labs that do research in the area of life sciences, and particularly immunology is one of the biggest area of interest. And our institute has a very dynamic environment because most of the groups are led by international trained scientists. So we struggle very heavily to conduct high quality science in a place like many countries in Latin America is resource constraining. So for this, because of this, from the very beginning of our institute, we decided to follow the strategy of using centralized facilities for all the large equipment. So we have this is the facility flow cytometry core, but we have a centralized platform for many other equipment, including microscopy, cell culture, animal facility, molecular biology, etc. And why we decided to do this? Because, as you may imagine, a centralized platform optimize equipment use and reduce the downtime. We can somehow focus the dedicated technical staff to support in the maintenance and training of researchers and students. This shared infrastructure lower the cost and improve sustainability, and also, and not less important, promotes the collaboration among the different groups and data standardization across labs. That it's very important now that the robustness of the research is a matter that it's very important. So in a few slides, I'm going to tell you that at the moment we have these 4 equipment in our laboratory. The oldest one is a FACSCAN 2. We have also an AttuneNext with an auto sampler that allow for the auto sampling of many experiment, and this is very important for drug discovery laboratories. We also have a Fortesa and our FACS area that is a cell sorter. So all this equipment works under a service cost that is covered by the every group, and the cost it's around $10 to $12 the hour, and this is probably much more expensive than running this equipment in other laboratories, probably in the States and in Europe. And this is because we have this Latin America aggregated cost of the shipments of all the stuff. This facility have a dedicated technical staff. So at the moment there are 3 people working on it. 2 of them are completely focused on the flow cytometry platform, and the third one is shared with the microscopy platform. All of them hold a PhD degree in biological sciences or immunology, and the 3 of them as well hold a permanent position as technician from the CONICET, and this is it's very important for us because somehow ensure the long-term knowledge retention and training capacity. And the three work together to ensure the maintenance and the user training and support for the student and the researchers. So they are the core of our facility, the most important people in the facility. So another very important part of our facility is that they belong to the national system for large equipment. And for me, it was a very smart strategy that was developed by the federal government of Argentina. That was the creation of this national system. So this is the web page, the official web page. The first started in 2008 with the microscopy national system, and currently includes 8 different types of equipment. So here we have flow cytometry, but also, as I say, microscopy, X-ray, high performance computing, among others. And these national programs work also with the same idea of networking and joining forces. So the idea is that they connect the major facilities of equipment in the whole country in Argentina to ensure open and coordinated access. And that's what they look for it, because most of this equipment were bought with public investment, that is with public funds. So somehow this network allow an efficient use of this large scale equipment and this investment of the federal grants. So how do you can access to this national system? The institutions have to register the institution as well each equipment, and they have to commit 20% of the time of the equipment for the community. So they are open to the community, and there are a booking system that is online and open to every user in the whole country. If you register and participate in the system, then you are eligible to apply for specific funding opportunities for upgrades in your equipment, to buy complementary tools, access to platform, and also to training. So I believe that this kind of platform at local, regional, and even international level could be an important source of collaboration. So as the participation in this system requires that you book your equipment use in the booking system, it's very easy to have a statistic across all the equipment in the country. And this is in our institution. This is Centro C2, so we were one of the first one to adhere to this program. And here in this graph, we observe the number of hours in the orange line per month since April 2016 until now. And then in the blue bars, the number of different users. And here you see that it was pretty stable, around 200 and 280 hours per month at the very beginning, before the pandemics, and around 150, 80 users, different users per month. And then here in this valley, it is the pandemic that everything was shut down, and then it recovered, but we are still not at the same level that before the pandemics. And particularly in the last year in Argentina, we observed a decrease. That is, we will talk about this a little bit later. It is because the severe funding cuts that we are having since the last administration change. Here in the right, we have the percentage of use according to the different institutions. So our institute allows for or use around 90% of the time. And then we have another users from the same university, other users from different universities, and also we have around 1% of industry or private companies. In this other statistic graph, we see the usage in comparison with all the different equipment that are registered in the program of flow cytometry. So in the blue bars are our 4 equipment. So we are the first in number of distinct users. Our fax canto is the equipment in the country that has most different users, and also our other equipments are pretty in the top. And this other graph is the cumulative usage of all the equipment that are registered in the system. So you see again, the Canto 2 is the 3rd one, and the Forteza is the 4th one in the whole country. These other 2 lines are from equipment based in Buenos Aires, that is the capital city. So somehow this clearly demonstrates that our equipments in our facility are a reference for the flow cytometry platform in the inner part of our country. So it is really a reference for all the research institute working in the inner part of Argentina. So, but we have been struggling very heavily to be able to somehow upgrade our capabilities, but we haven't been successful. However, at the moment, we do have a spectral cytometer that is a Cytec Northern Lines that is in a temporary base, because it was provided by the company by agreement, a 2-month loan agreement. We have been waiting a lot for this and are applying to different funds. But as I said, in Argentina, there are freeze in the national research funding. So this is the way we've found to be able to have this equipment. And so the equipment arrived around 2 weeks ago, and the ramp-up period was very brief. At the moment, all the students are very eager to use it, and it is under a very intensive use. In fact, we are trying to negotiate with the company to be able to retain the equipment at least a couple of months more. So we are convinced that this kind of centralized platform, as I said, share not only a local level, but also a regional level. It could be very important in areas as Latin America, in which we time to time have a very strong restraining in our condition, in our funding, because it allows to optimize the funding for equipment upgrades and acquisition. If you increase your capabilities, so you are much more competitive to obtain international grants that you can also use to support the facilities. In fact, in our institute, we have been able to secure 3 NIH grant and 2 GSK collaboration. So the thesis for doctoral training and also recruitment of early career is much more attractive, and can lead to an boost in international publication. In fact, in the last year, we have had more of 120 international publication based on the use of these technologies. But of course, in these resources constraining countries, time to time, we have like a new administration that we have to discuss with them why research is important. And then we have like freeze in different programs that somehow halt the science and the possibility to maintain or to upgrade our technology. At this moment in Argentina, all the research grants are free, so researchers are not getting the grants they were awarded, so the people cannot afford the fees. And somehow this delay the access to new technologies, and somehow compromise the long-term planning of our research. So this is the situation now. However, as I said, we in our institute we have a really very driven atmosphere, and we work very hard to try to do the highest research possible to and to continue our research with the highest standard. So now I will start the second part of my talk, in which I want to share with you some of the results that we can obtain using these and other facilities in our institute. And our research is aimed at deciphering the role of regulatory T cells along acute and chronic trypanosoma cruzi infection. As you probably know, Chagas disease, it's an endemic disease with relevance in the Americas. It's caused by the infection with the protozoan parasite Trypanosoma cruzi, and two phases can be easily distinguished. In the acute phase, there is a really high blood parasitemia and a strong pro-inflammatory response with a Th1 bias, high production of interferon gamma and CD8 cytotoxic responses that leads to the control of the parasite in blood, and also a moderate although transient tissue damage. And we have shown that during this period, the regulatory response is somehow limited. And after the parasite is controlled and many years after the acute infection, the patient develop a chronic phase in which the parasite is clear from the blood but persists in different tissues such as the heart, the skeletal muscles, and the gut. And in these tissues, there is a chronic low-intensity inflammation that leads to fibrosis and progressive organ damage. And the regulatory response during this period, it's pretty poorly delineated. So despite the important role of studying this disease due to the clinical relevances in our region, we also believe that this infection, it's a powerful model to dissect immune response adaptation throughout different stages of the infection. So let me start by showing some data that we published a couple of years ago. For that, we use FOXP3 GFP reported mice that allowed us to evaluate the course of the regulatory T cell responses along the time of the infection. So here we detect this Treg response that, as you can see here in this dot plot, the magnitude of this response is reduced throughout the acute phase of the infection, and the lowest frequency of this cell subset is found around the 20 post-infection, that is the peak of the parasitemia and the inflammatory response during the acute phase. When we analyze the phenotype of these guys here, and this is a star plot that is summarizing the very deep phenotype analysis, we detected that although they are reduced in magnitude, these cells are activated as shown by the upregulation in the red line of different typical Treg markers such as CD25, CTLA4, LEG3, and CD39. In fact, CD39 is one of the regulatory molecules that is earlier upregulated in the Treg. So remember this for later. And also we detected that not only these cells got activated during the infection, but also they upregulated genes typically associated with Th1 responses such as CXCR3, TBET, and interferon gamma production. And this Th1 polarization of these regulatory T cells here was also determined by a transcriptomic analysis that shown a clear upregulation of many genes related to Th1. So the Treg during the acute phase of infection acquire a Th1 specialization. So you probably know that Treg came in different flavors. This is called a specialization of the Tregs, and in this case we were showing that these Treg during T cruzi infection are specialized in suppressing Th1 responses. However, we were very also much interested in evaluating another specialization of the regulatory T cells that are the tissue repair regulatory T cells that this is a subset of regulatory T cell that is characterized by the co-expression of ST2, that is a receptor for IL-33, and KLRG1. These cells are particularly specialized in maintaining a tissue homeostasis and promoting tissue repair after damage. So in these plots here, in non-infected mice, you are able to detect this subset of cell in skeletal muscle, liver, as well as in spleen. And below in this plot, it is this subset was much less reduced in infected mice in all the organ evaluated. And this reduction in this subset of regulatory T cells somehow correlated with a significant decrease in the level of IL-33, that is the growth factor for this subset. And also, this reduction of tissue repair Treg correlated with an increase in the damage in different tissues. So this is an histological analysis of skeletal muscle in non-infected mice. The architecture is conserved, while in infected mice, there is a strong infiltration, but also cell damage as indicated by the necrosis and calcification of the fibers. So far I've shown you that during the acute phase, regulatory T cell responses are reduced in magnitude, but they are activated and they acquire an specialization in Th1 suppression, and while at the same time they have a reduction in tissue repair subsets. So we wondered whether it was the real effect of these cells. So to determine that, we used the Derek mice. Derek mice have the diphtheria toxin receptor under the control of the FOXP3 promoter. So if you inject a diphtheria toxin, you are able to specifically deplete of Tregs. So we infected these mice after 5 and 6 days, depleted of regulatory T cells, and analyzed the effect of this Treg depletion after many days after infection. So in this kinetic plot here, we evaluated the frequency of CD8 T cells specific for the parasite, and in the blue lines we showed that the depletion of Tregs increased significantly the frequency of CD8 T cells specific for the parasite, and this was also true for the spleen and liver. And this was at an endpoint of 20 days, so at the peak of the infection. And this increase in the CD8, effector CD8 T cell response impacted in the parasite control. In this plot here, we have the blood parasitemia. And again, we determined that the depletion of regulatory T cells allowed a better control of the parasite. And this was true not only for blood parasitemia, but also for the presence of the parasite in tissues such as heart and liver. So this Th1 specialized activated Treg, although not in high numbers, they were enough to suppress and to delay an effector CD8 T cell response and the control of the parasite. So at the same time, we wonder what happens if we are able to improve the presence of a regulatory T cell subset that will collaborate in preventing tissue damage. So we decided to test this by supplementing IL-33 in infected mice, and we did it at very early time points to avoid a high inflammatory response that somehow prevented the effect of IL-33. And shown in this dot plot here, the supplementation of IL-33 increased the frequency of cells with the tissue repair phenotype. And this was true not only for skeletal muscle, but also for liver and spleen. And afterwards, we determine whether this increase in tissue repair direct correlated with changes in the progression of the infection. So in this heat map, we are summarizing several different infection markers, such as the activity of enzymes like LDH or transaminases or other enzyme, or hypoglycemia, or lost weight, or the ability to keep hang in a hang test. So these during the infection are pretty much increase, and the treatment of with IL-33 reduce all these marker of disease, indicating that this increase in regulatory T cells with tissue repair function correlated with an improvement in the conditions of damage. So all the data I have shown you so far, it's from the acute phase of the infection. So we did focus on the chronic phase of the infection. And the first experiment were devoted to evaluate the features of the characteristic of the T cell responses. So here in this dot plot, it is shown the frequency of regulatory T cells in non-infected mice in the spleen and skeletal muscles in comparison to chronically infected mice. In this plot, you can observe that the frequency of regulatory T cells is still reduced in chronically infected mice versus non-infected mice in the spleen. In contrast, these regulatory T cells accumulate in target peripheral tissues such as skeletal muscle, and this is the statistic evaluation. So then we decided to analyze the transcriptome of these guys here in the skeletal muscle in comparison to these guys here from the spleen. And in this volcano plot, show that there are many differentially expressed genes between these two populations of Tregs. And when we compare these transcriptomic signatures with reported signatures associated with tissue repair or with Th1 specialization, we determined that the Tregs from chronically infected mice, the skeletal muscles Treg from chronically infected mice, have enriched expression of upregulated genes in tissue repair Tregs and downregulated genes in tissue repair Tregs. And also they show a Th1 signature because they have upregulated the genes that should be upregulated when they are specialized in Th1 suppression, and they regulated the corresponding gene. So these regulatory T cells from skeletal muscle from tissue from chronically infected mice have dual transcriptomic signature. They are specialized in Th1 suppression, and they are also able to perform tissue repair. So then we decided to evaluate the real effect, and we performed systemic versus local depletion. In this slide, I'm just showing you the results from the local depletion. And for that, we titrated very carefully the amount of diphtheria toxin that we have to inject in each of the muscles of the legs to deplete of regulatory T cells at local level with a minimal effect at systemic level. So this is the result. Once we injected diphtheria toxin in the muscles, at 7 days after injection, we see a significant decrease in the local number of regulatory T cells that is recovered after 2 weeks. And this is not only at the level of total regulatory T cells, but also of Treg with a tissue repair phenotype. And somehow, as expected, it correlated with changes in the damage as evaluated by histology. So this are the histology of skeletal muscle of chronically infected mice, in which you can observe certain inflammation and some degree of lipid or necrosis and calcification. And this damage, it's increased after the depletion of regulatory T cells with higher, particularly higher calcification and necrosis. However, to our surprise, the depletion of the depletion of regulatory T cells in muscle not only led to an increase in damage, but also we detected an reduction in the number of specific CD8 T cells in the muscle. As you can see here at day 7, there is a significant reduction of parasite-specific CD8 T cells, and this correlated with an increase in tissue parasitism that was more significantly after 2 weeks of depletion. So somehow these Treg in the muscle that have a dual signature of tissue repair, but also Th1, are not only preventing damage, but also sustaining local effector tissue responses. So this, we are finishing this manuscript, and we will submit for publication anytime soon. So with this, I arrived to one of my last slides, in which I'm just going to show you how all this data that I have shown you, the published one and the one we are finishing, somehow support this conceptual model of regulatory T cell function across the infection phases. We propose that during the acute infection, we have an activated Treg pool that is quite heterogenic. The main population is a regulatory T cell pool specialized in the suppression of Th1 responses that through CD39 mediating mechanism, it's able to suppress or to delay parasite-specific CD8 T cell responses and somehow delay parasite control that lead to tissue damage. Also, there is a reduction in regulatory T cell with tissue repair attribute, and I didn't have time to show you, but it's published in our paper. This correlates with the enhanced inflammation mediated by interferon gamma. Once the effector response, it's able to control, not clear, but control the parasite and the infection progresses to the acute, the chronic phase, there is an adaptation of the immune response, including an adaptation of the regulatory T cell response, and then the Tregs in the lymphoid organ system or the circulation somehow come back to a normal phenotype. However, there is an accumulation of regulatory T cells in the tissues, particularly in those tissues that are target of the infection. And these cells that accumulate, these Tregs that accumulate in tissues show a dual phenotype of Th1-like and tissue repair Treg, and they are involved in both phenomenon. Not only they prevent tissue damage, but also they sustain effector CD8 T cell responses that are required to control the parasite burden. And so somehow this showed that the biological relevance of regulatory T cells change from a deleterious role during the acute phase towards a protective role during the chronic phase of the stage. And this can be applied to our understanding of how to treat the human diseases. With that, I would like to finish with thanking and recognizing, acknowledging all my team. So the people in blue are the ones that were more involved in the result that I have shown you, and the different collaborators and supporting agencies. Thank you very much.
Thank you very much, Dr. Acosta. Are there any questions from the audience?
Thank you, Eva. Alexis Carragher is here. So First, thank you for willing to be part of this workshop. It's been a great day of immunology and flow cytometry. But I have a question on the second part of your talk. Did you check if your Tregs or your effector T cells are specific for the cruci antigens? Did you do any antigen-specific staining or priming in vitro?
So this is a very good question. We haven't been able to, for the CD8 T cell responses, yeah, we continuously use by using specific tetramers or by activating them with a specific an immunodominant peptide that is TSKB20, we always analyze both the total CD8 T cell pool as well as the parasite-specific T cells. This is not the same for regulatory T cells because for cruci, there are not really good tetramers developed for CD4 cells. So we have been able to track parasite-specific CD4 T cells, and these include Tregs. We have done some experiments by activating the Treg with parasite lysates, but we were not really convinced about the results. So at the moment, we don't know exactly whether all the results that we are observing are really requires of parasite-specific regulatory T cells or they are bulk T cells. So we don't know. Yeah.
We have another question.
Hey, Eva, thank you for your talk. I have also a question for the second part of your talk. So one specific cell type that is very important in chronic infections in different models are myeloid-derived suppressor cells. And some people have found that the influence of these cells are actually sometimes equally important as Tregs, and in some models they are actually more important than Tregs to allow this chronic infection establishment. And I wanted to ask you if you have studied the neutrophils and monocytes population during chronic infections. Do they have a worse, for example, antimicrobial capacity? What's the profile of these cells in your chronic infection model?
Yeah, thank you for the question. Yeah, we have done a very deep phenotypification of the different cell types that somehow are recruited or infiltrate the muscle, because we are pretty much focused to the muscle. And there are not really a lot of myeloid-derived cells there. So we did our best to try to define them, but we haven't been very successful. And since when we deplete, we don't see really much changes in myeloid subsets, we haven't gone deeper than that. We do see, I didn't have time to show you, but as we are working with IL-33, we also check routinely ILC2s, and we see really interested changes and possibly crosstalk between Tregs and ILC2 that we are trying to go deep in that, but not with myelocytes. Yeah.
Thank you very much, Dr. Acosta.
Thank you.
We now welcome Dr. Stephen Portelli, Professor and Chair of the Department of Microbiology and Immunology, Scientific Director of the Einstein Flow Cytometry Core Facility at the Albert Einstein College of Medicine in the United States of America.
Hello, everyone. I hope you can hear me, and can you see the screen okay?
I'm sorry, Doctor, we can hear you well, but we do not see the screen in presentation mode.
Well,
Now we see it well. Thank you.
Great. Okay, thank you. Well, thank you very much for having me today. And I must say, I'm very impressed with the organization of the meeting. It's really something to hear from people essentially all over the globe in one meeting. So congratulations to the organizers on that feat. What I'd like to talk about today is the experience that we've had here at the Albert Einstein College of Medicine with incorporating spectral flow cytometry into our core facility to enable many different projects. Although I won't be talking about very many of them today, I want to give you a little bit of background on the spectral approach. It's a bit redundant with some of the things you've heard earlier today, but hopefully will help to reinforce the major concepts. And it'll show you some samples of work that's been done in our facility with this approach. So when I first entered into this field and became very involved with flow cytometry here at Einstein about 20 years ago, there were many limitations. Although flow cytometry was already a very powerful and popular technique, it was clear that we were running up against limitations even at that time. And I would say only about 7 or 8 years ago or so, I was beginning to think that maybe fluorescence-based flow cytometry has already passed its prime and is going to be gradually replaced by some of the other techniques that had started to come along, especially things like mass cytometry with CyTOF, which was really expanding the number of parameters that could be analyzed per cell, and some of the sequencing-based techniques like CytSeq, which substituted oligonucleotides and sequencing technology for the fluorescence technology. So that was a little bit depressing, but actually things have really turned around in the last few years. And this is because of these several innovations, which I've listed on the slide here. And one is the dramatic increase in the number of fluorophores that are suitable for flow cytometry applications. We've heard a little bit about this earlier today, and I'll go over this again, but when I started flow cytometry, we really were limited just a handful of useful fluorochromes, and that grew up to the level of maybe a couple of dozen, but then it got stuck there for a while. But just in the last 3 or 4 years, we've seen a real expansion in the chemistry of the fluorophores and the availability of suitable fluorophores. And the second major innovation is the availability of spectral detection instruments. The spectral approach to fluorophore detection, multi-parameter analysis is not particularly new. I think the idea has probably been around for a couple of decades, but it didn't really get incorporated into readily available and user-friendly instruments until just the last 7 or 8 years. And this has allowed us to really take advantage of this explosion of new fluorophores and the increase in the number of markers and antibodies specific for those markers that are now available. And a third thing is the development of computational and software tools that have enabled us to better visualize the data coming from high-dimensional data sets like 20, 30, or 40 parameter flow cytometry studies, and tools for analysis of these data. And all of this then has led to a lot of effort to couple flow cytometry, in fact, sorting to other single-cell analysis techniques. Here, it's mostly been single-cell RNA-seq, but single-cell proteomics and other types of single-cell analyses are coming along. So all of these things together really revolutionized flow cytometry, I would say, just in the last 5 or 6 years. Now, in terms of the fluorophores, you can divide most fluorophores, possibly all of them that are used routinely in flow cytometry, into these 5 groups. And all of them have seen significant development in the last few years. The synthetic small molecules are the most familiar, and the original fluorochromes developed for flow cytometry. And the chemists have delivered many, many new versions of these, which have altered properties which are useful for flow cytometry. Then we have the fluorescent proteins, which include the translation genetically expressed fluorescent proteins such as GFP, YFP, etc., and variants of those, and the phycobility proteins, which have been around for a long time but remain useful, like APC, PE, PRCP. And these are further modified by the creation of the tandem dyes, in which a donor and a acceptor from fluorochrome are coupled together. This expands the wavelength range for individual lasers, which is very useful in building larger panels of fluorescent antibodies. And finally, you've got the quantum dots, and the newest are the polymer dyes, like the BD Horizon series. So all of these together have led to this remarkable expansion of fluorescent probes. This is one example of a sort of pileup of all of the probes which are currently considered to be useful for flow cytometry, or at least preferred for flow cytometry. There are 114 of them on this graph. It's a little bit too large for you to be able to read the font, I'm afraid, but take my word for it. You have in there probably all the ones you're familiar with, and then a lot of other ones which you may not be. The color of the bars relates to the laser wavelength, which gives them peak excitation, and they're arranged, the bar height is arranged according to the stain index, which is a composite measure of the intensity of the dye and its specificity. So you can see you've got here many dyes, and they're distributed over all 5 of the lasers, which are commonly used in commercial flow cytometers. And they have a range of different intensities, which is very useful when it comes to designing flow cytometry multiplex panels. I think it was mentioned earlier that you don't want to put, for example, extremely bright dye on an extremely high expressed marker. That will cause problems with the spillover effects. So you can, by carefully selecting from this large panel, you can now create optimized panels for 20, 30, and potentially even higher numbers of parameters per cell. So we got stuck at the 14 color level for many years. And that was because we didn't have spectral flow cytometers. We only had conventional compensation-based flow cytometers. The workhorse instrument for our facility for a long time was the BD LSR2. We had several of those, which could achieve up to 14 color analysis using the really innovative at the time arrangement of detectors and light paths in the trigon and the octagon arrangements developed by BD Biosciences. Now, these were great instruments. We really got a lot of use out of them for many years, but we were really running up against this barrier that we needed more parameters per cell to really be able to take the flow cytometry analysis to another level. And we broke through that barrier first in 2019 when we acquired our first multispectral Cytec Aurora analytical flow cytometer. This instrument, you can see in the picture, is a fairly compact benchtop type instrument. Everything is contained within that fairly small footprint box. The instrument that we obtained was configured with 5 lasers and 64 detectors spread over those 5 lasers. This instrument immediately caught on in our facility. This was one of the best investments I think we ever made. The transition from the conventional flow cytometers to the Cytec Aurora was so quick that we almost immediately had to figure out how to get more of these in order to satisfy the backlog of users that we acquired. So in 2020, we got a second instrument, which was identical to the first. And in 2023, we added a third. And we are now looking into adding a fourth. So having these instruments essentially all identical is really the only way that we can satisfy the demand for this technology here at this institute right now. So that was very satisfying. Now, what these instruments do, and I'm sure that a lot of this is very familiar to you already, but I'll just go over it very briefly. Instead of the approach of conventional flow cytometers where you're directing the fluorescence emissions from a single laser path through a series of filters and mirrors to a single detector that's tuned to a particular wavelength band. These instruments excite each event passing through the flow cell with all 5 lasers and collect emissions from each laser. And the number of detectors associated with each laser can range from 8 in the red channels, for example, up to 16 in the violet or UV channels. This gives you, you collect the data from all of these detectors off of each laser for each event and store it. And this gives you a spectral pattern or sort of spectral fingerprint for each fluorochrome that's detected on a given cell. Here you see a typical spectral pattern for one candidate fluorophore. This is PE Cy7, just picked as a representative example. And what you see is this kind of rainbow pattern, which represents the spectral emissions across all 5 lasers detected in all of the 64 channels associated with those lasers. Now, that's for one fluorochrome, and if you add another and another and another, up to 20 or more, what you're going to get then is a composite of this kind of spectral pattern, one laid on top of another. Now, that is rather confusing to the human brain, but mathematics and a clever mathematical algorithm can actually unmix these patterns and determine what fraction of the composite pattern represents each individual fluorochrome. And this can be done with high sensitivity, minimal spread and spillover, which enables analyses of cells that have 20, 30, or even potentially more fluorochromes bound to their surface or on their intracellular molecules. So just a little bit more about this kind of spectral display, since you see it a lot when you delve into the area of spectral flow cytometry. So you read this according to the wavelengths of the laser, going from the shortest wavelengths, UV, to the longest, red on the right. And then you have this, for each detector, you have this kind of rainbow box, which has red in center, and then is flanked by yellow, blue, and green bands. Now, what are you actually looking at here? Well, the best way to understand it is if you look off to the right of this figure, you see that each of those rainbow boxes actually represents a histogram, as if you were looking down on the histogram from above. So that the red is the area of the histogram that focuses closely to the median of the mean fluorescence intensities of the events. And then as it tails off, the colors become darker green and blue. So this is the way that these are usually displayed. I think that you can really focus just on the red and get the general idea. So with this particular spectral profile, what you're seeing is a fluorochrome that has peak channels in all 5 of the lasers, although the peak emission is in the violet laser channel. Now, these can be unmixed from other fluorochromes because there are unique features of the spectral pattern which can be detected and unmixed using the mathematical algorithm for the unmixing function. And here's an example of 2 fluorochromes which actually are both excited maximally off the same laser, the yellow-green laser. They have slightly different peak channels of emission, but these would be difficult to discriminate cleanly on conventional flow cytometers. However, when you have a spectral cytometer, you can see that by looking at the patterns with the other 4 lasers, there are significant differences, particularly like look at the red laser. So in the red, with the PE Cy7, you have a very clear peak signal, whereas with the PE Dazzle 594, there's essentially minimal signal from any of the red laser channels. So the unmixing functions are able to separate these 2 cleanly. And if you look at the patterns for the spectral emissions for essentially all of those 114 fluorochromes that I had shown on an earlier slide, those are all published in various places or accumulated on various websites. This is from the SciTech website, where they have shown all of the dyes that they have detected to have unique signatures in their 5-laser instrument. And there are 14 of them essentially at this time. So these are dyes, these are patterns which can be effectively unmixed using the unmixing algorithm associated with that cytometer. Some of them are easier to unmix than others and give less spread over and spill. So what you try to do is build panels which provide the least spectral overlap and which give the maximum signal intensity of emission on different laser paths. And with the large number of fluorochromes that are available now, it's pretty easy to do that if you're only looking at 20 colors. And as you get up to 30 or 40, it's still possible. It becomes a bit more challenging, requires more time for development of the panel. Okay, so building a panel of antibodies and fluorochromes for spectral detection is a challenge. There are a number of resources now available, and they're getting more developed all the time, which really make this challenge approachable for just about anybody now. The first part is selecting the fluorochromes, and there are tools available online at various sites, and all of the companies whose logos that I've shown below here have excellent websites with many resources, such as the fluorochrome selection software, which is available and in most cases is free to use. The other important thing is to understand something about which fluorochromes are best assigned to which markers, because if you have very highly expressed markers, you don't want to put your brightest fluorochromes there because you'll get more of the spectral spillover and spreading, which can complicate the data and lower the sensitivity of detecting specific populations. You want to reduce the spectral overlap of the fluorochromes that you use as much as possible, although you cannot completely eliminate it in most cases, but keeping it as minimal as possible will help to get clean unmixing. And then finally, you need special tools to be able to make some sense out of the data that you get and to visualize in a way that you can talk about it and think about it. We are pretty good as human beings at visualizing things in 2- and 3-dimensional space, but when we get above that, it becomes very abstract. And when you get up to 20 and 30 and 40 color analysis, you're dealing with a 20 to 40 color space essentially, with each of your fluorophores representing a different vector in space. We really can't imagine exactly what that space looks like and process the data very effectively. So we need the help of computers and mathematical algorithms. So for data visualization, which requires the process known as dimensional reduction, we need these mathematical algorithms for multi-parameter facts. So dimensional reduction means taking something from 20, 30, 40 dimensions and reducing it down to a number that we can easily think about. And in flow cytometry, that's usually 2 dimensions, although we probably could function with 3. But mostly it's 2 dimensions that we're looking at, various kinds of plots or output figures that are in 2 dimensions. And you've seen many of those already today. I'll show you more. To do this, the original most commonly used algorithm was the principal component analysis, PCA. This became extremely popular and useful when people started looking deeply into transcriptomics, and you're dealing with essentially a 20 or 30,000 dimensional space because each gene represents a dimension. And PCA was really the most useful way that people came up with to find a way of interpreting and thinking about data from such a complex data set. But PCA is not particularly useful in facts. In facts, we're not dealing with such high dimensions. We're usually in the 20 to 40 range or something like that. And 2 other mathematical algorithms, which are based on a slightly different baseline mathematical principles, have emerged as the most useful dimensional reduction approaches. And these are called the T-SNE and the UMAP approaches. And these 2 different algorithms have a lot in common, but there are also some significant differences, which I'll illustrate a little bit better as we move on here. But just to keep in mind that the T-SNE is particularly good at resolving the local structure of data sets. So things that cluster together are often very tightly put together with the T-SNE approach, whereas the UMAP, it also does that, but it also has the ability to preserve more of the global structure of the database, so spreading out more the unsimilar groups of things that are identified. And this can be quite useful actually in flow cytometry. And the other thing that's very important, that's different between the T-SNE and the UMAP, is that the T-SNE is more computational intensive, and it requires, depending on your computer processor, requires a fair amount of time to generate the output. Whereas the UMAP is a much more lean sort of mathematical approach, which is able to generate outputs much faster. And typically in our experience currently working with usually something on the order of 20 to 30 color analyses, the T-SNEs can take well over an hour to generate an output plot, whereas the UMAP usually can do it in a few minutes. And the reason why that's very important is because there are a few parameters within these programs which you can easily modify. And you can't always tell what the best output is going to be until you've played around with those parameters. And if you have to run the algorithm 5 or 6 times to find the best output, and it takes 3 hours to run, well, you're going to be sitting there most of the day doing that. Whereas with the UMAP, you can usually get through that process in less than an hour. So our users have almost entirely migrated their data analysis and visualization to the UMAP approach. And here's an example just comparing, this was a 23 color, I believe, analysis on bone marrow mononuclear cells carried out in our group. And the same data here has been put through both the T-SNE and the UMAP algorithms to generate these 2-dimensional outputs that we can look at, and we can select what we call clusters from the data. And there are software tools which help to do this in an unsupervised way. And with this T-SNE, what you see is 23 clusters, which have all been colored differently, so you can distinguish them easily. But they aren't all very well separated, and some of them actually touch. And you might even argue about whether some of those are even really independent clusters. Now, the same data, analyzed through the UMAP approach, also identified 23 clusters, the same 23 clusters probably, and colored with the same set of colors. But notice how much more separated many of these clusters are, and how much more distinct they are from each other. So this separation increases the resolution of your analysis, might allow you to generate more hypotheses about different subsets of cells. And as I mentioned, it's also much faster so that you can optimize the output in a reasonable amount of time with the UMAP. So I think this is why virtually everybody, at least in our facility, has taken this approach. The most commonly used analysis software in our groups is the FlowJo software package. And although they built in the T-SNE function early on in their release, they have subsequently added a UMAP plugin, which you can add. So that software will support this analysis. Now, let me show you just one example, which I think illustrates nicely the power and the value of the spectral approach. So this is a study that was done by Dr. Greg Laveau, who's a professor in our institute and works extensively with the Einstein FACS core facility to develop higher parameter, higher dimensional multispectral analyses. And in this study, Dr. Laveau and his colleagues are interested in looking at the subsets in peripheral blood of the leukocytes of patients who have the adult T-cell leukemia and lymphoma, or ATLL. This is a very serious T-cell blood cancer, and it's a virally induced cancer, which is seen in HTLV1 infected carriers. The HTLV1 has the ability to infect a subset of CD4 T cells and carries with it transforming genes, which leads to the production of this leukemia lymphoma in about 3 to 5% of all the HTLV carriers. So it's a fairly rare cancer, but it's very serious. And it's more common in the regions of the world that have high endemicity of HTLV1 infection. And that's what the map is showing you there. You can see that there are a number of countries which have a fairly high rate of HTLV endemic infection, 1 to 5%. And then there are also these hotspots in a few places in the world, which are the red asterisks, where there's very high rate of HTLV1 infection, and coincidentally, a relatively high rate, at least of ATLL. So it is a serious problem which doesn't currently have a really good therapy. In fact, the survival is probably the worst among all peripheral T-cell cancers with or without treatment. The combination treatment that is currently considered state-of-the-art, but it's probably not very effective, is zidovudine plus pegylated interferon alpha. So clearly, there's a need to understand better exactly what is the cellular lesion in this leukemia lymphoma, and what are the effective treatments for it. So to begin to get a better sense of what kind of subsets of T cells are present in the circulation of patients who have this syndrome, Dr. Laveau, with our facility technologists, designed a 28-color spectral flow panel for use on the Aurora. You can see the markers that are identified by the antibodies in the panel listed in the table on the left, and then the associated fluorophore with each of those markers. So this panel is selected to identify a lot of common T-cell subsetting antigens, but also a number of ones that haven't been looked at or really not extensively looked at in conditions like ATLL, including a number of transcription factors which control T-cell differentiation and function, as well as some molecules involved in T-cell suppression or exhaustion and immune checkpoints. Now, when you gate the lymphocytes according to standard approaches, such as shown in the top row of dot plots, one can select basically all of the leukocytes from the blood sample, and then you can display all 28 colors in the UMAP plot, which is shown here, and clearly segregates very cleanly the major subsets of leukocyte types. The B cells, the NK cells, the CD8 cells, and the CD4 cells and monocytes are all separated into clear clusters. An interesting thing, though, is this is a UMAP plot from the patients with ATLL, which differs from healthy controls in that large region colored in red that overlays the CD4 T cells. And that's because these are the expanded leukemic cells in the circulation of those patients. And we know that that part of the plot is enriched in the leukemic cells because many of those CD4 cells are CD7 negative. And although it's not a perfect marker for a transformed ATLL cell, it's one of the better ones that's available. Most CD4 T cells normally express moderate to high levels of CD7, and most leukemic ATLL cells in the circulation do not. And then you can look at the plot, the bar graph next to this plot, and you can see that there are some aberrations in the numbers of these subsets in the groups of healthy controls in the black bars and ATLL patients with the red bars. The major significant differences are in the reduction of B cells, the significantly increased level of CD4 T cells. I think I lost the significance marker there, but that's significant, and the reduction in CD8 cells. And this expansion of CD4 cells is mainly, if not entirely, due to the leukemic clones that are expanded in circulation. Now, with this analysis, one can go down deeper because you have 28 parameters that you can look at. You can gate on a population of interest. So one population of interest is the CD4 T cells. So here's the UMAP dimensional reduction plot on just the CD4 T cells, and overlaid in different colors here are the cells from the healthy donors, which are in the black contour plots. The ATLLs donor cells in both the blue and the red. And the ones shown in blue are presumably non-transformed circulating CD4 T cells. They express CD7, for example. And the ones colored in red are the transformed, or at least highly enriched, for the transformed ATLL leukemic cells. Now, once you have this, you can then go looking at each portion of this UMAP projection, and you can query the expression of each of the markers in your panel. And I showed 4 of the markers here. You can see with CD45RO, marker of memory, of T cell memory, that the ATLL cells are predominantly memory. You can see that the red color indicates high expression over that part of the UMAP. They also are highly proliferative, which isn't surprising since they're leukemic cells, which is shown by the Ki67 expression. And you can go on. There is expression of FOXP3 in the cells in that region of the projection. And there's also fairly strong expression of PD1. And we can keep going. There are many more markers to look at. I won't go through all of them, but here's a summary in more easily digested bar graph form based on that kind of analysis. You can see again this high level of memory markers like CD45RO and the high proliferative marker Ki67. It's also interesting, though, that PD1 is high on these cells, suggesting possibly some form of exhaustion or suppression. They also express the transcription factors FOXP3 and Helios. So this is a way of building a more extended phenotype for the leukemic transformed clone circulating in the blood of ATLL patients and distinguishing them from the untransformed cells, which are also there. This becomes actually important in monitoring the responses to treatment of these patients in trials to try to develop additional drug regimens to improve upon the one that we currently have. So by combining these markers, it's likely that we'll be able to improve on the ability to detect remissions or improvements in the disease. And that's one of the goals of this study now going forward. But there was one other thing that came out of this, which I think is very interesting and would not have been easily detected without the use of the high dimensional spectral panel and these tools for analysis. In the UMAP analysis of the cells gated for CD8, so all the CD8 T cells in either the healthy controls or in the ATLL patients, a unique subset of CD8 cells was detected only in the ATLL patients. So here, if you look at the overlay on the UMAP plot of the healthy control CD8 cells in the black and the ATLL cells in the red and also in this lavender color, the lavender colored cluster that's seen very clearly here is unique to ATLL, and there's nothing that segregates to that part of the UMAP projection in healthy control subjects. And this is quantitated in the graph shown to the right. So we don't detect cells of this type in healthy controls. And they're present in most or almost all of the ATLL patients and can be at quite high levels, up to more than 40% of all of the CD8 cells. So this was a striking finding. We think must have some significance to the immunological basis for what's going on in this disease. So we looked in more detail at what these cells are. So again, if you go back to the UMAP, it's the same one that you just saw, except it's now presented as a more of a heat map type coloring rather than a contour plot. And then you can lay over that the colors reflecting the level of expression of each of these 9 markers that are queried here from the panel. So again, you can see that the memory marker CD45RO is highly expressed in the region up here, where you have the unique subset of CD8 cells. The Ki67 is also there, so these are probably highly proliferative cells. They do express some level of granzyme B and perforin, so they maintained at least a modest level of cytolytic function potentially. And they have very high levels of some of the immune checkpoint negative stimulating molecules like PD1, TIGIT, and LAG3. This is all laid out in more detailed for each of the individual subsets in the scatter plots that are shown to the right. So there's a unique subset here. What is the significance? Well, we don't know what they do. One could say, well, they're simply exhausted cells. Maybe they have no relevance to the ability to control this leukemia, but we think that actually they may have some relevant function. And the reason that we think that is because of a second study that's been begun to look at another cohort of these patients. So here, another 12 patients with ATLL have been enrolled into a phase 1 treatment trial, which includes the histone deacetylase inhibitor belinostat to the standard therapy. And this is a very interesting approach because the HDAC inhibitor is thought to basically wake up some of the latent genes of the infecting virus. And this really builds on the same approach that's been taken for HIV therapy to try to increase the expression of HIV-encoded targets on the surface of infected cells so that they can be addressed by immunotherapies. So here, the same sort of thing was done. We treat these patients with the HDAC inhibitor and look for evidence of remission. Well, some of the patients do achieve remissions with this approach. First of all, you can see in the UMAP at the top left that in this cohort, we again identified this unique subset of CD8 cells, which segregates away from the CD8 cells in normal individuals and from the majority of CD8 cells in the ATLL, which are not infected with virus because this virus is tropic for the CD4 molecule on the CD4 T cells. Again, you can see the phenotype. If you go to that part of the UMAP projection and you focus down on specific markers, which are what the histograms are showing to the right, you can see again, like we saw previously, that there are memory cells with high CD45RO. They're highly proliferative with Ki67 expression, and they express some of the other molecules which are often associated with either exhaustion or suppression, like TOX, PD1, and TIGIT. Now, what do these cells really do? I mean, are they good? Are they bad? Do you want them? Do you want to get rid of them? Well, we're not sure, but when we look at the pattern of patient responses to this therapy so far, one thing that emerged as a strong correlation was that this unique population of CD8 cells is much more prevalent in the patients who responded, the 6 patients in this trial who showed clear clinical responses with remissions of their leukemia. And some of the patients were partial responders, and they had a lower level of these cells. And 3 of the patients had progressive disease and no impact of the HDAC inhibitor therapy, and they had the lowest level of these cells. And in this graph shown on the bottom right, you can see how the levels of these cells changed following the institution of the HDAC inhibitor at day 0. So you start out with an increased level of these unique cells in the patients who ultimately turned out to be complete responders. And that difference increases during the therapy, peaking up at day 21, and then at a later follow-up was found to have returned back down to the baseline level. So we really think at this moment that the best hypothesis is that these cells are participating in the control of the virally infected leukemic CD4 T cell clone, and that they're not cells that you necessarily want to try to target or eliminate or inhibit, but rather cells that you might want to try to augment to improve on the therapy of this leukemia. Now, the next step then, we think, to really understand those cells is to sort them out and then isolate and take the isolated cells and perform other deep analysis procedures, such as the single-cell RNA-seq and proteomics. Now, to do that, we're going to rely on spectral cell sorting, which thankfully is available now to us. We obtained the Thermo Fisher Bigfoot spectral cell sorter back in 2023. It took us about a year to really get up to speed with the use of this sorter, but now that we're familiar with it, we feel that this is an extremely efficient and powerful sorter. It's probably the fastest sorter that we've managed to actually put our hands on. It sorts up to, well, in theory, up to 60,000 events per second, but we can sort routinely in ranges of 20 to 30,000 events per second. And it's a spectral analyzer, although the optical configuration is not identically matched with the Cytek Aurora analyzers that we're using. This introduces a little bit of a challenge because when we take results from the Aurora analysis and then try to process them for sorting through the Bigfoot, we have to redesign the panels because the level of fluorescence and spillover are not identical between the two instruments. And this can be done, but it's an additional step, more labor and more time and expense. So to overcome this, what we've done is to acquire the Cytek Aurora CS, which is a cell sorter version of the Cytek Aurora analyzer. And this can be configured identically to the 5-laser Cytek Auroras that we're operating. It's also a pretty fast sorter, not as fast as the Bigfoot, but one can get up into reasonable levels of speed with this. And the really great thing about this is that the outputs are essentially identical to the outputs from the analyzers, so that any panel that we build and validate with our analyzers and any unique populations that we identify can then be sorted, analyzed and sorted with the same panel on the Aurora CS sorter and presumably isolated through the sorting approach. And this is just an example of how really similar the outputs of these two instruments are. So this was from actually a study done specifically to look at this by Joanne Lanigan and published with Cytek. What they show is that in a 40 color analysis of PBMCs, that the Cytek Aurora UMAP is identical, at least I cannot see the difference, from the Cytek Aurora cell sorter UMAP. So these reagents, same cells, are performing identically on the Aurora analyzer and the Aurora sorter. So this is where we stand at this point. I've given you just one example of many, actually, people trying to further subset various types of cells in cancers and various inflammatory diseases. We try to get down to greater detail to understanding how these subsets are interacting and influencing our ability to address the diseases, to diagnose them, to monitor therapy, and to develop new treatments. So I will stop there with just acknowledgement to our flow cytometry group. So this is the staff of our flow cytometry core. We have 2 faculty members, who are very experienced cytometrists. That's Jinghang Zhang in the center and Joey Zhang next to her. And then 4 technicians, all of whom have been extensively trained on the use of the multispectral instruments and are involved not just in the maintenance of those instruments, but also in helping investigators get used to their operation and to help them design the most optimal panels and reagent selections for their experiments. So I will end there and thank you all very much for your attention. And if there's time, I'd be happy to address any questions you have.
Thank you, Steve. Here's Alexis. Thank you for a great talk. And I have a quick question for you. So when you found this, when Gregor found this population of CD8 cells, first, how could you do something's going on? How can you actually prove if they are functional in terms of what could their function be? And also, since you have these new technologies and we start seeing new things, how quote unquote real do you think they are, and how much of a potential like deep sensitivity of the instrument could influence the appearance of these new subsets of cells?
Well, I think that they are real. I believe that the basis for the spectral discrimination and the clustering of the cells is pretty well accepted now, and it's being used all over the place with, seems very reliable results. So I don't have an issue with them being an artifact of the techniques. I think what we need to do is to rebuild the panel to create enough of a multi-parameter phenotype that we can subject the cells to sorting with. And then once we have sorted the cells, we should be able to get a much better insight on what their potential functions could be through single cell transcriptomics. So the other part of this was looking at their function. Well, what are you going to look at? Well, the first thing I would like to look at is the antigen specificity. So there are some well-defined potential CD8 epitopes for HTLV1, and presumably I would hypothesize that these cells are probably responding to HTLV1 viral antigens, and since they're CD8 T cells, most likely presented by MHC class 1. So building tetramer reagents, which have appropriate fluorochromes on them to incorporate into the multispectral panel, would give us the right approach to that. Potentially the other way one could do it would be with activation-induced markers. So that would require some in vitro stimulation of the cells with either viral peptides or some other antigen of interest, and then query the expression of activation-induced markers. CD40 ligand is a good one. CD69 seems to work for this too. And we could sort the cells out that way and then further characterize them. So there's a lot of things to try. I think we're going to do these things, but it's not clear, exactly how long it's going to take. There is some reagent development required every time you build a new panel. There's a lot of things available to work with now, but just figuring out which ones are the right ones takes a fair amount of time and some trial and error. So I hope that gets a little bit towards your question, if I understood it properly.
Thank you, Steve. I have a more technical question. It was a very nice presentation. When you compare the PCA, the UMAP, and the TSNE algorithms, so something that we do when we do TSNE is we downsample. So we do a step of downsampling because sometimes when you have complex samples, it's too hard to analyze them.
Yeah.
Do you still have to do that with the UMAP, and how is the potential resolution of UMAPs UMAP analysis compared to TSNE?
Well, I don't think you have to do that with UMAP. I think I haven't seen too many people doing that. Part of the reason is, it's so fast, even with a large full data set, that you can play around with the two parameters that you have that you can adjust and usually get a plot which is interpretable and looks optimal in terms of cluster number and separation. And you saw from the one example I showed how much better separated clusters tend to be with UMAP compared to an identical type of analysis with the TSNE on the same data. So I don't know the exact answer to your question, whether it's ever helpful to down select a subset of the data and perform essentially a smaller computational analysis, but I think it's probably not generally necessary with UMAP.
Hi, Steve. Leandro here. Great to see you.
Hi, Leandro.
So,
I don't see you, but I recognize your voice.
I was wondering about the data analysis. I think we discussed this before a long time ago, that with this automatization of the data analysis, it's different to the all gating strategy. Of course, it's impossible to do with 68 parameters, but I was wondering how we can actually really relate when we have a real population, especially for small frequent populations, and just an artifact. And that actually challenged me because, when I see all of this new UMA analysis and I see something new, it just can't relate how we can manipulate, because this is some kind of automated analysis. It's not the classic bioinformatic, approach when you have a person that actually program the algorithm.
That's a good point. We can't really think about it the way we think about low-dimensional data. So you could get clusters which you get very excited about, but ultimately you're going to find that, well, they're really not, cluster 6 is not really that different from cluster 7, 8, 9, 10, or whatever. And I don't think that we don't have the tools yet to perfect this. You have to pursue things that you think are interesting further. You have to then go and look at all the other markers that map to this, if you find, like in this case, a unique cluster, which you think is disease related, and maybe even treatment response related, you're going to have to ultimately isolate those cells and really figure out what they are and what they do in isolation. So your point, it's a very important caveat that you make. I mean, don't just look at this nice, brightly colored map and say, I have identified all these different distinct, unique types of cells. But I think that there's a lot of power in this, and it gets better all the time. So we're not wasting our time by pursuing some of these unique subsets. That's probably where the money is going to be in immunology research for much of the future. The things that were there all along, but people just didn't pay any attention to or didn't notice, like ILCs and such, relatively infrequent cell populations, which turn out to be very important. I don't know if that's a satisfying answer or not, but I think it's the best I can do.
Yes, thank you, Dr. Bertelli. Thank you very much.
Thank you.
We will now introduce Dr. Cristian Carvajal, Associate Professor of the School of Medicine of the Pontificia Universidad Católica de Chile.
Hi, good evening. Well, I'm thinking I was reading the program. I was a little bit the outlier here, but I will try to show you our results and try to get into the flow cytometry in the last slide. Okay. Okay, I have nothing to disclose. My topics today are I'm going to talk about the endocrine hypertension, and also a pathology that's called primary endostenosis. In primary endostenosis, we have studied for long years with Carlos Fardela, and we have a very nice data with extracellular vesicles, and then I'm going to make some point in that and show you some results. In the last slide, I'm going to show you some examples of three research that we are trying to reach out the flow cytometry. I don't see here. Well, as you know, arterial hypertension is a complex pathology. This pathology have a high prevalence in Chile. It's around 33% in Chile. Arterial hypertension have a lot of cardiovascular risk. Can you see in the picture? There is risk like a stroke, blindness, blood vessel damage, heart failure, kidney failure. And these patients can be associated to some endocrine alterations. As you see here, in Chile, we have an increased prevalence with the age. So when you are older, you get more incidence or prevalence of hypertension. I'm a kind of basic endocrinologist, so I'm gonna show you the RAS system. The RAS system is the system that mainly control our blood pressure. The blood pressure is mainly directed by 4 peptides and 1 hormone. And so angiotensin, angiotensin 2, angiotensin go to the adrenal. I don't know how it's pinpointed here. I don't see my picture here. My presentation is pinpoint. No sé si el cable o otra cosa. Yo creo que el adaptador. Porque acá salta todo el rato. ¿Por qué? Salta mucho todo el rato. Ya, okay. I'm sorry. Let's move forward. I'm gonna make the point. Aldosterone is a huge regulator of the blood pressure. So aldosterone joined to the mirocortical receptor in renal and kidney and the colon. And this system go back and have a negative feedback with renin go down. So when you have an unregulated activation of this system, aldosterone and mirocortical receptor, the MR system, you are unable to suppress the effect. So you have constantly a chronic and upregulated reabsorption of sodium and water, which leads to hypervolemia and hypertension. This turns the feedback go down. So renin, you have a very low renin. And when you ask, how can I identify patients with hypertension? Because hypertension is a disease that have a lot of hallmarks that are key in this disease. You can start by trying to elucidate what's happening with the endocrine system. So as just say, you can go forward and move to measure low renin or measure renin, and you see this is in very low concentration. You are in the 33% or one third of the patients with hypertension. In this course, we have measured in 20 years that there is a pathology called primary endostenosis that is in the 10% of patients. This pathology is characterized by a high aldosterone and unregulated. Aldosterone is able to activate the mirocortical receptor, and this have a prevalence 6 to 10%. That is in Chile and most of the country that are commitment with detect the disease. And PA also has been identified that leads to an organ damage. And more recent, we have known that the phenotypical spectrum of aldosteronism is go from normotension to hypertension. So you can have a normotensive people like us, I think so, and we can have a primary aldosteronism in our blood. When you go to the physiopathology, you can see the adrenal gland is the main tissue that controls the aldosterone. So in the adrenal gland, there is expression of one enzyme that is called CYP11B2 that synthesize aldosterone, and then this go to the MR receptor. Aldosterone, as just say, is synthesized and go to in the circulation and go mainly to the MR receptor in the kidney, so produce the reabsorption of sodium and water. But also you can have activation of MR in other systems like heart, vessels, immune system, and also adipocyte. This activation can induce, sorry for the Spanish, fibrosis, hypertrophy, inflammation, inflammatory phenotypes, and other kind of bad things. And how you detect PA in the real life? When the patient arrives to the clinics, if the patient is a young hypertensive patient or have a resistant hypertension, taking 3 or more drugs for control the hypertension, you may have to do an analysis of aldosterone and renin. So you cross this variable and you made a ratio aldosterone to renin, and you see when the aldosterone to renin is higher than 25, like this value, and you have a low renin, less than 1 or 0.5, you can say you have a screening positive for primary aldosterone. But that was like a 20 years ago. 20 years ago, this start to change because the number is not similar in several countries. So you can have a aldosterone renin cutoff of 25, or maybe you can have another form to detect the PA, like a plasma aldosterone, like in 9 and renin activity in 1. So there is some research that point to there is a phenotypical spectrum of the disease. And this phenotypical spectrum say that you can have over PA is the common PA with a higher aldosterone to renin ratio, but you can have a middle PA or subclinical PA when you have another all the factors identified in the primary aldosterone. May you have no hypertension, may you have no hypokalemia, may you have no another symptoms that are very close, are very related to PA. So it's necessary to identify novel biomarkers to support the diagnostic and prognosis of PA, since you can have high aldosterone ratio in your blood, but you are normotensive, and you can have a prognosis, maybe you have PA in the future. When you see the biochemical identification of this patient, you can more or less identify, this is very new, identify that the adrenal gland of this patient may are altered. This is a picture that is related to this triangle that can note that you, from your glands when are normal, have a very low or a regulated synthesis of aldosterone. And when you go through to the alterations, you can have a older adrenals that can have adrenal producing micronodules, or you can have a bilateral hyperplasia or idiopathic hyperaldosteronism. That also can increase your aldosterone in blood. And also you can have a aldosterone producing adenoma, that is a neoplastic alteration of the adrenal, and you have a very increased aldosterone in your blood. So the biochemical identification of this patient can say something about the adrenal of this patient. So what we know, the etiology is the primary aldosteronism that for us is a very important disease because it's 10% of the hypertensive patients have this. It's a chronic unregulated aldosterone biosynthesis. I just say that, and I want to highlight the phenotypical spectrum. But when you go and do the biochemical analysis of this patient, you have some pitfall and bias. These peptide bias are associated to the high antihypertensive drugs can change aldosterone and renin levels. If you use more salt in your diet, you can also increase the aldosterone. And there is other components that can affect this rating. There is also también, a limitation to confirmatory tests that you can do after you have a positive biochemical, positive screen in biochemical. And that is a bunch of reasons that you can say this PA diagnosis today requires a novel algorithm and novel complementary biomarkers. And here I have working from like 7 years, more or less, in the extracellular vesicle as a complementary biomarker of this diagnosis. When you see the literature or the state of the art in this disease about complementary biomarker, there is some study pointing that steroids like aldosterone, deoxycorticosterone, tetrahydroaldosterone are useful for big complementary biomarker. Also, there is some microRNA that have been also described for this problem, like be a complementary biomarker. NGAL and AGP1 and pendrin also have been protein and peptides added as complementary biomarker. And lately, our group and other group in Italy describe extracellular vesicle and extracellular vesicle cargo as a complementary biomarker for the disease. But none of these biomarkers are now in clinics and supporting diagnosis or prognosis of PA. 2 years ago, 3 years ago, we published a paper that we state somehow this problem. When you have a essential hypertensive patient, you do the biochemical screening for PA, you can find high aldosterone, low suppressive plasma renin, and high rate. So if these are very high, you have a ORPA, a patient that can have something in the adrenals, so you have to go to a therapeutic treatment or surgery. But appears also patient with a subclinical PA or possible PA that they have to do a confirmatory test, or I don't know if it's better, but alternative, you can use these complementary biomarkers that I just say. You can use some protein, steroid, or peptide in the blood or urine, or also you can use potential biomarkers like EB protein that we found, and also microRNA that we also found. In other groups, the microRNA 45, 16 is other group. So what are the extracellular vesicle? I know the people here have been hear about the extracellular vesicle, but these are vesicles that are released by most all the cells or tissue in our body, and also in plants and in all the organisms. And these are commonly have a size of 50 to 200 nanometers, more or less. Uh, 2 years ago, there is a task force for this and guideline for this, trying to separate all the extracellular vesicles that you have that we have in our blood or in biofluids. So you have microsomes, ectosomes, large exosomes, and apoptotic body. The size is important, but also the endocytic root that have these vesicles. But for our talk today, the important are the exosomes because they have signals and are commitment with the function like a biovector in our cells. What is inside in the membrane of the extracellular vesicle? You can find proteins, nucleic acid, and lipids. a protein like tetraspanin and MHC, the component of this system, the endocytic root. You can have microRNA, non-coding RNA. Also, you can have DNA in fragments or fragmented DNA. And you can have also all the membrane derivate or protein associated to the membrane. So we already know most of EV, so we can say, let's go to do something with it. Yeah, okay. EV function like a liquid biopsy. So you can have a saliva, blood, saliva, sorry, and urine. For to do this, you can have a good system for us isolate the extracellular vesicles, like ultracentrifugation, ultrafiltration, SEC, and you can have your particles. So you can do all your experiment that you want to do and start to identify or associate exosome with your disease. But before you do that, you have to you need to have a reliable isolation and characterization and quantification of EVs. You can not go through, at least you made this. So you can find a good system to isolate your EVs. You can, I just say, you can use size exclusion chromatography, polymer precipitation, immunomagnetic beads isolation, and also differential centrifugation. We used this one, differential centrifugation. We had an ultracentrifuge, and I show you here the 3 samples that we investigate in our lab. Urine, plasma and serum from patients with PA and other endocrine disease, and also extracellular vesicle from cells, culture cells. These are, well, you need a higher number of cells to have a good number of exosomes. These are our techniques. We use NTA, Western blot, we use the TEM for characterizing all the steps that you need to have your EVs. This is the way this is the result that we have. We use the nano-tracking particle analysis that you can see in the video. The particles are put in a nanoflow cell. This nanoflow cell can identify the movement of the cells with a means by an equation. It transforms the movement of the cells in quantity and size. You see the TEM. Okay. So I'm going to show you three papers that take this concept, the primary endocytosis, the EB as biomarker, and we are trying to find out how we manage this information. So we have a cohort study with a patient from 18 to 65 years old without comorbidities, without treatment for hypertension, and the ratio that I say in the first slide, over 25, and the renin activity low than 1 nanogram per ml per hour. So you identify patients, in this case, 16 patients with primary aldosteronis, and you see here aldosterone is higher than control. The plasma activity that I say in the first slide go down, is very low, and the aldosterone renin ratio is higher. Okay, and so you characterize your patients with this. Then we start to do these things with the extracellular vesicle. We isolate the extracellular vesicle by the ultracentrifugation system. And then we see the extracellular vesicles, I'm going to say EVs by TEM. And then we go further and we identify a protein from the surface of the extracellular vesicle, CD9, CD63, and one of the inside, TSG101. And we have the curve of the concentration against size. So this is a typical curve of nanoparticles as EVs. When we start to do this, we identify firstly exosome from urine. So we must to standardize this protocol. So we see here the urinary creatine correlate with particles. So you have more urine, you have more particles. So you have to do the ratio. And PA have more particles than control or normotensive people, and but are smaller. Okay, more particle but smaller. After that, we isolate this particle and we do proteomics. We have 16 patients proteomics against the 16 controls, and we identify one protein. This protein is called AGP1, also is called oromosucoid protein 1. And we test by Western blot. And we also observed that this protein was in higher concentration that appears in control people. We published this paper. So we add a novel biomarker to this mixture of the complementary biomarkers. Other paper, the second one, in the camera is the second one, is this one, a characterization of gene expression analysis of serum-derived ABs in primary aldosterone. This work used for first time flow cytometry. You know, flow cytometry for me is far because my vesicles are lower than 200 nanometers. So you have to do some things to be able to measure the vesicles. This study made the quantification like us by NTA, and they made a second quantification or analysis by fax and in an equipment called Cytoflex, the Beckman, and are able to measure CD31, CD42B, and CD45. This use bits, so increase the size of the particles, so they are able to measure. You can see here again the NTA. You can see the exosomes or EVs. You identify by Western blot CD63, TSG101, and other protein of the membrane, protein 1. And also, they see the measure, they made a measure of diameter and concentration, and in blood, they see the similar that we saw in urine. So we're very happy with that because we are doing the work, different part of the world, and find most the same. The patient with primary aldosterone have higher extracellular vesicle and have a lower or smaller display. But they add with this system, the Cytoflex, other point that they found the contribution of some when you have the blood of this patient, you can have a pool of all of the tissue of your in circulation. So they point out the vesicles that they are identifying came from the endothelium, platelets, and leukocytes. They also made some expression of 4 factors that they found in a microarray that they do, and find some interesting protein that's caspase-1, endothelin 1, and the receptor of factor 2, and the M-oxygenase. So it was very interesting to know about this data. And also, they add a very important point for us that when you this patient have an adrenal an adenoma, it's a neoplasia in the adrenal gland. So when you go to surgery and you take out the adenoma, you see rapidly, like I don't know, the next day, a big fall of the nanoparticles in blood. You can see here post-surgery, the particles go down. Also, the markers that they also identify go down. Another paper, the third, that you can see with the camera, use add is the same group that the second one. Add another way to see how is this vesicle. So they do nanoparticle track analysis because the guideline of the ISEV, the International Society of Extracellular Vesicle, is also you have to do that, the NTA. And they made flow cytometry with max quant analyzer. They can identify 37 EV surface antigens and mostly CD. And also they add a high resolution flow cytometry in the Beckman equipment. But they increased the paper that they saw, the last one in 2019, they increased from 3 antigens to 8 antigens. You can see here the blood samples by serial centrifugation obtain the exosome. The exosomes are loaded or combined with beads attached to some dyes, and they made the immunocapture and they read in the this is for the point 2. They showed this NTA and the protein that are critical for rise these studies. A protein that is in the endoplasmic reticulum that you don't have to see in URVs. TSG101 you have to see in URVs. Synthetic 1 you have to see in URVs. APOE1 is in the particles, the lipoparticles, lipoprotein particles. So you don't have to see in your isolation of EPs. And CD81 you have to see in URVs. So they perform a study taking together this analysis in the Max Plan platform, Max Quant, sorry. And they arise this figure. In this figure, you can see they made the analysis of most of the 37 antigens. And when you see, I don't know if you can see here, this is the normal. This is the people with the aldosterone producing adenoma. So the picture or the heat map is very different between this one is the normal and this one is the normal. So they say we have a signature for these patients. Also, you can see here there is some difference between the people with PA, primary aldosteronism, against people with essential hypertension, which is similar to normotensive. So PA separate in most of the CD antigens in a good form. So they speak about the signature of this antigen of the member. They also remark this when you see in PA patients the antigens of the surface, you identify most of the protein came from the endothelium, the red one, and also the platelets. But also they say there is a very good contribution of T and B cells, which is important because the hypertension is not only a disease that reabsorbs sodium and water from the kidney, also is an inflammatory disease. I think this is the last one. And finally, as um, a, a image from this paper also show this. When you have another a neoplastic in your adrenal gland, you can go for surgery. So this is called APA, the hyperplasia. I'm sorry, the neoplasia. The red one and the orange one are the cures for this, pre and post surgery. When you have your extracellular vesicles in the disease, you have a very high peak of nanoparticles. When you take the surgery, you go down with your particles, and you also you are more or less in the level of the normal people. So there is a very good contribution of this very tiny adenoma for increase your nanoparticles in blood. So there is something apart from aldosterone that can be increasing your nanoparticles. We are studying that. And when you go for pharmacology treatment like spironolactone, you have also a decrease of your nanoparticles in your membrane. So when you block the MR, also the effect of aldosterone and MR is blocked. So there is a contribution also in this system. Well, the conclusion of this presentation are these. I hope you can take this like a take-home message. Primary aldosteronism is a prevalent and underdiagnosed cause of secondary hypertension associated with a significant end organ damage, and also can increase your cardiovascular risk. PA exists along in a spectrum, so I explained that. Requiring refined tool for accurate diagnosis and prognosis. The the extracellular vesicles, the particles that I'm saying, and their cargo are emerging as a novel biomarkers in this pathology, and also can be a mediator of the pathophysiology of PA. So you can take this out and read what's happening with the extracellular vesicle, but you can also ask if these are communicate something between tissues in your body. And a specific cargo that we published in 2 years ago, and also the 2 publications that I show you, suggest that there is potential application for clinics rising from this study in these biomedical labs. That was my first slide. Of course, I acknowledge to all the people that work with me and all the collaborators. Thank you.
Thank you very much. So we have time for only one question, if anybody in the audience has one.
Thank you, Christian. I have one question. When you talk about the exosomes and the vesicles, how different is the content of these vesicles? How conserved or how different is the content? In pathology within different patients.
With PA?
Yes.
The PA is a disease that I think should be similar between patients with the same phenotypic. If you have high aldosterone, you have higher chances you have a phenotypical spectrum that is similar to other PA patients. But there is some bias. When you see females and males, there is some difference in that. Also, you have bias. You have talking only about PA patients. If you have younger PA and older PA, you also have another profile. So we should standardize that between sex and between age. So you can have around 6 groups when you have to have a profile, a signature according to that profile, to that patients.
You know, first of all, really interesting. And I have a question about how do you stabilize your sample, right? Because EVs are shed, right?
Yep.
So if you have control over your sample from a clinic to come into your lab, then that's a very controlled space. But if I have to ship my sample around in a box that gets thrown around, how do you control the kind of EV that you look at inside your sample?
Mm-hmm. It's a good question because EVs are main nanoparticles made from membranes, biological membrane.
Mm-hmm.
And the first difficulty that we have is frozen the particles. So because the particles, if you don't store in proper condition, you can broke out. Actually, we lost like a 30% in every sample we froze. So there will be a chain of GMP, good or GLP, good laboratory practice, to avoid any bias with that transfer from when you take out the blood, withdraw the blood, and finally when you put in the machine for read what is inside. Okay. We have to work in that, but for this time, I'm very happy with this, and we need to move maybe in a short future, to some technology that can store better the exosome, because we already know we lost 30% of exosome in every 1,000 freeze cycle that we do.
Okay. Thank you. Thank you very much.
Thank you, Dr. Carvajal. We next welcome Dr. Arturo Borsutsky, Associate Professor of the School of Medicine of the Pontificia Universidad Católica de Chile.
Hello, everyone. That's better. Thank you to the organizers and the Institute for inviting me to participate today. I think I need to take this off. There. So I'm going to talk today about flow cytometry in the clinical space for immune diagnostics. So as you well know, this all starts with our patients. We have all sorts of patients. Personally, I'm a pediatrician, so we see all kinds of different patients with mild diseases, moderate diseases, and very, very severe diseases as well. And many of the diseases we see in immunology are in that severe spectrum. And throughout the recent history, we many times have had the problem of not being able to diagnose them adequately. And in particular, studying the immune system, as you all know, is something quite difficult. And that means that having good quality and precise diagnostics in immunology is of utmost importance, and probably a long and hard endeavor, and very costly as well. So clinical applications of flow cytometry, this is a very summarized version, but this, as you probably all know, this started in the 80s with HIV, measuring CD4 T cells, and that's the reason many of the hospitals, starting in the United States and Europe and then elsewhere, started doing flow cytometry to being able to measure the CD4 T cells. So it started probably in infectious diseases space, and then rapidly went on to hematology, oncology, with a diagnosis of leukemias and lymphomas, and side by side with immune deficiency. But this has expanded once transplants were started in many places. Flow cytometry became a very essential part of transplant medicine, and then to new areas, including autoimmunity, allergy. This is very strong nowadays in many parts of the world, and many other things like rare cell detection, tumoral cell detection in blood, and stuff. So my area of specialty is pediatric immunology and rheumatology. So we're going to center this talk in primary immune deficiency, which is the main reason I got involved in flow cytometry, and especially in the clinical side of it. So primary immune deficiencies now is also called inborn errors of immunity, IEI. So we used to talk about PIDs, and now we also talk about IEIs. So this was first documented, one specific immune deficiency was first documented by Dr. Bruton, who discovered BTK in 1952 in children with A gamma globulinemia, so no antibodies in the blood. But now, 70-something years afterwards, there's more than 500 different immune deficiencies. And the list grows every month, I would say. So it's about 10 new every year or even more. So it makes it quite complicated to know everything that's going on in the field. And we usually said that immune deficiencies were characterized by recurrent or severe infections, and that is still the case. But nowadays, the spectrum is far wider. We have all sorts of autoinflammation, autoimmunity, cancer of several types and other tumors, and many times severe allergies as well. And there's different combinations of these. So there's some kids that have infections plus recurrent fevers, or severe allergies plus risk of lymphoma or things of various sorts. And the prevalence of these diseases is growing, probably very likely because of better diagnosis, and because of the fact that we have better treatments nowadays. So these patients don't die very early in life as they probably used to in worldwide. And this is a figure we published about 10 years ago that we measured hospitalizations due to immune deficiency. So with discharge diagnosis of immune deficiency, and we saw that in 10 years, the hospitalizations due to immune deficiency doubled in the country, and this has probably gone up much further since I last looked at it. Just remind you of what alerts us to a primary immune deficiency. So this is the Jeffrey Modell Foundation's warning signs of PID, and then they made an adult version because adults have less infections than kids. So having 4 or more ear infections in a year in kids prompts a suspicion, but in adults, like any of you guys, if you have 2 ear infections in a year, it already raises suspicion that you may have something in your immunity, and so on and so forth. So use, obviously, of flow cytometry in these diseases has expanded to now very complex functional studies. So we went from counting CD4 T cells to everything that you have seen today, which is very outstanding. However, the local capacity was limited to very few studies until very recently. So we and others in Santiago decided that we should do something about it because it was not feasible to be sending samples to the US or abroad for flow. The shipping takes at least 24 hours, and many times the cells died on the way, and it was very expensive to do it that way. And we started developing a project to have immune deficiency and immunity in general testing in our clinical lab. And this is our current list of flow cytometry testing. So this has about 5 years of existence, and it has greatly expanded our capacity. So lymphocyte populations and leukemia phenotyping that was there from before, and I'm not in charge of that at all. But all the rest are sort of new tests we have and have greatly changed our possibilities to study our patients and to do very fast diagnosis many times. So I'm going to show you some clinical vignettes of different patients where we have used flow cytometry for diagnosis, for follow-up, for confirmation. So this first one is a 9-year-old boy who presented 2 years ago at our institution for fever and a respiratory infection. So he came in with a cough and fever to the clinic. He went to see one of the bronchopulmonary specialists, and he was sent home on antibiotics. Notably, he had a history of recurrent ear infections, sinus infections, bronchitis. He had been recently hospitalized because of a pneumonia. That was what the parents told the doctor that day. But it appears from what the doctor wrote that they didn't give much of a severity impression to him. And digging deeper, we learned that at 4 years of age, he had had a salmonella infection with positive blood cultures. So having a bacteremia by salmonella is usually a sign of some immune deficiency. And then at 6 years of age, he had a prolonged fever with lymphadenopathy in the neck, which was found to be due to Bartonella. But then so people suspected at that time that he had some immune problem, but they were lost to follow up. They didn't come back. And what they told the doctor that day is that the kid had a normal immune workup and even a genetic study that didn't find anything. So he had been sent one of these panels of 200, 300 genes, now they're larger, to the United States. And there have been some variants of uncertain significance, which we call VUS, which are variants that are not really pathogenic, but we don't know. But nothing serious had been found. So he came back a week later and was admitted immediately due to high fevers. He was ill appearing, looking a bit septic, but had a normal chest x-ray because everybody thought he would have had a pneumonia. But his other studies were not looking good. He had very elevated inflammatory parameters, low albumin, low platelets, large liver, large spleen, and he went to the critical care unit. An immune deficiency was suspected, possibly complicated by a hyperinflammatory condition we call HLH, which is hemophagocytic lymphohistiocytosis, which is kind of a storm of activation of your T cells. And that was suspected because of the fever, the splenomegaly, the low platelets, and it's something we see much more commonly these days. So one of the things you could readily suspect with the history this kid had of infections was a chronic granulomatous disease. So this is a problem of your neutrophils in particular, and also monocytes that are not able to have the respiratory burst inside the phagolysosomes and destroy the bacteria or fungus as we would like them to. So this is a study we did in our lab where we can see that the control is able to uptake a pigment called dihydrorhodamine. And when activated with PMA, the cells, the neutrophils of the person change this dihydrorhodamine to rhodamine, which is a pigment, and then we see it move in the x-axis, right? And our patient, you see that upon activation with PMA, the MFI is right there, almost in the same place as it was. And when we look at the mother, because this is an X-linked disease most of the times, you see a double peak. So the mother had half her cells with the problem and the other half without, but because those cells were enough to fight off disease, she didn't have any history of infections, which is the common scenario. So this is quite a serious condition. I put these slides one before the other. So this is, as I said, the dihydrorhodamine. You gate on granulocytes. Normally, this shifts. There are some variants that you have different kind of curves, but normally you have this figure when you have the complete deficiency. This you see when you have some of the other genes that cause autosomal recessive CGD, and this is what you see with carriers. But this patient did not only have that. This is a test we implemented very short time ago, where you see CD8 T cells, and that we mark them with CD38 and HLA-DR. Probably you know these molecules. These are hyperactivated CD8 T cells, and this is very specific of this HLH condition, where you have this storm of CD8 T cells and NK cells and others causing this major inflammation in the blood that is usually fatal if untreated. So this kid not only did have a problem fighting off infections and probably had an infection going on, but had this hyperinflammatory state that needed immediate treatment. So we started treatment with prednisone and high-dose intravenous immunoglobulin. Started antibiotic and antifungal prophylaxis, which is what we usually do with this condition. And the patient was able to go home in good condition. And we offered them, the parents, hematopoietic stem cell transplantation, so bone marrow transplantation, but they declined. Not everybody with CGD gets transplanted. If it were my child, I probably would, but they decided to continue prophylaxis with antibiotics. And then I think we lost them once again. Things that happen here. So this is a very different kid, 2-month-old. So the kid was born here at our hospital, and then at 2 months got admitted due to a parainfluenza 3 bronchiolitis. So this is very common picture, kid with bronchitis, wheezing, cough, and nothing was very out of the ordinary, except that the kid started not responding very well to the treatment we were giving him. So we did a very common complete blood count, hemograma in Spanish, and the hemograma showed 880 cells, which is far lower than a kid aged 2 months should. And because of that and the ongoing infection, we deepened our immune studies and found that he basically did not have any immunoglobulins except for the IgG, which was in normal range. Remember that IgG gets passed through transplacental transport from the mother, but he didn't produce any of the other ones. And when you look at the T cells are almost entirely gone, and he has no B cells. And on the other hand, NK cells were quite high, right? So we have a kid that evidently has something serious going on. So we try to look at those T cells in our lab to see if they proliferated upon stimuli, and we usually do PHA and stimulate T cells through that. So, you know, the PHA stimulates the TCR from outside, so it's not an antigenic stimuli, but mitogenic. And we see here that the kid has very little T cells, as you saw in the previous slide, but they almost did not proliferate. So this is a CFSE staining assay, where you see that upon proliferation of the lymphocytes, the CFSE gets diluted day by day. And that's what we see in the normal, but not in the patient. So very few T cells that don't work. We also looked at whether those very few T cells were memory T cells or naive T cells. A kid that has 2 months of age usually has almost every T cell is naive because they just came out of the factory. And as you see here, the control, who is an adult, usually we take from the blood bank, has very good numbers of naive T cells and also a portion of memory T cells. But our patient, the few T cells he has are memory. So those are activated T cells. And last, and probably the most precise test we do for diagnosing a kid that has a severe combined immune deficiency, as you probably guessed, is the measurement of recent thymic emigrants. So you stain with CD31, CD45 array, and CCR7, and you see here that the patient does not have any in the upper right quadrant. So the thymus is there. He has a thymus, but the thymus is not outputting any T cells out. In this case, that's a T minus, B minus, and K plus severe combined immune deficiency. So unfortunately, this patient, when we already received him, he had a very severe liver dysfunction, probably due to the infection, and progressed rapidly to respiratory failure, died before hematopoietic stem cell transplant could be performed. And his genetic studies that we were able to send out confirmed that he had RAG2 mutation, which is one of the relatively common genes that cause SCID. And in Chile, we still don't have newborn screening for SCID, something that is available in every state in the United States right now. Kids, when they are born, are screened to see if they have severe combined immune deficiency. And if they have it, they undergo transplant as soon as possible before they get infected. Unfortunately, most of our kids with SCID get infected before they are transplantable, except those that are born of mothers that already had a kid before with SCID. So there's work to be done there. Third case, 8-year-old Chilean girl, previously healthy. It's a very unusual case. She started with a swelling of her eye, and that obviously got to the hospital because of that, and she had a mass in the orbit of her eye that got biopsied, and it was a lymphocytic kind of mass with IgG4 positivity, but she also had this autoantibody, it's called ANCA, anti-neutrophil cytoplasm antibodies. And finally, the biopsy confirmed that she had a rare kind of vasculitis in children called granulomatosis with polyangiitis, previously known as Wegener's disease. You may have heard before. And she was treated with corticosteroids, mycophenolate, rituximab. Rituximab is the anti-CD20, so it eliminates B cells from your circulation. And she improved and was discharged in good condition. But a few months later, she came back with a genital cellulitis, very unusual infection due to salmonella in this case. And she was found to have very, very little immunoglobulins in the blood. So rituximab can cause that your immunoglobulins go down. And that was suspected at that moment that rituximab just hit her too hard. So she was started on intravenous immunoglobulin replacement every month. But she also progressed to have bronchiectasis, which are these dilatations you see in the bronchi in that chest CT, which is not a common finding in this vasculitis. Vasculitis usually has nodules and granulomas and things like that, but it's rather a sign of immune deficiency. So very rare, this vasculitis does not usually associate with any immune deficiency, but we started looking around. And first we looked at her B cells and did a study of naive and memory B cells. So this is commonplace nowadays. You'll stain for CD27 and IgD. And as you can see, the control has a good number of CD27 positive, IgD negative B cells, which are the ones that are class switched already. But our patient did not. So she had few class switched memory B cells. And so we, in the end, decided we had to do a genetic study because we were not going to figure out this very easily. And we found a heterozygous pathogenic RAP27A variant that had been previously reported in atypical Griscelli syndrome. So this was quite a surprise. Griscelli syndrome is something very rare that usually are kids that have very low neutrophils and have albinism. So their hair and eyes are white, right? But also Griscelli syndrome is autosomal recessive, so you need 2 mutations, and she didn't have albinism, so her hair was okay. But so we started looking in PubMed, and there were very few cases described with something called atypical Griscelli that had immune dysregulation and strange things and without albinism. So if you remember your basic immunology and cell physiology, RAP27A is a protein, is a GTPase that helps docking vesicles to the plasma membrane and then helps them open up. So that's why they have albinism, because the melanosomes need to dock and open to synthesize the pigment. But also in our immune cells, like the NK cells and the T cells, you need those vesicles to dock onto the plasma membrane. So specifically in NK cells, that is very important. And we have an assay for that, which is the CD107 degranulation assay, where you stimulate your NK cells with a special kind of cell that we have in the lab. And that makes the CD107, which is the like rectangular protein that is in that vesicle, open up and expose itself to the membrane transiently. So you stimulate the cells and you see how the CD107 appears in the flow cytometry. I don't show you here with the unstimulated, but that's not there. And as you see, our patient had very little of that CD107 mobilization. So we confirmed that she had definitely a problem opening up the vesicles of her NK cells, being able to confirm that even though she had one mutation in the genetic studies, she did have a disease compatible with this atypical Griscelli syndrome. So now she's being treated for a severe autoimmune disease and a severe immune deficiency at the same time, which is quite a challenge. And getting to the last one, this is a 13-year-old boy with X-linked granulomatous disease. So the first one we saw, CGD, this one was transplanted at age 10, but had lots of complications. He had reactivation of cytomegalovirus, adenovirus, other infections. But the transplant grafted well. So at day 30, he had 95% chimerism. That means that 95% of the bone marrow cells were the donors and 5% were his, but which is a good result. And had, most importantly, a normal respiratory burst. So that dehydrorhodamine changed to rhodamine had normalized due to the transplant. But then again, 1 year later, the chimerism had gone down. So his blood cells started reappearing, and the respiratory burst was showing that half were normal and half were not, half were mutant. So we usually say that if you have more than 20% or 30%, you should be okay. But this kid started decreasing, and we did this surveillance post-transplant, and you can see that you go from a normal respiratory burst further to the right to having this half and half condition. And then this is the last one, which was about a month ago. You see that he has very little of the normal cells remaining. It's about 8%. So in this case, it's a very small example of post-transplant surveillance and how it helps decide the future and the treatment of the patient. In this case, prompting the decision to retransplant probably at some point in the very near future. So acknowledgement, special acknowledgement to Dr. Rodrigo Hoyos, who is a pediatric immunologist that was in our institution and left very recently, but he was the one who set up this flow cytometry lab. So it's more of his merit than mine. And that is the very small facility where our cytometer is. Marianela is the lab tech in charge. So it's pretty amazing, but it's one person. And if she goes on vacation, she gets a replacement and there's another person. And that is one of our fellows right now who's learning about flow cytometry, because we really believe this is something that should be part of the core curriculum of all our immunologists. That's it. Thank you very much.
Voy a correr y de vuelta.
So thank you very much, Arturo, for your talk. I'm quite surprised that the age of disease, of relevant disease that brings the person to the clinic for a diagnostic occurs so late in life. I would have expected that, given our typical immunology courses, that because of these problems, you would expect much earlier on severe problems. So how come does it occur so late on?
It's a great question. And what we know nowadays is that usually we expect immune deficiencies to appear in the clinic in the first 2 or 3 years of life, but more and more we see adolescents, adults, even 50-year-olds come to the clinic with immune deficiency. There are some like common variable immune deficiency that can debut at 50 or even 60. So this is something not only pediatricians should know of, but all doctors. No, well, if it were not for hygiene, they would have died earlier, maybe. But this is just because of under recognition. Before we had no idea. And many times when we started seeing these patients, you heard these horror stories of 30 years being ill with different infections or lymphoma, and lymphoma once again, and nobody thought of an immune deficiency.
Thank you for your talk. Very, very interesting. In regarding the immunodeficiency in kids, how about the response to vaccines? For instance, the BCG that is inactive attenuated vaccines. Is there any like sign upon vaccination that this kid could have a immunodeficiency?
So most of the vaccines we use nowadays are either dead or inactivate or component vaccines, and those are no problem. So either the kids will respond normally if they have B cells and T cells, or to the spectrum goes that they won't respond at all, but they won't cause any harm. The problem is with live vaccines, both viral and bacterial, where you can have severe rotavirus, measles, BCG dissemination, and those can be life-threatening, for example, for a kid with SCID. And that is a long discussion regarding the BCG, because some of our experts say that BCG, some say that it should not be used, and some say it should be vaccinated at 30 days of life, because you give time for the newborn screening of SCID, and then if that's okay, you put the vaccine in. But we have had SCID kids with BCG dissemination.
Thank you very much, Dr. Borsuski. So to close this session, we welcome Dr. Leandro Carreño, Associate Professor of the Universidad de Chile and Millennium Institute and Immunology and Immunotherapy Deputy Director.
Thank you very much, Antonia. We'll wait for the presentation. So I'll try to be short because I know you're all eager for the coffee break. So I first, thanks. I'm very grateful for the organizers, Alexis, Hernan, Bob, for inviting me and have this great opportunity to be among these incredible colleagues around the world. So I'm going to talk today about one of the passions that I developed. Sorry, it's just here in our lab for the last years. And this is about how the innate immunity controls the adaptive immunity. And I will try to give I know we are all immunologists here, but I think it's good always to have a refreshment and just recollect the idea that, sorry here. Okay, that for the adaptive immunity and the difference with innate immunity is about time. So first, the innate immunity recognizes danger. We have different cells here and soluble components, including the epithelial barriers. And we can distinguish the danger, like, different infections, different virus, bacteria, etc., but not specifically which bacteria, which viruses. So for that, we need adaptive immunity. We need T cells, we need B cells, we need antibodies, we need different type of T cells with different ways of T cells to develop. So, and then we can actually distinguish this. And this is what I call the dogma of the adaptive immunity. When you have danger signals, the PAMs or any kind of danger, the dendritic cells, they undergo a process of maturation when they recognize this danger, and they can process and present antigens to T cells, CD8 T cells, CD4 T cells. But because of the danger signals, they also are included in this interaction, the presence of costimulatory molecules that give the signal to CD8 T cells and CD4 T cells to get activated. So when a T cell, get activated, for example, a CD8 T cells, then can go and recognize the same antigen that they recognize when they go activated, and eliminate infected cells, for example. And that the same way, a T cell can have a conversation with a B cell that actually capture the same antigen, because the BCR recognizes a pathogen, so they can present via MHC class 2 the same antigen that got activated the CD4 T cell, and then the 4 T cell will give some signals to the B cell, including also the presence of co-stimulation for being transformed in a plasma cell and secrete antibody, and then the antibody can block the pathogen and so on. So this looks good, but as you can see here, there is also the presence of cytokines, because it is not the same when we have different pathogens. It's not the same to having to attack an intracellular pathogen like a virus or an intracellular bacteria, an extracellular bacteria or a fungi, or something big like an helminth, for example. So then the cytokines will shape the response of the CD4 T cells, given the differentiation for Th1 cells if it's an intracellular infection, Th17 cells if there is an extracellular infection, or Th2 cells, which is something bigger, for example, helminth, and we have this very associated with allergy, as we will heard from the last talk by Arturo. So the effector cells that are involved in these different type of responses, type 1 response, type 3 response, and type 2 response of the immune system are different. And here, this is something very interesting. We can identify this by flow. The naive T cells, CD4 T cells, then they grow, when they get activated, then they grow a program when they can be a Th1 cell with distinct transcription factors, Th2 cell, Th17 cells, a follicular helper cells, and actually, I'm not showing here, Bob talked about that in the morning, the Treg cells. So we can have a panel. So this is, my talk says advanced flow cytometry. I think it's advanced, and now we have ultra advanced. So I will let you know how we are doing this and what we expect to do with this new equipment that we have here in the Alexis and colleagues lab. So we have a panel, for example, here that allow us to have, for example, one experiment. You can have the spleen of an animal, or you can have a sample in blood. You can actually analyze the frequency of Th1 cells, Th2 cells, Th17 cells. You can even buy some kits, nice kits from within here to have this optimized. So, but the problem is, actually not the problem, the challenge is these things are changing. It's not just one phenotype, Th1 cells, Th2 cells. We can also have the mixture of this, and this is a very old paper that when I was in my time in the STEEP lab, in collaboration with Arturo Casadevall and Rafael here, which is in Madrid right now. You can have a panel here when you can actually analyze the CD4 T cells and the different presence of different cytokines that are associated with Th1 production, but also with other phenotypes. And you can perform a Boolean analysis to see whether these cells secrete 1 cytokine, 2 cytokines, 3 cytokines. And this is, I think, is a nice example when in this paper, we analyze the importance of the capsule of the BCG for vaccination, and how when you challenge, you actually need the capsule to have this phenotype of multiple cytokines secretion or production by different T cells, and these T cells are actually important in the protection. So with this in mind, I will talk about now why innate immune cells are important, why we think is important, why we really believe they are important. Because in the moment of the priming of the T cells, when they actually shape the phenotype for Th1, Th2, Th17, and the magnitude of this, they can be actually modulated by different innate cells, so-called innate lymphoid cells, NK cells, NKT cells. and also by eosinophils, basophils, neutrophils, and macrophage. And actually, which is very interesting is that after the initiation of the adaptive immune response, they actually are recruited back to be effectors of the immune response. So I'm going to talk about today two examples. One is about NK cells. I will show data for other or groups, and then about NKT cells to highlight one concept I think is important when we analyze these multiple parameters nowadays that actually is quite a challenge. When we talk about NK cells, and if you remember what you learned in the undergrad and your undergrad studies, you will recollect that NK cells, they detect the lack of MHC class 2 or class 1, and they kill the cell that actually downregulate the MHC class 1. But if you can see here now, the accepted view of the NK cell regulation of T cell immunity is not also, it's not only about the elimination of the cells that actually have dampened the expression of the MSC class 2, but they also have other outcomes like regulate the response of the T cell, regulate the response of dendritic cells, or even being involved in what we called before the memory of innate memory that now we know and that we accept that is trained immunity that actually was so important in the pandemic. So in this slide here, you can see that the NK cells actually can have multiple phenotypes because they express a lot of receptors, and the combination of the receptors changed a lot. And what is important is that a person that actually got immunized with something, and the NK cells, they shape the expression of the receptors that explain the trained immunity that it will decay, maybe 1-2 years after the immunization, but it does sustain the ability of the expression for a couple of months or even a year or 2 years, explaining the trained immunity. So when we started like 10, 15 years ago, and Bob, you can actually correct me if I'm wrong here, this came off, the CyTOF. And we were all excited about this incredible technology that we have an antibody with an isotope. So you can have as many colors, I would say color, as isotopes we have. You have a cytometer, the CyTOF, and you destroy the sample, and then you analyze just the isotope. You quantify the isotope, so you have multiple parameters. So what the first time we have a lot of parameters. And here, this is the first paper that came out for the NK cell diversity, and you can see these incredible branches. As this was the first type of analysis before the t-SNE. Actually, t-SNE was actually available here, but this branch analysis, they actually you can see the incredible diversity of NK cells. But the problem is, you destroy the sample, so you cannot further analyze the sample, and this is a drastic limitation of CyTOF. But then the multiparametric, the advanced cytometry came off. So you can actually have these nice t-SNE plots. You can identify the cells. These are all NK cells. You can do single cell transcriptomics, and you can see now there is an incredible diversity, and this actually reflects, the trained immunity that you can get. This is mouse when you just have one immunization of an antigen. This is very incredible. So now I will move forward from something that actually I'm very interested in this last 10 years, let's say NKT cells. And NKT cells are T cells. They are T cells because they have a receptor, a T cell receptor. By contrast with what happened with the T cell receptor that is extremely variable, the T cell receptor of NKT cell is semi-variable, so the alpha chain is almost always the same with a very limited ability of beta chain, but they also express some of the receptors of NK cells. These receptors they were very variable, so they have killing mechanisms going on, but also have a regulatory mechanism going on. But is a T cell, so they can have a ligand, and it's a T cell, and they have a lot of regulation mechanisms by secreting cytokines, and this is something that we can see here. So the TCR, the T cell receptor, recognizes an antigen. This antigen is a glycolipid bound to a molecule that is very similar to the MHC class 1, they call CD1D. It's also an alpha with 3 domains, alpha 1, 2, or 3, with the beta 2 microglobulin attached here. But at contrast to what happened with the MHC class 1, is not variable. Actually, it's very conserved in different species. They have some difference, but it's very conserved. And what happened is that this interaction between the dendritic cell presenting an antigen to NKT cells is extremely rapid. So the NKT cell, they have a kind of a memory-like phenotype, so they can secrete large amounts of pro-inflammatory cytokines and actually many different kind of cytokines. And this is happening when the T cell priming and with the B cell priming is happening. So since this happened before the actual priming of T cells and B cells is done, they can influence the phenotype of CD4 T cells, CD8 T cells, and B cells, and also NK cells and other effector cells. So the glycolipid, so the best identify is as a glycolipid derived from a sponge, marine sponge. That's why the sponge Bob. This is just a casualty, Bob. Sorry, coincidence. But the problem is that the first studies with this compound is alpha galcer. This is a galactyl ceramide here with 26 carbons in the lipid acid change with no insaturations. They have a mixed pro-inflammatory and anti-inflammatory response depending on the context. So there is a lot of effort. There were a lot of effort to make some compounds that have some modification in the structure. Here actually, for many years, I have collaborated with Steve and Amy Howell for the University of Connecticut, and she provides different variants of this compound. That, for example, here you have this compound that has some insaturations here in the lipid chain, and it's actually the chain is shorter. You have here the prototypic, the benchmark alpha galcer, and you have here another one that has a replacement on the O-glycoside linkage for a carbon, for example. And because of those modifications, the ratio of interferon gamma to IL-4 change. So this is actually something good because when you want to provide a more direct and unique cytokine signature to perform the immune regulation for the other cells. So they have been modified in every single part of the molecule. Identify some compounds that they were called Th1 basin because they promote more interferon gamma than IL-4, or Th2 bases because they promote more IL-4 than interferon gamma. But in the recent years, this Th1-like and Th2-like has changed a lot, and this is also thanks to flow cytometry. And we now have accepted that actually NKT cells can undergo a differentiation program that is actually extremely rapid, and they can be NKT1 cells, which is very similar to Th1 cells, NKT2 cells, NKT17 cells, and this is very interesting and was actually launched actually 10 years ago, specifically 10 years ago by the group of Mitch Cronenberg, the NKT10 cells. They can secrete IL-10, and which are very important for negatively modulates and or induce tolerance in the immune response. So in my lab in the recent years, we were interested on see how this can modulate the activation of B cells and T cells, and using what we learned about these interactions to see whether we can actually use this information when we have pro-inflammatory response to treat infections and cancer, or when we have anti-inflammatory responses to treat allergies and autoimmunity. So for this talk, I will just focus on this first part here, and to induce and response, and I will go back a little bit. that we have antigens to be recognized by NKT cells and B cells, or NKT cells and T cells, we have used liposomes. So liposomes are very easy to use, and thank you, Christian, that you give a nice introduction about nanoparticles, so I can skip that part. And we have been doing these nanoparticles. We can have the antigen inside the nanoparticle, or have the antigen, the T cell or B cell antigen, outside the nanoparticle, and with the glycolipid embedded in the lipid bilayer. This is just an example of the size. This is a machine called nanosite. You can have here, you see here the liposome just going through the flow. Actually, this has been done in Christian lab. And this is just for fun. We did this last year. This is an atomic force microscopy, where you can see cut it in half, and the liposome, very nice. But importantly, they actually can be measured by flow. And this actually, at the beginning, was so hard to see them because they're so small. They're 150, 200 nanometers. And this is done in a normal cytometer. And this liposome actually contain in the membrane an antigen. This is our protein, so you can have an antibody and detect them. Nowadays, because this is done with the 4088 laser for detecting forward scatter and side scatter, and now we have the capabilities in our facilities to actually change that and use the 4 or 5 laser, and that actually allows to detect more easily the nanoparticles. So actually, this is a great advance for us because now we can formulate different size and really see the difference in size, in complement what we do with the nanosite characterization. So one thing actually that was very interesting for us and actually very unexpected is that because we're using these nanoparticles to formulate the glycolipid preparations, we compare what happened with NKT cell phenotypes when we use soluble glycolipids on glycolipids contained into liposome. And this is just the result of an extensive gating, including NKT cells, B cells, T cell, excluding the cells that are not important. And you can see here the characterization of an NKT1 cells, NKT2 cells, and NKT17 cells. And which is very unexpected and interesting for us is that when you have the soluble glycolipid or the glycolipid is the same glycolipid, there are different glycolipids here, but containing to liposomes, the distribution of the differentiation of the NKT cells is so different. And we came out with a regime. This regime I can show you because this is something actually we are working in some of these PI protected grants, but we were able also to induce a great expansion of the cells called NKT10 cells. These cells are very important because they secrete large amounts of IL-10, and they can be used for purposes induced intolerance, and we are using this for treatment of allergy. As you can see here, there are these 3 glycolipids. They induce very low amounts of NKT10 cells, but when they are contained into liposomes, you can have here up to 30% of induction of NKT10 cell, which is very interesting. So we were also interested with using these liposomes to see how is the interaction between B cells and NKT cells? Because in most of the immune response that involve antibodies, the human immune response, you need the help of CD4 T cells. You have an antigen here. They contain an antigen that activates CD4 T cells. The same CD4 T cells is being activated by the dendritic cells, and then this antigen-specific conversation will enhance and will actually change the isotype, will be involved in the germinal center formation, etc. So we wanted to know whether NKT cell can surrogate this and how it can affect this by using different glycolipids. And this is just one review that we got a couple of months ago that we just gathered some data that we have, and so for others, what happened with T-independent B cells or the T-dependent B cells and with different glycolipids. And as the hypothesis here is that there are the surrogate of NKT cells actually can have a tremendous effect of on the outcome of the difference immune response for the human immune response. As you can see here, and this is very interesting for us, is that you have this liposome containing different glycolipids. When you explore after a prime boost scheme the production of specific antibodies, and this is a liposome that actually contains ovalbumin outside attached to the membrane, and you compare what happened with different glycolipids. The glycolipids induce an enhancement of the human response in general, but also the different glycolipids that induce different phenotypes in NKT cells. You can see that the isotypes are different. The isotype is changed, which is very interesting because the isotype, for example, IgG2b is very important for viral infection. So in this case, this glycolipid could be a good adjuvant for vaccine preparations against virus, for example. And also, there are two glycolipids here that enhance the affinity, the relative avidity of the antibodies. So we also were able to detect the specific germinal center B expansion, as you can see here. And this is very interesting because compared to the most used adjuvants, actually the use of these glycolipids enhance the production of germinal center B cells, which explain actually why the affinities are a little bit higher. And also, we were able to see this phenomena with the human response are actually T-independent, with T-independent antigens. As you can see here, you can see that we can get some isotype switching to IgG1 and IgG3 when we use these liposomes. So I will finish with this because the obvious questions with these results is if there is any difference between the antigen presentation pathways when we have the glycolipids in soluble forms and in formulation with liposomes. So we really think there is something going on here. This is the most used pathways for the antigen presentation that involve CD1D. We know from before that there are some pro-inflammatory inducing NKT1 cell, for example, glycolipids. They are destined to lipid rafts. So we really wanted to know what is happening here and how this modulate the differentiation of NKT1, the NKT cells, and then actually how that can eventually have an effect on T and B cells. And for this, we need the equipment for actually quantify, but see what is happening inside the cell. So this is something that we really need to do with image flow cytometry. We actually, when we started with some experiment right now, and we're mixing flow with confocal microscopy, but to really do it in the setup that we used to do, which is in vivo experiment, these cells are really infrequent. They're very, very, very few. And the problem is the cell lines of NKT cells, they don't actually keep the cytokine signature properties that they have in vivo. So this is something that we really need to solve using these new technologies. So I have here, I think there was a message here, yeah. And I'm agree with you, Bob. Don't let technology choose your questions. Let the questions choose the technology. But gosh, having the technology open our mind to new questions. So there are really questions that you can even think about if you have the technology. So this is my current lab, the NKTs, natural killer thesis guys. And well, of course, extend a very important network of collaboration. I'm happy to take questions. Thank you very much.
Thank you. Do we have any questions from the audience? Maybe we're all hungry.
I'm sorry. I was wondering what kind of antigen presenting cells can express this type of um, no, this type of antigens, those glycolipids, specifically, I want to know what other type of immune cells can present in CD1D, those
That's a great question. Actually, most cells express CD1D in, actually CD1D expression is at low levels. So dendritic cells and B cells are the ones that express the most, I would say. Years back, there was this hypothesis that anti-inflammatory Th2-like glycolipids were presented by B cells. Dendritic cells were the responsible to present the Th1-like glycolipids. And actually, I had the opportunity when I joined Steve Labs that we actually I was involved in a really great paper in immunity that showed that actually are the dendritic cells that the ones that are the most important CD1D presenters for NKT cell activation. The difference is that the destination of lipid rafts is different, and also actually it's not every single dendritic cells. They are the CD1Cs. And that time, that nomenclature wasn't there. It was just CD8 alpha positive cells. But to be very honest, there's some evidence right now showing that probably NKT10 cells or NKT17 cells, they would be involved other cells. So actually something that we want to explore. We want to explore the difference between antigen presentation in dendritic cells by soluble forms or nanoparticle-based form, but also we want to relook at the antigen presenting cells, because maybe for this case on the nanoparticles that could change. The liposome is, the glycolipids, it's the self, they have some problems with solubility. It's not that easy to inject an animal with a glycolipid. You need to do it right, actually, because they are, they are hydrophobic. So maybe that could explain that too, because dendritic cells actually are better to do that job than other cells.
Thank you for your presentation. I have a question. Nowadays, treatments for some disease prefer to use immunomodulation rather than immunosuppression because of the benefits for patients. And you talked to us about how the immune cell receptors can be changing along the time because they have a really strong plasticity. So it could be a problem for the future that these immunomodulators, I mean, I'm talking about drugs that binds to a specific receptor. So if the receptors are changing, could be a problem that these drugs will no longer be effective for treatments?
Yeah, that's a great question. It could be actually for NK cells. You don't see a lot of therapies that actually direct NK cells. So where we see that it might, they are not the only cells. IL-6 actually, I think they played a extremely important part on this, and we just don't understand how they got there because they don't have TLRs, example, etc. But for NK cells, for example, that the part of the trained immunity, that the so-called trained immunity, is because they change the diversity of the receptors that they retain. That's why actually in my lab we like NKT cells. So NKT cells, they undergo an expansion and they differentiate, and then they go and go back to normal levels. I mean, as far as we know. Of course, with this new technology, maybe something changed there. But if you pick up a naive animal, for example, and you explore how they're the distribution of NKT1, 17, 10 in normal conditions actually is quite similar in the time. So, Bob, you have a question?
I just had a question because it's not my field exactly. Are APCs impacted negatively in their function with telomere length? With telomere length. So the telomere length inside there, does it impact APCs?
Yeah, so the question by Bob is that if the APCs are impacted by the telomere reduction, right? That's the for the sense. Yeah, I don't know. Yeah, I wouldn't say. I don't know. Actually, it's not here. So Lucas, he's a not second year PhD student in my lab. He likes senescence, so he's studying senescence. I'm sure he would know, but I don't know. I'm sorry.
Thank you very much, Dr. Carreño. So we will now go on a break of 10 minutes and see you, okay, in 10 minutes. Nos vemos en 10 minutos. Todos los que siguen están online, ¿cierto?
Sí.
Ya.
¿Seguro?
Yo encuentro que es lo que corresponde. ¿Partimos? ¿Sí? ¿Partimos? Okay. So to begin this session, we welcome Ms. Cheryl Kim, Flow Cytometry Core Director of La Jolla Institute for Immunology of the United States of America.
Thank you so much. I hope you can hear me okay. Does it sound okay?
Yes, we do.
Okay, great. I'd like to thank the organizers for inviting me to present at this wonderful symposium and for moving me up in the schedule so that I can do so. So to just give you a little background, I've been in the field of cytometry for over 24 years and at the La Jolla Institute for Immunology for over 18 years. My focus, like many core managers, is evaluating new technology and new instrument implementation. I'm also involved in cytometry education and training and help organize community and networking events. So for today's presentation, I'll briefly introduce you to our institute and my core facility. I'll take you through our timeline of high parameter implementation at LJI with a focus on cell sorters specifically. The majority of my talk will be about our experience as the last BD Biosciences Center of Excellence and the rare opportunity to evaluate the FACSDiscover S8 cell sorter. A few experiments, projects that I'll share with you. First will be an instrument comparison between spectral and conventional using an optimized in-house core panel. Second will be an instrument comparison sorting rare precursor B cells in non-human primates, something that we do quite regularly. And lastly, as Bob presented earlier, one of the major applications that can benefit from spectral and real-time imaging, the study of cell-cell complexes. LJI was founded in 1988. We're an independent, nonprofit biomedical research institute, and as many of you may know, a world leader in immunology research. We have a total of 21 principal investigators, 8 adjunct professors, and around 500 employees, all centralized in one building. We have many collaborative partners and affiliations, including Kyowa Kirin, the Sanford Consortium for Regenerative Medicine, and the University of California, San Diego. We have 4 research centers at LJI: autoimmunity and inflammation, cancer immunotherapy, vaccine innovation, and the Center of Sex-Based Differences in the Immune System. We also have 9 scientific core facilities. In the last few years have added histopathology and immunometabolism. LGI researchers are super fortunate to have so many resources available to them, including flow cytometry. And they may not realize it until they actually leave and move on. But surprisingly, many scientists actually come back as external users if they stay in the area. So we're one of the largest flow cores in San Diego. There are a total of 7 of us staff that keep the lab running. We manage 16 instruments, and luckily we're centralized in one large space with just a little more room for expansion. We support 20 out of 21 internal labs, and that's just because the one lab that doesn't use this is mainly computational. We support over 30 external labs with over 250 users. We have a wide variety of sorters and analyzers from both BD and SciTech. We also provide the site license for FlowJo and omic analysis software and recover this through recharges. So just to mention a few key achievements, we were awarded 2 shared instrumentation grants from the NIH over my time there. We were also awarded a $1.17 million grant by the Conrad Prebid Foundation that we used towards instrumentation a few years ago. We collaborate with industry leaders on novel technology. We've been a beta and early access site. We implemented the first BD Fax Melody, which is a user-friendly cell sorter. We implemented the first BD Fax Symphony S6, which is a high-dimensional sorter, the first one in the US. We purchased the first SciTech Aurora CS full-spectrum cell sorter, And again, I'll talk about us being the center of excellence in 2024. Here are all the instruments in our core facility with varying configurations and capability. In addition to the core instruments, 2 labs of their own, Dr. Crotty and Dr. Setti, have their own analyzers upstairs. Between them, they have 7 analyzers. So we have 7 analyzers in the core and 9 cell sorters as well. So our first high parameter sorter was purchased in 2019 as an early access unit. This was a joint purchase by 5 different PIs, including a partial award to Dr. Shane Crotty from the Gates Foundation. The Fax Symphony S6 is a conventional sorter that we configured with 5 higher powered lasers, and it can detect up to 28 colors. Alba Grafoni, who will be presenting soon, published a seminal paper in Cell in 2020, identifying the target regions of SARS-CoV-2 and the importance of T-cell cross-reactivity to the common cold virus. This was the first publication ever that used Fax Symphony S6, where a 21-color panel was used to assess the immune cell profile in patients recovered from COVID-19. This was referenced by Dr. Fauci during a congressional hearing, pleading for more funds towards COVID research. And since then, we've purchased 2 other Fax Symphony S6 cell sorters with the same configuration that gets used on a daily basis. LGI received many grants and philanthropic support during the pandemic, and we have been fortunate to have purchased 7 Aurora analyzers at LGI total. Dr. Mateus, formerly with the SETI lab along with Alba Grafoni, published one of the first papers out of LGI that uses full spectrum cytometry for the AIM ICS assay, which Alba will present more of her work in more detail. In 2021, Cytech developed a full spectrum cell sorter, the Aurora CS, and Dr. SETI was awarded a gift from an anonymous donor that was used towards the purchase of the CS. The CS has the same configuration as the Aurora analyzer, making a seamless workflow from analyzing these AIM-positive cells to sorting them for 10x single-cell RNA-seq. So last year, we were designated a BD Center of Excellence. And what does that mean exactly? This was a mutually beneficial agreement that gave us access to the FACSDiscover S8 for a 9-month period. We were able to evaluate this new technology and had direct access to many of the resources at BD without paying the large price for it. For BD, this gave them the opportunity to interact with our core users directly on LGI applications. We also provided a lot of feedback on hardware and software. We served as a demo site for the community and hosted several educational events at LGI last year. Bob presented a way more exciting animation that showed how imaging detection works. But for those of you that are just joining in, the S8 uses BD CellView image technology. The primary 48 nanometer laser is split up into 2 different optical paths, one for imaging and one for spectral detection. And for imaging, an acousto-optic deflector is used to split the laser into an array of beams, where each is modulated at a unique frequency. And the waveform is decoded to provide fluorescence and spatial information. And real-time images can be derived without the use of a camera. And this can happen just as fast as conventional sorting, up to 9 to 10,000 events per second. So for those of you that have experience with BD instruments and FACSDiva software, although our work is serious, we thought it'd be fitting to name all of our cell sorters after divas with matching personalities. So we have their pictures posted by each of these instruments, and just as divas have good and bad days, these instruments can certainly act up. And so we wanted to pick the perfect diva for the S8, and it was a unanimous decision. Our S8 was called Beyonce. So our first instrument comparison experiment was conducted by Denise Hines on my team. She's a scientific lead. The goal was to evaluate and compare all 3 high parameter cell sorters using an already optimized 19-color in-house immunophenotyping panel. And for this experiment, 3 healthy donors were used. Reference controls were optimized on the Aurora, and the same controls were used for the S8 and the S6. We used the QC optimized settings that were on the S8 and the SCS, and Voltration optimized PMT settings were used on the FACSymphony S6. This 19-color panel is an in-house panel that provides a convenient and cost-effective way for our core users to test and get preliminary data for grant submission without having to redesign a panel. So Denise optimizes panel for both the CS and the S6. This panel can identify and phenotype up to 10 different immune populations based on surface expression without using any of the exotic dyes that have recently come out. So here is the initial cleanup gating strategy used for all the populations that I'll be showing you next, where each donor is shown across all 3 cell sorters. So the S8, CS, and the S6. So this is showing CD4 and CD8 T cells. This is CD4 memory and naive cells. This is for B cells, memory and naive. Basophils. And all the monocyte subsets, so non-classical, intermediate, and classical. So in summary, the panel optimized on the Aurora and S6 could also work on the S8 with basically no modifications. The reference controls and compensation controls that were optimized on the Aurora and the S6 could also be used on the S8, and population resolution is pretty comparable between all 3 types of technology. The population frequency was similar enough with a few minor differences in basophils, naive B, and the non-classical monocytes. The next instrument comparison experiment was to evaluate and compare the cell sorting capability of all 3 sorters. So this was conducted by Monalina Schill in my lab, who also has a joint position working with Pat Madden, the postdoc in Shane Crotty's lab. One of their main projects is working on HIV vaccines and assessing different boost conditions in 9 non-human primates. And so they regularly sort these precursor B cells that are in PBMCs. For this experiment, an 11-color panel that was optimized for the S6 sorter was used. Fluorescent antigen probes are made by combining fluorophore conjugated streptavidin with biotinylated trimers. In a normal workflow, the samples are usually hashtagged since these cells are really rare. They're sorted and pooled for 10x single-cell RNA-seq. So for this experiment, the PBMC from NHP was stained with this 11-color panel and split up between the 3 sorters. We used the 100 micron nozzle, which we would usually use the 85, but our Aurora CS was still awaiting upgrade at this time. We used the sort mode, the purity mask for sorting. And in the same experiment, it was a quite large experiment. Monalina tested different reference controls, testing different probe iterations. And then the 2 populations that were sorted were these double probe positive cells. And then we also took a QC population, which is usually taken during these sorts, just to assess purity and viability before the 10x workflow. And then we usually do a purity check of the QC population just because this probe positive population is so infrequent. So here is the cleanup gating strategy that was used. And on the S8, we're able to detect the doublets by imaging, as Bob pointed out. And so these results are pretty comparable. And then just to show you the post-sort purity, so this is the pre-sort sample sorted on all 3 cell sorters. And then this is the post-sort. And again, we took the QC population just because this is like less than 500 events that were sorted. So in summary, the panel optimized for the S6 can also work on the CS and the S8 with relatively little modifications. The population resolution is comparable between all three sorters. The sort purity was mostly comparable. There were some discrepancies with the population frequency and the sort yield. But this was also due to the fact that the sorters were set up consecutively by different people. And also the amount of pre-sort sample that was used for the instrument was variable. And so these are the stats that came out. I left out the CS because it wouldn't be a fair comparison. There was more pre-sort sample that was used for the reference control optimization. And so these are the numbers that came out. And a few months ago, Monolina was able to repeat the same comparison experiment without the S8, but we couldn't fit the repeat experiment before the S8 left. But comparing the CS and the S6, I'm not showing you the results. We used the 85 micron nozzle, and the sort count and process events were almost identical from these two sorters. So for the last section, I'll discuss the projects that took up most of our time with the S8 and using the imaging capability to detect immune cell doublets in human blood. So this work was done by Julie Burrell, senior staff scientist in Dr. Bjorn Peters' lab. She recently presented this work during a webinar a few weeks ago and presented a poster at AAI just this past week. So biological immune cell doublets are pairs of two immune cells of similar or distinct lineage that are physically in contact. In single cell technology, they're usually considered technical artifacts and excluded from any data analysis. So in previous studies, Julie has shown that doublets fail to be categorized by conventional cytometers, contaminating single cell data. The imaging parameters are best features to distinguish doublets. So as someone mentioned earlier, the Amnis image stream was the work that was done here. And that a significant fraction of T-cell monocyte doublets in human blood show biological relevance during infection, inflammation, and vaccination. So there are protein, RNA, TCR signatures that show immune interactions. So Julie's focus has mainly been on T-cell monocyte doublets. And with the S8, Julie was able to further characterize these cell-cell complexes. So here's the cleanup gating strategy used on the S8. And using the side scatter imaging parameters, doublets can more accurately be detected. And so this CD3 positive, CD14 positive was put into flow-sum clustering and was done using all the parameters, including these imaging-derived parameters, along with manual analysis. And so this new gating strategy was established to identify the different T-cell monocyte phenotypes. So here are all the different T-cell monocyte doublet phenotypes using real-time imaging with the T-cells in green and the monocytes in red. And so this all comes from this double positive gated population that looks homogeneous when looking at the non-imaging parameters. But now we're able to even further divide these synaptic interacting cells. So this shows you coincidental, so in the same plane of view. And so they're further apart. And these three synaptic subsets were identified based on CD3 expression, either dim CD3 or bright CD3. And then this correlation high positive population looks like the cells appear to be in closer contact. And so these doublets were sorted from tuberculosis patient samples and used for functional characterization by single-cell RNA-seq. We also wanted to push the limits of the S8 and take full advantage of the spectral and imaging capabilities. So currently, as Bob mentioned, there's no compensation available between the imaging parameters, making it difficult to use all three channels together. In addition, you have to avoid using fluorochromes on the spectral side that could potentially overlap into the imaging detectors. But with the emergence of new dyes for spectral cytometry in the last few years, we were able to successfully develop a 22-color panel, including all major immune cells, to come up with an immune cell doublet atlas in the human blood. So we use these three imaging parameters. So T cells are in green, monocytes are in red, and B cells are in blue. And as you can see, there are many different types of doublets: T cell monocyte, T cell, B cell monocyte. And these other cells that are categorized, we can use the non-imaging parameters to identify what type of cell that is. And as you see, there are also doublets comprised of two cells of the same type, so T cell, B cell, and monocytes. So this data still has to be analyzed. So I think we've accomplished quite a bit in the last 9 months. Usually implementation for new technology takes at least 6 months and up to many, many years. And we were literally cramming every experiment in the last few months, of course. We ran the last experiment the day before it was deinstalled. So in summary, here are the 3 high parameter sorters that were used for evaluation and comparison, along with their technical specifications and capability. So the FACSDiscover S8 uses imaging and spectral. Aurora is spectral. The FACSymphony S6 is conventional, but can certainly be upgraded to spectral. They can all be configured with up to 5 lasers. And just to note, the FACSDiscover S8 doesn't have the 70 micron nozzle, just because the imaging component needs slower velocity to capture the images, but we regularly use the 85 micron on all of our sorters. And they can all sort up to 6 ways. And software-wise, the FACSDiscover uses a newer software called FACSchorus that's more user-friendly and wizard-guided, but it is a little less flexible. You can also use the BD Research Cloud to design panels. And on the Aurora side, this uses SpectraFlow. It's user-friendly and ultimately flexible. You can also use the Cytec Cloud to create experiments and import them directly in, and also has a seamless workflow from analyzer to sorter. And then the FACSymphony uses FACSDiva that's been around for many decades. It's fully manual, but it's also, we know all of its quirks if you've worked with the technology for so long. I also put here that service contract costs are quite a bit, especially for the FACSDiscover S8 and the S6, but we are very lucky and we get VIP service for service. And I thought it would be entertaining to show you the very first cell sorter that I worked on back in 2001 at the Scripps Research Institute. This was an analog system that used oscilloscopes and these huge water-cooled lasers that are back here. And we could only detect up to 4 colors at this time. So I think we've come quite a long way. In closing, I'd like to acknowledge my team in the Flow Core, Julie Burrell for all the work done on the S8, all of our collaborators at BD who provided us with support and advice throughout this whole process, especially our COE team that made this all possible, and our Cytec team for their constant Aurora support, and our funding sources that have given us the opportunity to stay cutting edge. Thank you for your time.
Thank you very much, Ms. Kim. Is there any questions from the audience?
Thank you, Cheryl. Alexis Calar is here. Very nice to see the last slide because I used the same machine when I was doing my PhD thesis, that sorter in 2000. So that was funny. I also want to say that one of our students will probably be working with you as a collaboration we have with Alva and Alessandro.
Okay.
So they are thinking on experiments that they will probably learn to do here because we also have an S8 and they will just probably apply with you over there and learn from you. Thank you so much.
Great, thank you.
Thank you very much for your talk. It was very interesting to see the comparison between equipments. And I wanted to ask a question about that point, that experiment. So when you identify each cell population, you barely don't see any difference between equipment, but did you look at, for example, cell viability or cell activation? How long do you take to get some, for example, 100,000 cells in your tube? Is there any difference in that point of view between different equipments?
Okay, are you talking about the sorting experiment or just?
Yes. Yeah, exactly.
Yeah, so actually they're comparable when you use the same nozzle sizes because the same frequency or around the same frequency is used across all the instruments. And so speed-wise, as long as the frequencies are somewhat close, then yield-wise, it actually ends up being pretty much the same. And so as long as you keep the speed consistent and the efficiency is running around the same percentage, then we expect the same yield. And so for the rare cell populations, as you see, the difference between gating like 0.006 versus 0.003 is quite significant. And so usually on a normal day when we sort side-by-side machines, they usually want to use the same technology. So say if they're running a big experiment, then they'll book 2 of the S6s, and we have like a standardization protocol that we follow that actually yield very similar results. So I'm not sure if that answered your question.
So I have another question. Thank you very much, Cheryl, for your presentation. So this might come out of the bloom because you did not mention it in a specific, but I wanted to know your take on populations that come up when you do analysis, multiparametric analysis, non-supervised, when you have all these populations that aggregate as discrete populations, but they're currently not identified as such. Do you think those are populations, or are they yet to be identified? Do you have any methodology you think in the future could be used to find functional aspects related to those populations?
Yeah, that's really tough. I heard that question earlier. And so you still have to use other technology to confirm the results. And so the project that Julie actually started was because this technical artifact was showing up in the data. And so it kind of led her to further look into these cell doublets. So unfortunately, this costs a lot of money, right? So like sequencing would be the thing to do, and not many people have the funds to do that. So you still have to use other technology to confirm these results and to see if it's a real population by microscopy, but pretty much single-cell RNA-seq is the way to confirm these results.
We have one last question.
Thank you very much, Ms. Cherry. I'm Daniela. I'm PhD student, and I would like to know what type of analyze do you recommend us for presentate all data we can obtain of the Discover ACE because we have obtained many data.
Are you asking what analysis package, or I'm sorry, I didn't understand the question.
Yes.
Oh, analysis package. I know, there are a lot of these automated algorithms that people have mentioned. And so most of our users or some of our labs actually have bioinformatics support. But for people that don't have access to that, OMIC seems to be more user-friendly. FlowJo is definitely becoming more of an easier workflow. And so in version 11, I think that if you have access to FlowJo, or do you have any data analysis software that you can use?
FlowJo. Yes.
Oh,
We
FlowJo.
have FlowJo. Yes.
Yeah. So if you just wait for the latest update, which is version 11, then all the tools are integrated within. So right now in version 10, you have to use a plugin, which makes it like an extra step to do this analysis. But in FlowJo 11, they'll have many more tools that are integrated. I hope that answers your question.
Yes, thank you very much.
Yeah, no problem. But you
Thank
can
you very
also,
much, Ms. Kim. I'm sorry.
yeah, no problem. You can also look at OMIC too, which is not as well known as FlowJo. But for these more complicated algorithms, it makes the workflow quite easy.
Thank you.
Yep, no problem.
We now receive Dr. Denise Moraes from the Institute of Biomedical Sciences of the University of Sao Paulo, Brazil.
Oh, good afternoon or good evening, everyone. I'm sharing my screen. Can you see it?
We can, yes, we can now see it.
Is that okay?
Yes.
Okay.
Yes, it is.
So thank you very much. First of all, I'd like to to thank Professor Alexis Kalergis for the invitation and also to congratulate for the amazing meeting. It has been really good. And today I'm gonna talk a little bit about how we have been using flow cytometry in my lab to answer some immunology questions. So I have been the coordinator of the flow cytometry facility for a few years. I'm actually the former coordinator, so I left the coordination, but I have been coordinating the flow cytometry facility here for over the past 5-6 years. So when I started, we used it to have a very basic core. So we had the basic analysis machines like Canto, Fortesa, then we acquired the Symphony machine. We have fax area sorters. And over the past 2 years, we really upgraded our facility. So we were the first lab in Latin America to acquire the BD Discover S8 sorter. And now we have the S8 sorter. We have the Symphony SE. We have the Aurora Cytec spectral flow cytometer. And we are now moving towards the spectral technology. But most of the data that I'm going to present to you today, it's a work that we're finishing for publication. So we are using this work mostly the conventional traditional flow cytometry, except for the sorting that we made mostly on the Discover S8 fax machine. So today I'm going to talk, I'm going to move towards the other screen. So I'm going to talk about the impact of the gut microbiota dysbiosis on the function of T gamma delta cells and consequently on the neutrophilic inflammation in the lungs in response to ketogenic diet. So over the past few years, we have been learning a lot about the concept of the human meta-organism. So this concept says that we are not alone. So we share our bodies with millions, trillions of microbes, bacteria, fungi, viruses, and even archaea. And all of these microbes that are occupying our bodies, they are not just using our body as their homes. So they actually share with us very important functions, and we can also get a lot of benefits for host physiology from these microbes. So they are essential for the nutrient absorption. They can help metabolizing and synthesizing vitamins, bioacids. They help in the short chain fatty acid metabolism. They help in the production and regulation of neurotransmitters. And also they can impact cognitive function, for example. But today I'm going to talk about how microbiota nutrients can have an impact on the immune regulation, not only locally, where they're mostly localized in the case of the diet, in the gut, but they can have long distal side impacts. And why this is important? So we know that our barrier tissues and our mucosal tissue, specifically the gut, they are environments that are constantly exposed to antigens. And most importantly, those antigens, they share the exactly same physical localization. So dietary, commensal, harmless antigens are sharing the same localization as antigens coming from the pathogen, in a way that we need to have a very specialized immune system associated to these barrier surfaces in order to promote inflammation when I have a pathogenic challenge, and this inflammation needs to be just enough to control the pathogen, and the immune system needs to restore homeostasis as soon as possible to avoid the breakdown of immune tolerance. And in our lab, we are interested in understanding how these challenges can break down the immune tolerance and prevent the immune system to restore homeostasis. So in this way, we try to understand how environmental challenges such as infection, dietary changes, microbiota changes can interfere in this mucosal-associated immune system and prevents the restoration of homeostasis, driving the development of several immune-mediated diseases such as allergies, inflammatory bowel diseases, failure to respond to oral vaccination, and even metabolic syndrome. So to understand this, we first need to understand how the immune system works in the mucosa, and specifically in the gut for this talk. So we have three main layers of immune cells of barrier immunity in the gut. So we have the first physical layer that contains most of the epithelial cells that produce antimicrobial peptides, mucus, IgA that will segregate the commensals and pathogens from the host. Underneath this first physical layer, we have an active layer that contains the immune cells. So we have mononuclear phagocytic cells, macrophages, monocytes, dendritic cells that are going to be able to instruct the activation of the proper immune cells in the gut mucosa, such as Th17, Tregs, IgA producing B cells. We have a third layer of specialized lymphoid tissue, such as driving mesenteric lymph, tissue associated mesenteric lymph nodes. And on top of it, we have the microbiota that's shaping all of these immune cells that are in the mucosal layer. So we have these three main layers of immunity that need to work in a way that is going to promote compartmentalization, immunity, and regulation of the responses every time I need each of these components. And on top of it, we most recently learned that this mucosal immune system, it's not isolated from the other barrier tissue. So there is a constant communication between mucosal and barrier tissue. Specifically for this talk of today, I'm going to talk about the communication between gut and lung. So we know that there is a constantly communication between gut and lung immune system, and on the other way around. So I'm not going to talk on the lung-gut communication today. I'm going to focus most on the gut-lung. And we know that this communication is mediated by microbiota changes that can impact the production of metabolites, the translocation of microbial components to the blood and consequently to the lung, where it can activate immune cells. It can be mediated to pathogens that can enter through lung or gut and establish this communication. It can be mediated by tissue damage caused by infection, caused by dietary changes or microbiota changes. It could be mediated by the traffic of cells between gut and lung. And today I'm going to focus on the role of dietary changes in activating this gut-lung axis and activating this communication. And why dietary changes? So we talk about diet all the time. And we have been talking a lot about food insecurity forever. So we're always talking about food insecurity. And over the past years, the face of food insecurity has been changing a lot. So we still have situations of lack of nutrients, of starvation. So we have, it's one of the aims of the United Nations Sustainable Goals. But nowadays, we have been watching the rise of this kind of food insecurity, where we have people eating unbalanced and unsafe diet that's going to have very important impact on the metabolism and host physiology. But in my lab, we're very interested in understanding the immunological safety of those dietary changes. So we have been studying very different diets. So we study malnourished diets and how can impact the immune system. We study occidental diets, high-fat diets, high-sugar diets. And for the talk of today, I'm gonna focus mostly on one of the very fashion diets that people have been used to return this situation here to the homeostasis. So the ketogenic diet or the low carb diet. So this work has been performed for Marina Caçador Ayupi, a very talented PhD student in my lab. And then when Marina arrived in my lab, she was very interested in studying this diet because when she arrived, it was like the most fashion diet of the moment. Everybody was eating ketogenic diet to lose weight in a fast way, an efficient way. And the thing is that ketogenic diet is not a new diet. So we use ketogenic diet. We have been using ketogenic diet for over at least 100 years. So or more than 100 years. So that the 1921, the physicians started using ketogenic diet and mostly for medical purposes. So it's a diet that has been used for diabetic people to control the glucose levels, to treat obesity, and to treat refractory epilepsy, mostly in kids. So the ketogenic diet has a very important anti-inflammatory role in the central nervous system, contributing for the control of epilepsy in children. But Marina decided to study how the use of this diet by healthy individual, not obese, not diabetic, and not epileptic, how this could impact the immune system. So mostly what we were doing, we want to lose weight fast, we don't really need a dramatic intervention, so we tried the ketogenic diet. So what Marina did is that she took healthy black 6 mice, no obese, no diabetic, and she treated these mice for 4 weeks with the ketogenic diet. And as expected, the mice lost weight. They showed a reduction in the glucose levels in the blood and an increase in the ketone bodies in the blood as well. So it was the ketogenic diet doing the same effect that it does in humans. And then she started looking at the gut-associated mucosal tissue. So the first thing Marina found by flow cytometry, she found an increase in neutrophils in the gut. It was a very discrete increase, so it's a very slight increase in the neutrophils, but it was there. And this increase on neutrophils was also associated to an increase on IL-17 producing gamma delta T cells, also in the lamina propria of the small intestine and not in the large intestine. Next, she went to look at other mucosal tissues. So she went to look at the immune cell compartment in the lung. And to do that, we do use flow cytometry, but we do this approach where we inject mice with anti-CD45 conjugated with FITC through the intravenous route. So we can stain all the cells that are in the blood. And then we digest the tissue, and then we stain again with another anti-CD45 labeled with another fluorochrome, so we can have the immune cells that are in the blood and the immune cells that are in the parenchyma of the lung, and then we start studying the immune cells in the parenchyma of the lung. And Marina found a very quite dramatic effect of the diet in this population here, the red one, which are the neutrophils in the gut, in the lung, sorry. So we found like a 4, 5 times, sometimes even 10 times increase in the neutrophilic infiltrate in the lungs after the ketogenic diet. And 1 week of diet, it's sufficient to promote this switch. But in this case, she was using 4 weeks. And as the same way as we found an increase on IL-17 production, we went to look at the IL-17 production in the lungs as well. And again, we found an important increase on IL-17 production by gamma delta T cells in the lung, but not by IL-6 or by CD4 T cells. So the same way in the gut, but we found a much more pronounced effect in the lungs. Neutrophil recruitment and IL-17 production by gamma delta T cells. Next, we started wondering whether these cells are migrating from one tissue to another, because as I mentioned before, the migration of T cells, ILCs, between gut and lung is one of the pathways of communication between the gut-lung axis. So to check this hypothesis, Marina is stained by flow cytometry the different gamma chains of the gamma delta T cells in order to identify if the increase on IL-17 producing gamma delta T cells in the lung was made by V gamma 1 gamma delta T cells, which are the gamma delta T cells that are from the lung, or in the gamma 4 cells, which are mostly gamma delta cells coming from the gut. And she found that the gamma delta cells that are increasing in the lung after the consumption of the ketogenic diet were actually coming from the lung and not coming from the gut. So we started wondering whether this IL-17 production by the gamma delta T cells could be mediating the neutrophil recruitment to the lung, because we know that IL-17 can recruit neutrophils from different pathways, directly or by activating stromal cells that produce chemokines that will recruit the neutrophils. So to test the importance of the IL-17 for the neutrophil recruitment after the diet, we next used IL-17 receptor knockout mice with IL-17 receptor knockout mice, and then we fed these mice with diet or not. And Marina found that in the absence of the IL-17 receptor, there was no more neutrophil recruitment to the lungs after the diet treatment, meaning that IL-17 is essential for the neutrophil recruitment to the lungs. So we know that IL-17 can act in the endothelial cells or in the stromal cells that will produce chemokines, which in turn will recruit neutrophils. So to test if this is what's happening, we decided to remove the IL-17 receptor from the stromal cell compartment. And to do this, we irradiated wild type or IL-17 receptor deficient mice, and then we reconstituted both mice with bone marrow from the wild type mice, meaning that here I have just a wild type mouse, and here I have a mouse, oh, this is wrong, sorry. This mouse was reconstituted with wild type bone marrow. And here I will have a mouse that doesn't have IL-17 receptor in the stromal compartment, but the hematopoietic cell, its compartment is normal. And then what happened is that the treatment with the ketogenic diet was still recruiting neutrophils to the lung, meaning that the IL-17 was not acting in the non-hematopoietic cells. So this was not what was happening in this case. So we hypothesized that maybe the IL-17 was working directly in a hematopoietic cell that will in turn recruit the neutrophils to the lung. So to test this, we took wild type mice. We irradiated these mice, and then we reconstituted these mice with bone marrow from IL-17 receptor deficient mice, meaning that in this case, the stromal cells will respond to the IL-17, but not the immune cells. And then after treating these mice with ketogenic diet, we saw no differences in the neutrophil recruitment to the lung, meaning that the IL-17 produced by the gamma delta T cells in response to the diet is acting in the hematopoietic cells, which is in turn recruiting the neutrophils. So our next question was, is this IL-17 working directly in the neutrophil and recruiting these neutrophils, or is this IL-17 acting in another cell that will in turn recruit the neutrophil? So to test this, we made next bone marrow mixed chimera, where we took a wild type mouse, we irradiated them, and we reconstituted this mouse with half bone marrow from IL-17 receptor deficient cells and half bone marrow with wild type cells, meaning that these cells here, they are wild type, and these immune cells here, they don't have IL-17 receptors. Then we treated these mice with ketogenic diet, and then we went to look at the neutrophils in the lung of these mice. And we found the similar proportion of neutrophils between wild type neutrophils and IL-17 receptor deficient neutrophils, meaning that the IL-17 is not working directly in the neutrophils and recruiting them. So probably there is a mediator in the between that will induce the production of a chemokine that will in turn recruit the neutrophil. And one of the most important cytokine and chemokine that are involved in the neutrophil recruitment to an inflammatory microenvironment is the IL-6. So our next question was to test the contribution of the IL-6 to this phenotype. And indeed, if we treat mice that are deficient on IL-6 production, they don't have the recruitment of neutrophils through the lung, meaning that the IL-6 is essential for the neutrophilic recruitment to the lungs after the treatment with ketogenic diet. But mostly interesting, most of the time we see the IL-6 here in the beginning of the pathway, in the differentiation of the Th17 or the T gamma delta 17 cells. But in our case, the deficiency of IL-6 didn't impact the IL-17 production by the gamma delta T cells, which means that probably the IL-6 is in another step of the pathway. So it's probably we have the diet promoting the IL-17 production by the gamma delta T cells that will in turn activate another cell that will produce the IL-6, and these IL-6 will recruit the neutrophils. So we went to look which cells were making IL-6 in this context. And after doing flow cytometry as well, and here again, we look at for the cells that were in the vasculature and also in the parenchyma, and we found that in the parenchyma of the mouse, after the treatment with the ketogenic diet, there was an important increase in the production of IL-6 by inflammatory monocytes, as you can see here and here. So we have twice as much IL-6 production and also IL-1 beta production by inflammatory monocytes in the lung parenchyma after the ketogenic diet. So what we think is happening so far is that somehow the diet is activating gamma delta T cells. We don't know how the diet is doing that yet. And these gamma delta cells, they will produce IL-17, which will activate inflammatory monocytes that will produce IL-6 and IL-1 beta, which will recruit the neutrophils. We cannot exclude so far that the diet could also drive somehow the production of the IL-6 without the IL-17. And to test this, we started exploring several different mechanisms. So we were analyzing how the metabolites derived from the diet can work on this process, or how microbiota-derived antigen. We still didn't finish, so this is a work in progress. But we started asking what could be driving both IL-17 or IL-6. So how the diet could trigger the IL-17 production and the IL-6 production in the lung. So we, first of all, hypothesized that maybe microbial components could be involved because of changes in the microbiota composition. So to test that, we used MYD88 deficient mice. And when we treat MYD88 deficient mice, we don't have the neutrophil recruitment to the lung. I'm not showing here, but we also don't have the IL-6 production by the inflammatory monocytes, and we also don't have the gamma delta 17 producing IL-17, meaning that probably there is a microbial component involved in this process. So of course that the next step was to sequence the gut microbiota, and as expected, there is a huge change in the composition of the gut microbiota after the ketogenic diet, mostly in the small intestine. And then we see the increase, the rise of more pro-inflammatory species, the decrease of species that are more anti-inflammatory in the gut. Then we went to check whether this change in the gut microbiota could be responsible for the phenotype. So to do that, we used germ-free mice. So we treated germ-free mice with the diet. And then we found that in the germ-free mice, the diet did not increase the recruitment of neutrophils to the lung, meaning that this phenotype is microbiota dependent. So we went to look what was promoting this effect. So we went to do fecal matter transplantation to these mice. So we took white SPF mice, we depleted the microbiota of these mice, and then we reconstituted these mice with microbiota from the ketogenic treated mice. And then we found that the microbiota from the ketogenic treated mice was not sufficient to promote the neutrophil recruitment to the lungs. So they need to have the ketogenic diet. So here I have just the ketogenic diet without the fecal matter transplantation. Here is just mice that received the fecal matter transplantation from ketogenic diet treated mice. And these are the mice that have fecal matter transplantation and ketogenic diet. So we need to have the diet to have the neutrophil recruitment to the lungs. So we need the microbiota. So the phenotype is microbiota dependent, but it's not microbiota specific. So I cannot reproduce the phenotype just by transferring the microbiota. And the same goes for the IL-17 production by the gamma delta T cells, and also for the IL-6 production by the inflammatory monocytes. So the phenotype is microbiota dependent, but not microbiota specific. So we went to check whether we could have the translocation of microbiota components to the other tissues that will therefore promote this systemic inflammation. So we checked for microbiota transportation in the portal vein blood, in the liver, spleen, mesenteric lymph node, and we also checked for the gut permeability, and we saw no microbiota translocation and no increase in gut permeability in the mice treated with the ketogenic diet. But for our surprise, when we culture the lung tissue, we saw a huge change in the composition of the lung microbiota in these mice. So as you can see here, there is an increase in density and also in the species that are growing in the lung from the mice eating the ketogenic diet. And when we sequenced this microbiota, we found a big change in the composition of the microbiota of mice eating ketogenic diet. So they have a change in the composition of this microbiota. I'm sorry, it's switched here. So they have more proteobacteria, they have more Enterobacteriaceae, suggesting that somehow the ketogenic diet is changing the composition of the lung microbiota. This microbiota from the lung, not from the gut, is activating either the inflammatory monocytes or the IL-17 producing gamma delta T cells in an MYD-dependent pathway. And these cells are producing IL-17 and IL-6, which will recruit neutrophils to the lung. And to finish, our final question was, so what? What is the functional impact for us, healthy human beings eating ketogenic diet? And at that time, the COVID pandemic was just starting, and we were seeing that people eating ketogenic diets and having more ketone bodies in the blood, they had more severe diseases. So we tested whether these mice could be more susceptible to the development of pathology driven by infection. So we used we also tested, not tested, but we also evaluated in patient based on the dietary forms that they fill it up. And we also tested in hamster and in mice infected with the SARS-CoV-2. So we gave mice the ketogenic diet, we infect them with SARS-CoV-2, same with hamsters. And we found that these mice, they had no change in the weight gain over time, no change in the viral load, but they had much more morbidity. So they died over 1 week of infection, all the mice eating ketogenic diet. They had a more severe score of disease, and they also had much more inflammation. So they had an increase in IL-1 beta production detected by flow cytometry. They had an increase in IL-6 production, and they had much more inflammatory infiltrating to the lung. So in conclusion, the change in the diet, more specifically in this case, the ketogenic diet, can change not only the gut, but the lung microbiota. We still don't know how this diet is changing the lung microbiota, but it's in a gut microbiota composition dependent way. And this change in the lung microbiota can activate the production of IL-6 and IL-17 that will recruit neutrophils and make the tonus of this tissue more prone to immune pathology mediated diseases. And to finish, I'd like to thank all the people involved in this work, especially Marina, Clara, Amanda, Leo, and Marcelo, which were the people doing the experiment, and our collaborator, Tim Hand from Pittsburgh, who helped us with the germ-free experiment, Marco Vinolo from Unicamp, José Carlos, who gave us the transgenic mice, people working on the hamsters, and our two core cytometry facilities, so the flow cytometry facility from Cefapi and the flow cytometry facility from our department, we have all the equipments that allowed us to do these experiments. And thank you very much for your attention.
Thank you, Dr. Moraes. Are there any questions?
Thank you very much for your talk. This is Susan Bueno here. Regarding the last association you were doing, it seems like probably the requirement of the microbiota changes and also the ketogenic diet to promote the increase in IL-6 and also the neutrophils maybe is related with the metabolites produced by the bacteria, right? That are fitting with the lipids or the components of the ketogenic diet. So I was wondering whether you are addressing some of these.
Yeah, we're starting to do some metabolomic analysis in the blood of these mice to see what is increasing. I can tell you so far that we found a change in the short chain fatty acid production. So this is for sure changing. I don't know whether this is a consequence on the change of the microbiota, or if this is, or whether these short chain fatty acid are actually acting in inducing the IL-17 production by gamma delta T cells. So we started doing very simple experiments, just checking whether the serum from the mice eating the diet can transfer the phenotype. We still don't have conclusive results. And another thing that we are addressing is that we are trying to isolate microbiota components from the lung, and we're trying to do a lung microbiota transplantation to check whether specific microbial components from the lung can transfer the phenotype. So these are the two ways that we're trying to approach this now. But for sure, I think there is a metabolite, probably a phospholipid-based metabolite because of the gamma delta cells. So this could be activating the gamma deltas. But we still don't have anything conclusive so far.
Hi, Denise. Thank you for your talk. Hernan Peñalosa here. I want to focus on the neutrophil side of your talk. And I have two questions. The first one is, did you evaluate whether just the keto diet increase neutrophil activation at baseline?
What do you mean by baseline? Just comparing neutrophils?
Just the diet, not any infection or any inflammatory stimuli.
So I look at for nets, and I look at for surface activation markers. There was no changes. We started wondering whether these are neutrophils or myeloid-derived suppressive cells. So we are now sorting these cells and doing suppressive function to check whether these are real neutrophils. We run these cells in the Discover, staining them with DNA stain to look at the nuclei to see if they are polymorphic nuclei, but they are not classically activated neutrophils. So they are weird looking like neutrophils. They could be myeloid-derived suppressor cells. I don't know yet. Did I answer?
Yeah, and that it comes my second question. So you use a viral challenge to when you compare the immune response between keto diet and control diet. Have you looked using a bacteria? Because extracellular bacteria, there are neutrophils are very important to fight cellular bacteria, and you can see the role of these cells in that model where they are actually participating in pathogen clearance and not just inflammation.
This is a great question. We are doing the bacteria right now. So we are doing Streptococcus pneumonia. We are just testing the model. What we did, that it's a bit counterintuitive, but I wanted to see, we did asthma. And this is why I'm looking for the suppressive capacity of those cells, because at first we thought that if we induced asthma in these mice, they would have a more severe neutrophilic form of asthma, but actually they are super protected from asthma, so they don't develop asthma at all. Even if I stop the diet, wait for 2 months, and then induce the asthma, they don't develop asthma. And another thing that's important, these neutrophils, they don't go away from the lung parenchyma. So I can stop the diet and the neutrophils are still there. This is why I think it's because of the microbiota change. But we are doing the bacterial infection right now, so I don't have the data yet. But I would say that they don't clear better the bacteria. I would say that because they have more microbiota density. So they have more neutrophils there and they have more lung bacteria there. So they're not even clearing their own microbiota. So they're not controlling their own microbiota. So I don't believe they're the classical activated neutrophils, like they're probably something different.
Denise, Alexis Kalergi is here. Thank you for being patient, and we are a bit late with the symposium because we have a very active discussion. I have one last question. Have you, at the end of the answer to Hernan, you touch on asthma, right? But I was wondering if these changes that you see with diet could influence, or have you looked at chronic inflammation models?
The only chronic I looked at was asthma. But it's the classical model of asthma, so it's not a very chronic model. We don't use the systemic immunization with ova-alum, so we do asthma by giving the mice papain and ova intranasally, or just DERP1 intranasally, and we give several doses, and they don't develop inflammation in the lungs, not eosinophilic inflammation. So the neutrophils are still there, they don't increase, but they don't develop eosinophilic inflammation. I haven't looked at other types of chronic inflammation in the lungs. So this is something that is worth trying. It's something that we want to do is tuberculosis, is TB. At some point we're going to do that, but we haven't done any other model of chronic inflammation except for asthma.
Thank you very much, Dr. Morais.
Thank you very much for the invitation and congratulations for the conference.
Next, we welcome Dr. Svetlana Masel, Director of the Flow Cytometry Resource Center of the Rockefeller University of the United States of America.
Hello, I hope you can hear me.
Yes.
Very good. Okay, first of all, I would like to say that it's amazing endeavor which you all almost achieved. And I would like to thank organizers who did plan this event, and also for invitation, which I'm very fond of. I really think that it's the great opportunity to present different approaches and different places take, and I think we will all learn a lot from today's presentations. So on my side, I am in charge of the Flow Cytometry Resource Center at the Rockefeller University, which is quite unique place, and I hope I move with slides. That's the question. Yes. So I hope it is very important to understand where we are and what we are talking about. We are talking about the place which has very flat structure. It's very advanced university, private university with the only graduate studies in place. We have 72 biomedical laboratories, which actually have broken into some research areas. And you can see that in here we have only 20 labs, which we can one way or another way, talk about immunology involved. So we have a lot of clinical research, which is part of the research we do to do the implementation into the clinics. And we have a lot of collaborations and partnerships with tri-institutional places and other places in not only in New York, but it's really very dense population of biomedical research in the Upper East Side of New York. So out of the scientific resource centers, which we have, who provide a lot of possibilities for the researchers to use advanced techniques, flow cytometry is one of these 18 centers. And we do have our website. It's internal website, but external shows almost the same thing. We try to tell as much as we can through this reach out, and a lot of information is there. We actually moved almost 10 months ago to the new place, which has been built for us after multiple years of planning and implementation, and we luckily made it accomplished. And the plan has been made to have the place which is very much compartmentalized, and we are now located in one location. Before, we have been spread quite a bit. So if we talk about the flow cytometry, you know that it has a long history, let's say almost 60 years of commercial flow. And we can say that even talking about it, it keeps moving, and the pace of move is quite tremendous. During his talk today, Bob Balderas was talking a lot through the history, and other people were touching it. I also want to say I'm in flow for many years, and I did start at Rockefeller in 2001. And during the first couple years, we did get 2 new instruments, and one of our sorters, which we had at this moment, has been upgraded with a digital software, which is DIVA, which is well known and has been mentioned already by Cheryl Kim. So as Bob referred to the things, the whole historical pathway of the flow is development of the dyes and development of the instrumentation. So we all have been through it, and we can see the things progressing. You can see I put the same slide as Bob did. And during these years, I want to show some milestones. And in 12 years from the first slide I showed, you can see that the whole landscape completely changed. None of the instruments which have been listed in 2003 appeared to be on our list for 2015. We got 3 ARIAs, we got image stream, we had 3 LSRs in Van Accurate. You can see that we have been very much oriented into the BD instrumentation, which was the reason because it was the definite leader in the field, and we strongly believe in having good instrumentation in place, which we have been always updating the instrumentation to the best and possible standards. You can see that even from this slide, we have minimum 2 lasers on Accuray, which we have been using for some screening purposes, but everything else, all the instrumentations have at least 4 lasers. All the instruments have 561 laser, and we had even couple instruments with very unique laser, which is 445, which has been requested by one of the labs to use for their CFP detection, which was extremely important. So as Bob pointed, that limitation was 18 fluorescent parameters, And we have been restricted in this area, but luckily in a couple of years, we did get the new instrumentation, which was Aurora cell analyzers. Sorry, I have the call. One sec. I'm really sorry. It was unexpected call. So we did have the instrumentation which we got, and we were very happy to have it in our hands. We are extremely happy and satisfied with the Cytec Aurora, and we got actually 2 instruments of this type. Sorry, I cannot hang up on that. So historically, what happened, the commercially available analyzers and cell sorters were mostly from BD and Beckman Coulter. After that, other instrumentation came on board, and we started considering this instrumentation to have on board. Most informative for everybody was to get the instrumentation like analyzers, Cytec Aurora ID 7000, and after that, few cell sorters came on board, and the cell sorters actually made us think where and how we will be progressing forward. Because for many years, we had the complete match between the sorters and analyzers. We did have very similar configurations of the instruments, which we were able to match the things in there. When we got Cytec Auroras, we started thinking of possibility of having the instruments which we will be able to use as a sorter. And luckily, BD Discover came on board because it gave us additional opportunity to look into the instruments with the new eyes, which will be using not only spectral, but also imaging. And we did get the Cytec BD Fax Discover S8 in 2024, which actually was very much referring to what we had before, which was the image stream, which we did stop during the pandemic, and we are using image stream at our sister facility in the Columbia University, which we did make an agreement to go with, and we are happy to use it. So we give this opportunity to our users to use different techniques, and even if we don't have them on hand, we can give this opportunity to use the close neighbors facilities around. So when we were thinking of the match of the cell sorters, we did consider having Aurora CS, and which was a very straightforward decision to go. But when we got money last year and BD Fax Discover came on board, we were thinking of possibility to expand our capabilities by using not only spectral flow, but also being able to look at the images. I will not put much time on explanation how it works, and luckily I'm at the very end of the day, which made everybody already making good background for me to present. So that's our instrumentation, which each instrument is very unique for certain things, and we always try to have possibility for many researchers to have the advantage of different advantages of the instruments. What is important when we got the new instrument, which has been installed in August 2024, it started being very much instrument for discovery. And I will not present any scientific data in here. I will talk about how shared resource laboratory with the advanced instrumentation can help people to solve the problems which they have not been able to solve before. So if you look in here, I'm talking about one case, and the case is about the Hue 7.5 cell line, which gave us possibility to look at the importance of the image-derived parameters to be used in the research. And we have been using the total intensity light loss thing to show possibility to sort cells based on the light loss. And you can see in here, if we gate for the low one, if you go gate for the high one or for the intermediate one, we get very different morphologically significant populations of cells, which we have been sorting, and we put them in culture. The users put them in culture, and they started working with this for their downstream applications. The second case I would like to show is the case of the cell line, which has been very highly expressing EGFP. And you can see, if you look in here with the original settings, there are a lot of saturated parameters, which we did do the approach to bring the saturation down, and with the gains adjusted only on the spectral channels, we decided to use the imaging channels to go deep into the granularity of the system. So what we did use the imaging channels to look at the stuff in here. And what we did, if we gate cells based and sort cells based on the image parameter 1, we are looking at the relatively low expressors in GFP. If we look in the image parameter 2, you can see the intermediate, which have been saturated in here in the image parameter 1. And if we go for the image parameter 3, which will be very far emission-wise from where GFP is, but the expression is so high that we can still distinguish and record the cells. We were able to sort them, and in the future, we plan to use this approach for the time when we need to sort cells and look at the morphology of the cells dependent on the level of expression. We think it will be very helpful in the long run for us. And other opportunity we did have, actually one of the labs at Rockefeller has been in the process of submission the paper, which actually has been, believe it or not, it has been published today. Well, it has been accepted. And after that, we did provide some additional data, which helped them to publish the paper. Over here, you can see the first picture, which is picture from the paper. It's about the B cells, and you can see even of the relatively small type of the cells, you can still see some morphological changes and approaches. And in the supportive materials for this paper, you can see more information which we have been recorded for them on our discovery site. So for sure, instrumentation is important, but the most important part of the resource center is the knowledge and the approach which we are having, connecting and collaborating with our users. It is very important not only to have advanced staff, but also to have good IT support, which especially in the situation with the site, which has a lot of issues with the data management, our IT person is extremely helpful for us to proceed. What we are doing, we are providing for many years a lot of services on our instruments. There are equipment-related services. We also move on with all the upgrades, and all of these things are happening because our users need things to be more advanced. We pay a lot of attention to the education. We do have classes going on and the consultations going on. We train people on different instruments, and after that, for the analyzers and Sony cell sorter, our users are operating them without assistance. But when we talk about more complex projects, we spend a lot of time with our users on the consultations, instrumental help, troubleshooting on the experimental design, and we also help people to do the data analysis. So what happens, we have all the booking and billing done at the FCRC at Rockefeller through the PPMS system, which is part of the StrataCore. We're extremely happy with this system. In what we're using the forms for the training requests, which you can see there are many different training requests in place for people, for our researchers to convey to us what is needed. So on our website, we talk about different training steps, and we have also sample preparation equipment and analysis workstations, which our users can proceed on and analyze the data on. So our training scheme is quite cumbersome. I will run through it quite fast, but it's a lot of work to make sure we provide the customized services. So we have couple sessions, which are group sessions to start the introduction to the flow cytometry technique. At the same moment, we meet with each person individually for the C1 experimental design consultation to look into what kind of questions people have, and we provide people with the assisted run to decide what the next steps should be done. So based on the results of the assisted run, we move on and decide if the person is ready to be sorting the samples, or the person wants to be even trained for the unassisted run. If people plan to do cell analysis, we decide based on the results we acquire by staff, we decide which type of the instrumentation will be the most efficient in terms of usage for the exact experimental project. We stopped training people on the BDLSRs because it's most likely will be used for just ongoing projects because these instruments have end of life. But what we train people on are other analyzers, 2-cell Cytec Auroras, Agilent, NovaCyte Pantheon, and Thermo Fisher Attune. We also can advise people to go and use a Cytec image stream, as I told you, at the other institution. So C1 consultation is one of the most important things which we are doing because it's one-on-one discussion with the user, what they plan on do. What happens, because biology matters, we start the consultation as collaboration between researcher and FCRC staff. So during this conversation, or even before this, we asked researchers to provide us with the following information, which is not exhaustive. We found out without this information, we cannot give good advice and proceed further. We need to have the short overview of the project to know what the goals are and hopefully understand better how we can help. Detailed Word document for the sample preparation where we can give suggestions how to proceed and what we can commonly advise people with. Reference papers are very important, and also we need to know about if somebody, co-workers or at other places, people were doing very similar experiments. Otherwise, we cannot help enough. As I told you at the very beginning, the issue at Rockefeller is that immunology is not the major approach for us, and we have very different cell types on board. We all have some immunological experience and expertise, but we cannot be experts in all biological directions. So what happens is we advise people to look at the omics, which have been published with the spectral flow cytometry. We have been looking into the huge advantage which has been taken by Cytec by changing to version 2 because new fluorochromes came aboard and better approaches were taken. BD was publishing the OMIP 102 and Cytec was publishing OMIP 109 with more colors on board. And as Bob said, that in a month or so, it will be more information about even more colors on board. But for us, we are not only using the immunological samples, and different sample types provide us with a lot of challenges in terms of how to proceed and deal with this. So we have learned a lot of information from omips and mainly from supportive materials from omips. We are suggesting people to do titration of antibodies. We found out together with our users that all the steps which have been pointed on the omips matter for the best possible outcome of the flow cytometry data. We do use this panel design tool, which has been influenced by Cytec, and we really took it into our pocket as an important tool for all the instruments we have. With the cell types we are using, the huge issue is with the high autofluorescence, and using spectral instruments for the autofluorescence extraction is absolutely essential. And what you cannot see without autofluorescence extraction, you start being able to see with the autofluorescence extraction in place. We do suggest people to use viability and vitality dyes, which is extremely important. And we have been pointing in for years. We do have this materials on our website, which actually helps people to proceed with their experiments better. And it's part of the panel design also. So I will say that besides the instrumentation, which is exactly flow cytometry type instrumentation, we found out it's very important to provide people with a sample preparation equipment, which is listed on our website. It's available for people to use at Rockefeller to use for free. And we take care of all the maintenance on this equipment. We have a swing rotor centrifuge. It could sound to be funny, but it's not funny because many molecular biology labs are not knowing about the difference between the way cells could be centrifuged and the difference which we see in the sample prep is tremendously high in terms of recovery and survival of the cells. We have the GentleMax, which is not only used with the kits provided by Miltenyi, but also as a uniform way of preparation of the single cell suspension. We have the cytospin centrifuge, which helps people to look downstream and do the assessment of the sample prep in the single cell suspension. We do have Luna cell counter, which we found to be absolutely essential for the very challenging samples like very small nuclei, for example, from ant antenna, which you cannot see on the other cell counters. We do have the laminar wash system from Curiox, which is not in heavy use because we don't have uniform type of sample runs, but it could be used by our researchers for example, for titration of antibodies. We use the Toledo liquidator to transfer samples from one plate type to another one. So we try to be helpful for our users to be able to assess the sample prep before and after the cell sort. We have two types of fluorescent microscopes, non-fluorescent and fluorescent microscopes at our place, and we did put the Revolve hybrid microscope next to SA to be able to assess the performance of the instrument in sample preparation part. We also have, oh, sorry, I jumped. We also have the orbital shaker, which I think changed a lot the life of our users who use the LegendPlex bead array assessment using the flow cytometry. So it is very important. We also suggest people to use very funny tools, but these tools help people to get downstream applications streamlined. We suggest them to use different tubes, different aspirators. For us, it's very important because cells which our users are running are extremely sticky, and using polypropylene tubes is one of the essential steps in there. When we suggest people to do titration of either dead cell exclusion dye or the antibodies, we usually suggest to do 7-step dilutions, as I told you, and for the dead cell exclusion, we suggest to partially kill cells, mix them together, and after that, use this guy for the dead cell exclusion titration. So we give the handout to our users how we suggest to better proceed on all of these things to make it not as laborious and as scary as they thought it could have been, to make the process, which is essential to approach, to make it as streamlined as possible. So this way, when they do the titration of the threefold dilutions of either antibodies or dead cell exclusion, make sure that you have the negative control and transfer everything to the cells for the downstream processing, either not wash if it's dead cell exclusion dye, or yes washes according to the protocol we present. And in general, it could be used not only in the Eppendorf format, but also in the bullet tube format. Dilution factor is huge with this titration, and we usually cover the whole range of whatever is necessary to find the best possible concentration of the reagent to be used in the field. So we use this bullet tubes aspirator and multi-channel aspirator as a way to explain to our users that many things could be done. They can borrow these things from us because our main goal is to have good experimental sample prep, and the sample prep is the key for the good flow cytometry experiment. I'm done. I want to thank the organizers of this conference. I hope that for everybody who is participating, it is possible to find very key points to see the best out of what can be done in the field and to find out how we can proceed better with the flow cytometry. Thank you.
Thank you, Dr. Massel. We have time for one question, if anybody has one.
Thank you, Silvana. This is Alexis Kalergis. Quick question about the that you included a computer person that was like a new addition to your team. Could you just explain a bit more how important that person has been and what kind of work he does?
Okay, thank you for asking. Actually, this is not the person who is doing data analysis. It's the person who is making sure that all our setup at the Rockefeller works because, as you know, we have some instruments, for example, with FACS Diva, which means Diva 8 is Windows 7. Windows 7 is not allowed to be used in the open world. So we have all our instruments on the special instrumental network and making logins through the Rockefeller web access, and also to have possibility to move the data within the environment, which is safe and secure, is extremely important. We found out that the IT person whom I did mention, and I really think it's one of the keys of success of the streamline approach in here, is very essential because with Diva, we have learned a lot, but at the same moment with FX Chorus, which appeared to be new software for us, which is not new in the field, but SA data present very complex I will say they present very complex organization of the data management, which we found to be quite difficult to approach with the conventional methods. And that is why we developed the workflow, how to move the data for the users to be analyzed. Chorus cannot be installed on currently on the standalone computer for the data analysis, which our users got used to do the data analysis. Software of the FlowJo doesn't show things in exactly way as Chorus does, and relying on the images, it's important to have these things available for the users to use the Chorus. So with the help of the IT person, we're able to make all the logistics to make the best out of available tools ready for the users. And at the same moment, we are waiting for, as Cheryl was talking about, the FlowJo 11, which is supposed to come on board by the end of May, as we know, and the new version of the Chorus to be more user-friendly. But in the meanwhile, to do research and look at the best possible outcome of the amazing instrument as S8 is, we need to have a lot of support to make things work. We have the 10 gigabit per second network installed. We have the NAS backup for the data because the data are huge, and to make sure the move of the data is streamlined, it's important to have good support from the IT. Not only for data analysis, but data analysis we're approaching together with our users, and so far we are quite successful in there.
Thank you very much, Dr. Massel.
Thank you.
To continue this session, we welcome Dr. Alba Grifoni, Research Assistant Professor of the La Jolla Institute for Immunology in the United States of America.
Hello, everyone. I would like to take the opportunity to thank for the kind invite and also to thank Cheryl that saved me some time on basic of the fantastic flow cytometry that we're doing here at LGI. Is everybody able to see my screen? Just double checking before I start talking. I assume you are?
Yes, we are.
Okay, sorry about that. No, that's totally fine. Okay, and so and my talk is gonna dive much more into the science. So what are the scientific questions that you're gonna ask and how you can use flow cytometry to answer them? First of all, in general, I would like to emphasize when we think about immune response against infection or what type of immune response is really important to induce in a vaccine to protect against a specific infection, what we need to highlight is what do we mean with protection? Because of course, when we think about protection from detectable infection, then a major role is played by the antibody response and particularly neutralizing antibody response that blocks the virus to infect the cells. However, and that is the mechanism with which many of the vaccines we're receiving actually work, protection can also mean protection from disease severity and protection from hospitalization and deaths. And so in a way, the majority of the SARS-CoV-2 vaccine were designed with this goal in mind. And in that case, there are multiple components of the immune system that can play a role in actually minimizing disease severity. And my lab has been particularly interested in understanding the role of T cells. And so in general, there are several lines of evidence that are showing the importance of T cells in SARS-CoV-2 immunity. And pretty much there are three major concepts that are summarized in three representative studies. But as you know, SARS-CoV-2 has a lot of literature to dive into, and in particular, many of the work done by people in that room. And so one component that is very important for T cells is quantity and coordination. The stronger the T cell response is, the better, but it also has to coordinate with different components of the immune system. Another component that is very important in terms of T cell response is kinetics. The faster the response, the better. Finally, the last piece of the puzzle that has been a little bit less studied and spearheaded by our lab is really the breadth. So the more antigen the T cell response are able to recognize, the lower is the disease severity. And so that is a concept that I'm gonna bring up a little bit when I'm gonna talk today, because when we think about the immune response against SARS-CoV-2, it is very important for T cells to think outside spike. And indeed, we know that immune system is teamwork, and just as a reminder, the reason why everybody is very focalized in the spike response is that that is very important in the context of antibody response. But once again, T cells can virtually recognize all the protein of the virus as long as they're chopped and presented in the context of the HLA molecules. And although each one of us has the favorite immune subpopulation that is studied, in reality it's really teamwork, and neutralizing, non-neutralizing antibodies, CD4 T cells and CD8 T cells, they really work in concert to really try to fight the infection. So a lot of work has been done in the past years to really dive into the role of the immune response against SARS-CoV-2. One feature that I didn't mention before that T cells have particularly as a feature with respect to antibody response is stability on viral evolution. And in fact, we and others have shown in the past that T cells that are induced by either vaccination or infection are able to recognize variant of concern. And so in a way, T cells are always able to catch up with viral evolution and provide a background immune response that is able to slow down the virus and prevent disease severity. One concept, though, that at the beginning of our study was really unclear is what is the effect of an infection that happens after vaccination on T cell response. And at the time when we initiated this study, the study in the literature were really providing contrasting opinion about it, from the group of Wendy Burgers that actually suggest that getting subsequent infection boosts the immune response, to the work of Paul Thomas that showed an increase, but a milder one than the one shown by the Wendy group study, to the group of Ruth Montgomery and Danny Altman in the UK, actually suggesting that, if anything, breakthrough infection were causing exhaustion of the immune system. So in this picture where it was really unclear the effect of BTI, the role of the study that we designed was really to understand more in detail several questions. And the first one was, what are really the immune perturbations that the T-cell response experience following breakthrough infection? And so in order to answer that question, we leveraged a longitudinal cohort. This was a vaccine cohort in which some of the participants actually experienced a symptomatic infection. So we have the unique opportunity to have pre-breakthrough infection samples and post to really pinpoint at the single individual level what's happening to the immune system following infection. The second question that we were having is, given the SARS-CoV-2 evolution, do we start observing differences that are SARS-CoV-2 variant specific? And that is very important because it really suggests the importance of keep monitoring the effect of viral evolution on T-cell response, if it's necessary. And the final one is really understanding if you get a vaccine, and I would like to remind that in the United States, the majority of the vaccine that we received were spike-based, we were really interested to see if an infection occurring following vaccination was able to broaden the antigen repertoire and the epitopes recognized following infection. And so normally I will dive into the data, but given this is a flow cytometry conference, I'm just taking an extra slide to dive into the approach. And as Cheryl has shown you before, our lab and in general at LGI, we have been heavily used what we call activation-induced marker T-cell assay, which is based on the combination of two activation markers that can be identified following antigen stimulation. What we have been doing in this study and in our subsequent study is try to really combine this original assay with the more classical intracellular staining assay that has been used even before the pandemic. And the way we have doing that was also to try to time the kinetics of T-cell response so you can have an efficient identification of cytokine produced by CD4. They usually tend to have a lower kinetics and CD8 that have a faster kinetics. So this is the assay we came up with. We co-culture our PBMC with our peptide pools. For people that have going collaboration with us, they know that we like them to call mega pools, but pretty much it's the same as a combination of peptides that try to reduce the complexity of the virus in a single pool so that you can have extensive understanding of the T-cell response, taking into account the different protein of the virus. And that's what we do. We co-culture these for 20 hours. So we give the time for the T cells to express on their membrane this activation marker. And then the last 4 hours, we add preferring enhancing to have the cytokine accumulation. One critical point that emerged while we were trying to combine this assay is the fact that the addition of preferring enhancing naturally can cause downregulation of the activation marker. And so the way we overcome that was to add concomitantly with preferring enhancing this antibody to preserve the expression on the surface of the cells. And so here the results show you essentially why we're doing this. If we want to look at activation marker in our AIM protocol, you see an expression. If you do a simple ICS protocol where you are preferring enhancing but not this component, you do have a decrease in this activation marker, which is significant. It goes back in line with what I was saying, that preferring enhancing can cause reinternalization of this marker. The moment in which we add this antibody in these steps, we were able to reestablish the natural expression of our AIM markers we would expect on a simple AIM protocol. And that is true whether you look at CD4 and CD8, and we originally devised these in a previous study and much more recently brought it to the next level. My lab published it this year, where we also show that you are able to remove supernatants at these steps to have a more in-depth assessment of the cytokine prior to go to the next step of the staining. The other point, of course, and like naturally, we want to make sure that this was not affecting the cytokine detection, and it doesn't. So pretty much we were able to add this extra step, improve our AIM detection and maintaining the cytokine production. I was mentioning the MAGA pool, and I'm not gonna go too much into details, but pretty much using a combination of bioinformatic and previous knowledge in the IDB database of the component of the virus we know are important for T cells, we're able to filter this complexity in smaller pools. And depending on the question, different strategies can be used to really pinpoint information about the virus or other infectious disease of interest. In this context, and particularly relevant for our study, our lab, and particularly the lab of Alessandro Sette and spearheaded by Dr. Antunes' work, were able to devise a T-cell assay that much more in-depth was able to discriminate if people that receive a previous vaccination also have a previous infection. And here I'm just showing you that our T-cell assay has pretty much 94% capability to detect infection as compared to the serology that was run at the time. And of course, the serology has been improved lately and the fraction of performance has increased, but yet still not reaching our T-cell assay threshold. So going back to the original question, what do we see when we want to go and look at the perturbation that a breakthrough infection is inducing on our T cells? And so to answer this question, we look at CD4 and CD8 T cells. We look more comprehensively, not just to spike, but also to the non-spike. And we apply this AIM-ICS assay so we could detect both activation of our T cells and cytokine production. First, we look at the pre-samples, and based on our findings and PCR testing that these people were receiving, we were expecting to not have previous infection. But that was not the case, and actually 30% of our cohort show a non-spike specific response, consistent with the hypothesis that these individuals experience an asymptomatic infection without knowing. And indeed, when we stratify our donor based on that information, we do see an increased response in the spike, CD4 T cell response, and also neutralizing antibody. So consistent with this, we look at the data, combine the entire cohort, but also we wanted to see whether the previous asymptomatic infection already was having an effect in the way you respond to post-symptomatic breakthrough infection. And so here I'm going to show you the response for spike and non-spike. In the baseline are individuals that we hypothesized experience a previous asymptomatic infection. And indeed, when we look at our CD4 response, we do not see a boost in the T-cell response following infection. Conversely, individuals that didn't have a baseline response, they do see a significant boost in the T-cell response following breakthrough infection. And once again, that is still the case when you combine the entire response. And once again, that's because the individuals that are at the baseline have symptomatic infection were just a smaller fraction of the entire cohort. For instance, if we were to repeat these experiments now, then the majority of the individuals already have a previous asymptomatic infection, probably we will not see necessarily an increased boost in T-cell responses. The other point that we really want to address is what do we see in terms of the quality of the response. So one of the concerns was that you do not see a boost, and instead you change the quality of the response and you see exhaustion of the antigen-specific cell. So we went back and looked at this data, and we start looking also at the co-expression of markers that have been in the past associated with exhaustion or downregulation of the immune response. And our data are consistent with the fact that there is actually no change in this parameter, and if anything, in some cases, a decrease of markers associated with regulation of the immune response. Therefore, our conclusion is following breakthrough infection, we do not see an exhaustion phenotype as previously hypothesized. We then were interested in really understanding what about the quality of the immune response. And so this is the combination of the different cytokines we use in the assay, and consistently with what we saw in the context of the AIM assay, we see a generalized increase in the cytokine production. Then we dive more into that, and we pinpoint which cytokine were the major driven of this increase, and those were to ascribe to interferon gamma and IL-2. And in particular, when we focus into polyfunctionality, we saw an increase in polyfunctionality following breakthrough infection. So with answering the first step of our study, now we were interested in understanding is the finding that we're observing different whether we look at people that got breakthrough infection during different wave time of SARS-CoV-2 variants. And so to do that, we collect a cross-sectional cohort that was experiencing thematic infection in different documented waves of SARS-CoV-2 variants, and particularly Delta and the two sublineages for Omicron. And so in this case, we wanted to have a more generalized understanding of the immune responses. So together with the T cells, we also partner in collaboration with the Crowdy Lab to look at the B cell component. And so whether you look at CD4, CD8, or B cell response, pretty much the uniform messages, we do not observe any difference whether you go from Delta to Omicron, which are pretty different between each other. And I don't have time to show, but like that was even true when we look at different sublineage of Omicron. And so with that one, we went to the third question, which was if you experience an infection following vaccination, are you able to broaden the response of the T cells versus different antigen pertaining SARS-CoV-2? And so what you're seeing here is essentially a large-scale analysis where we look in each pool at the 25 different proteins of SARS-CoV-2. Clearly, we have a majority of the response with spike, which makes sense given that you receive 2 or 3 doses of spike vaccine and on top the infection. But we also start noticing additional protein-specific responses consistent with the fact that the infection has generated novel response against antigen outside the spike. Then we wanted to dive more from the protein level to really the single epitope that is recognized by the T cell response. And so in doing this type of experiments, we were following the hypotheses that I'm going to highlight here before. So what happened when T cell wants to recognize a specific epitope? If you have SARS-CoV-2 ancestral, a specific peptide is going to bound to the HLA molecule and it's going to be recognized by the T cells in a nutshell, making it an epitope. When you have a mutation that is caused during viral evolution, there are 2 possible case scenarios. One case is the mutation disrupt the capability of the HLA molecule to bind the epitope, but the other one is that the mutation still preserves that binding. And at that case, you can have an increase in recognition because the TCR can still recognize that combination and actually get boosted by the fact that it gets response to that. You can have a decrease of responses, meaning that the T cell response is unable to recognize the epitope any longer. And then there is an additional hypothesis that has been understudied in the past. What about novel TCR recognition? So if you lose a TCR, but at the same time you generate a new T cell response, then when you look at the response, you're not going to see a change in the response. So you're not going to see a booster, you're not going to see a decrease. And so that is an indication that a novel TCR recognition occurs. So this in general is the expectation when you think about viral evolution. Another point that has been even less studied is what happens if the original ancestral peptide was never able to bind to HLA molecules, so the T cell response were unable to see it, but now with the variant, the HLA molecules are able to bind and present it to the immune system. So in that case, you should be able to have a novel TCR recognition and generate a completely new response that we were not able to see before. So when we did our epitope identification, we were trying to see which one of these mechanisms was present following a breakthrough infection. So this is where things becomes interesting because what we notice is that we have a little bit of everything. So I'm going to focus much more on the Omicron, given that he has much more mutations, so it's easier to make the point. But in looking at the responses, we see some indication of increase of response that suggests that the epitope recognition is preserved, but there is a boost in the response, and therefore you see an increase in magnitude. Then you have some cases where there is no change. That suggests that the original T cell was not able to recognize this anymore, but a new T cell was generated and pretty much you see no boost in the immune response. Then there is the third case of decrease. You used to have a good response, the mutation has disrupted, and so you have lost T cell response out of it. And then finally, and that was very exciting for us to notice, we were able to see variant-specific response mounted following infection. If you try to combine all of this, that's what you observe. So as a reminder, when we look at the entire response, the average response that we observe is roughly 0.8% of CD4 and CD8 T cell response, even when you focus on the worst case scenario of the point where there are mutations, there is just a smaller fraction of the response, so you still have this component that is 100% conserved, and on top of that, out of this smaller fraction that has mutation, in most of the cases, the response is not affected, and that's because it is composed by different mechanisms: decrease, no change, and increase of de novo response. And so that is true whether you look at Delta, and it's even easier to see, but it's exactly the same principle also in the context of Omicron. And so to conclude, we found in our study that breakthrough infection are able to boost the T cell response by 3 different mechanisms. The first is the boost in magnitude, and that is particularly evident if no baseline is observed, while instead, if you have a previous recent asymptomatic infection, probably the T cells response is in plateau, so you're not going to be able to see a boost of the response. There is a boost in breadth. When we dive into the different antigens recognized by the T cell response, we start noticing non-spike specific T cell response, although the individual have been vaccinated with a spike-only vaccine. Also, we see the generation of new variant-specific epitopes, suggesting that T cells are able to catch up with viral evolution and constantly generated de novo response variant-specific. And then finally, this is consistent regardless the particular SARS-CoV-2 variant analyze. And so our study has been also corroborated in the context of other variants, and in particularly this follow-up study is only bioinformatic, and long story short, we try to compare the difference in evolution between ancestral and a more recent variant, the pyrola, maybe some of you may recall. And what we see is that if you compare the original one to the most recent one versus a more recent sublineage, the de novo response induced by the exposure with the recent sublineage kind of bridge the gap for the subsequent one. And so the T cell response is more and more stable across variant evolution. And this was a bioinformatics study and has been corroborated experimentally by 2 individual collaborators in different geographical locations. So with that one, I would like to conclude that immunology is teamwork, and the majority of the work that I show you has been spearheaded by 3 members of my group and by a collaboration with the lab of Alessandro Sette and Shane Crowley. And with that one, I'm happy to take any questions.
Thank you very much, Dr. Grifoni.
Thank you very much, Alba. Here's Pablo Gonzalez.
Hi, Pablo.
So, great talk, amazing amount of data, very valuable results and questions. So congratulations for all the work. So I have a question regarding maybe what you did not show, and hopefully you may have analyzed this in other studies, but I'm very intrigued on knowing what happens with Tregs. Are they expanded upon all these boost, breakthrough cases? Because we usually tend to, we're looking all the time for AIM positive, for exhaustion, but we haven't looked that much into Tregs. So I wonder whether you have done some of that work.
Yeah, that's a fantastic question. So the AIM marker that we're using, particularly for CD4 T cell, OX40 and CD137, do include Tregs. And if you wish not to have them in your assay, you actually need to use a different combination. Often it is recommended to use OX40 CD40 ligand. I'm saying that because follow-up studies that are not available yet and done mostly by Alex lab shows that the single sequence RNA-seq level also a signature of regulation. And I'm not gonna dive much more into the detail because it's their study, but I'm happy to share with you the findings once the study is out. It's actually under review, so it should be out soon. But long story short, yes, there is a regulatory specific antigen response, which may have been affected by different booster or infections. So that's definitely something that we observe in our data. To parallel with data already published, you might be familiar with the work of Dana Farber that actually she looked into that in her cadavers donation, and she does see a population of immunoregulatory antigen-specific cells that increase following booster vaccination. So I hope that answered the question.
Yes, yes, it does. But overall, there's much less studies on Tregs than on AIM, certainly. I mean, the focus is more on the activation than the regulation. And so I have another question, if I may. If we compare the immune response to COVID to influenza, do you see parallels in terms of this repeated exposure year after year? And do you expect a difference also throughout the years if you continue with these analysis in terms of getting to a similar type of response as influenza in terms of this ceiling on T cell responses to repeated antigen exposure? Because here we have the great chance that we know the infection date on adults for the first time, which we do not have in influenza. Influenza we get as a child, and responses in child are very different from adults. But here we have a disease where we can actually measure the start point of infection from adulthood and then follow up throughout the years and see what happens with these types of markers in terms of exhaustion and T cell activation ceiling.
Yes, so let's unpack. This is a beautiful question. I love it. First, what do we parallel this with influenza? Nobody has done that. I am actually trying to do that, and it's very hard to convince the field that T cells matter in influenza. If you ever manage to do that, let me know, because I've been trying. I'm telling you, I've been trying. In terms of your question about following multiple exposure, we have done follow-up on multiple infection exposure. So far, our data holds, we see no exhaustion. Now, in the context of influenza, and this is just speculation, I would love to generate the data, we need to remind ourselves that influenza vaccine are based on the same PR8 backbone that is given over and over and over to T cells. And so the question is, and this PR8 is very, very different in reality from a viral evolution point of view to the circulating influenza strain that we receive like as human, infecting each other with a different strain. So one question could be, maybe the period specific T cell response after getting vaccination on a yearly basis over and over can get to exhaustion, yet the exposure to the viral strain that are different from the backbone may still generate new activated T cells that are able to control the infection. In truth, nobody has done this type of study, so there are a lot of speculation. What I would like to say is that, unfortunately, it's hard to make parallelism with SARS-CoV-2 and influenza because nobody has done a deep dive in influenza as much as it's done with SARS-CoV-2. It's possible that the two infections are going to behave the same. It's also possible they don't. Also, in the context of corona, let's remind ourselves that you also concomitantly get exposure to other common cold coronaviruses, and so the diversity that you get exposed to might even be broader than what you get with influenza in general. So I would be cautious in trying to parallel the two too much. That's at least my personal view about it.
Very true. Thank you very much.
Hi, Alba. This is Susan. How are you?
Very good.
Very nice
How are
talk.
you? Thank you.
Thanks. Seeing your data, I was wondering about the possibility that these variants that generate new peptides that binds to the MIC could indeed cause changes in the affinity with the with the peptide MIC TCR with the new variants that could actually inactivate T cells in some way that this, if you have seen something equivalent, if you vaccinate with the one strain, for instance, for one variant, and then you get infected with another variant, and the peptides that are presented to them in the MIC can change and change at the same time the affinity with the TCR. Do you think it's possible that you can get inactivation in some way, like a immune escape for this variant as well?
So the data that I show you clearly cannot speak about affinity, right? For that you need TCR sequencing. However, when we do the epitope repertoire, we also compare the average epitope repertoire of the individual following breakthrough infection as compared to the original data that we did at the beginning of the pandemic with a single infection or vaccination. We don't see a difference in the breadth of the immune response. Also, in terms of immunodominance, we do not see the novel T-cell response having an increased magnitude over the conserved ones that might justify, a disbalance of the T-cell response, as you suggest. So as of now, based on the data we have, I do not see this hypothesis to actually be possible. However, once again, we didn't dive into the TCR sequence, and really to pinpoint the analysis you're asking, you need to follow after multiple infection, what is the avidity and expansion of the clone based on TCR sequence analysis. However, there is a group in the United States that is particularly famous to do that. I mentioned at the beginning of my talk, that is the Paul Thomas group. And so what they see in the Minervina paper, they already are looking at people that got infection before, vaccination after, and breakthrough infection after. And they do see a modulation of the clones, but they do not see the novel responses necessarily taking over or reducing the clonal expansion of the original clones. So although their data clearly have not looked at the multiple exposure and the situation that we have right now, not even their data suggests that that's the direction the T-cell response is going to. But once again, great question, and we need more data to answer this question maybe in 5 years.
Thank you. Thank you very much, Dr. Grifoni.
Sure, my pleasure. Thank you, everybody. It was lovely to be here. Bye.
Okay, so to close this session, we invite Dr. Ved Prakash from the International Center for Genetic Engineering and Biotechnology from New Delhi, India.
Hello. Hello. So first of all, I would like to thank Professor Alexis for giving me this opportunity to present my work. My screen is visible.
No.
Okay.
Doctor, now we don't see you nor your screen, actually.
So yeah, I'm trying to share my screen. Yeah.
Yeah, I think you have permission, but to share your screen. This is Alexis. How are you? So try to go to down on the screen and then try to share your PowerPoint. Here you go.
Now we see your presentation, but we don't hear you.
So visible now?
No, we saw it and now we don't see it again. Now we see it.
Okay.
Yes,
Now,
perfect.
yeah. So, hi, very good evening and good morning to me, because it's really peak morning for me here in India. And I would like to thank Professor Alexis for giving me this opportunity to discuss my work in this symposium. So, I'm Ved Prakash Dwivedi. I'm leading immunobiology group here at IC. It's International Center for Genetic Engineering Biotechnology. Am I audible? Hello, I'm audible?
Yes, we can hear you loud and clear.
Yeah. Just. So yeah. So it's the International Center for Genetic Engineering and Biotechnology, New Delhi. I'm also taking care of flow cytometry facility at ICGB New Delhi. So, as we all know, understanding the host predictive immune response against TB is really very complex. It is still unclear which antigen or which immune cells provide predictive immunity against this deadly disease. So today and every day we are trying to unscramble the complexity of this disease and associated immune responses. So today I'll be discussing about the um, uh, role of host predictive immune response against TB. So first of all, I would like to give a glimpse of what ICGB is and what ICGB does. So basically, ICGB is International Center for Genetic Engineering Biotechnology, and our vision and our mission is science for development, and our vision is to be the world's leading intergovernmental organization for research, training, and technology transfer in the field of life sciences and biotechnology. And again, our mission is to combine scientific research with capacity enhancement, thereby promoting sustainable global development. It has three main components. One is in Trieste, Italy, and other one is in Cape Town, South Africa, and uh, our is in New Delhi, India. And other one is one regional research center in China. Although there are 68 member states which are recognized by ICGB, and uh, we are doing here translational research, and we are developing poorly developed and underdeveloped countries to facilitate their research for so that they can be able to do translational research for human benefit. So as we all know, as per WHO, 1/4 of the global population is latently infected with Mycobacterium tuberculosis, that is causative agent of tuberculosis, and is responsible for millions of deaths each year. It has spread worldwide, but more prevalent in Southern and Central Africa, South Asia, and Latin America. So Mycobacterium tuberculosis mainly infects through respiratory tract, where the alveolar macrophages engulf airborne bacilli. Bacteria replicates inside the macrophages. Macrophages secrete some cytokine and chemokine in response to bacteria to activate the lymphocytes. Once they get activated, they migrate to the site of infection and form granuloma. The main function of the granuloma is to prevent the spreading of bacilli from lung to other organ, as well as concentrate the immune response at the site of infection. If the immune response is stronger, then bacteria will be retained, granuloma will be reduced, and characteristic will be asymptomatic. However, if there is weak immunity, then bacteria will replicate, granuloma will caseate, rupture, and spill the bacilli into the airway to infect others through coughing and sneezing. So, although TB is present on the earth since ages, but it is still considered as global pandemic. The problem lies both in its prevention, wherein we have only one vaccine, that is BCG, which is very effective in case of disseminated and meningeal TB in young children. However, has questionable efficacy against adult pulmonary TB. And if we talk about its treatment, which consists of multiple expensive antibiotics with no emphasis whatsoever on the host. Although there are multiple T cell subsets which are known to play a crucial role in containing MTB infection, but still the exact correlates of post-productive immune response against TB is not known. So today I'll be discussing about some role of some novel T cell subsets during TB pathogenesis. And here in ICGB New Delhi, my lab is basically involved in the formulation of various vaccine and immunotherapeutic candidates by exploiting various host factor, bacterial factor, and epigenetic factors. So to develop a vaccine candidate, first we need to understand why BCG, the existing vaccine BCG, doesn't work. So for this, we performed a very simple experiment where we compared the immune response between virulent strain of Mycobacterium tuberculosis S37RV and vaccine strain BCG. And interestingly, what we observed that at different and then we infected these mouse with S37RV and BCG, and then at different time point we took out the organ to look at the bacterial burden and immune responses. And interestingly, what we observed that initially both strains replicated in similar manner, but at later time point growth of BCG got gradually diminished. So it might be the reason that adaptive immune response plays a critical role in the clearance of these BCG bacteria. And then we looked at the protective cytokine responses, and we observed that both strains induces comparable level of IFN gamma producing Th1 cells. However, BCG was unable to induce significant population of IL-17 producing Th17 cells. So what is the reason behind this difference? So as we know that as compared to virulent strain S37RV, vaccine strain BCG possesses multiple deletion mutations, and these mutations are known as RDs, region of differences. And among these RDs, RD1 is the most dominant, prominent, and important one for providing virulency to Mycobacterium tuberculosis. So we thought, is RD1 responsible for the differences of immunogenicity in S37RV and BCG? For this, we deleted RD1 from S37RV and put back RD1 into BCG, and then we again we performed the same experiment as I discussed earlier. And what we observed that RD1 deleted S37RV was behaving like BCG. However, RD1 knock-in BCG was behaving like S37RV in terms of bacterial burden and immune responses. Therefore, we concluded that yes, RD1 causes differences in immunogenicity of S37RV and BCG, and so as also is responsible for the induction of IL-17 producing Th17 cells. And with this experiment, we concluded that even S37RV could be a better vaccine candidate than BCG itself. But for obvious reason, we cannot use S37RV as a vaccine candidate. Therefore, we thought to design a MTB mimic molecule. The idea was to generate the immune response without causing any pathogenicity. So for this, we screened various MTB peptide for their capacity to activate T cell as well as protective cytokine responses, and we selected few of them, combined together with TLR ligand, and absorbed these assembly onto the liposome to facilitate their entry into the lung of mice through intranasal injection. And we named this molecule as PTL, and we patented this molecule, and right now this molecule is in national phase of various Southeast Asian and African countries. Then we asked whether this PTL molecule can enhance the vaccine efficacy of BCG. And for this, we performed a vaccine experiment wherein BCG immunized animals were subsequently boosted with this PTL molecule. And after a very short period of rest, these mice were infected with MTB through low-dose aerosol infection model. And then at different time point, we took out the organ and looked at the bacterial burden and immune responses. And interestingly, what we observed that PTL co-immunization significantly enhanced the BCG-induced antitubercular immune responses, as evidenced by reduction in bacterial burden by more than 2 log in lung, liver, and spleen of the mice. We also observed that PTL co-immunization significantly enhances the polyfunctional cytokine responses and memory responses, which are required for the long-term protection against tuberculosis, which further led to the significant reduction in the rate of TB reactivation. which is major challenge being faced due to current anti-tuberculosis therapy. And interestingly, we observed a significant increase in the double positive T cell, that is T cell having both the arms, CD4 and CD8 positive T cells. And you can appreciate that as the percentage of these double positive T cells are getting increased, the bacterial burden is getting down. So now we know that these double positive T cells are produced in the thymus and are involved in the generation of single positive T cells, which eventually migrate to the periphery and provide protection against different disease conditions. However, the presence of these double positive T cell in the periphery has also been reported in a number of viral infection, cancer, and autoimmune diseases by a number of techniques like flow cytometry and confocal microscopy. But the functionality and significance of these double positive T cells has never been studied during TB pathogenesis. So here in this study, we would like to emphasize the role of these double positive T cell during TB pathogenesis. So initially we thought whether these double positive T cells are artifact or real population. So for this, we performed staining wherein these double positive T cells were stained with antibodies having tagged with different fluorochromes, and we concluded that no, these double positive T cells are not artifact but real, which are getting infiltrated into the lung of MTB infected animals. And then this study was further validated by confocal microscopy, where you can clearly see these double positive T cells. Then we further analyzed and we observed that these double positive T cells produces increased polyfunctional cytokine responses, that is IFN gamma, TNF alpha, IL-17 and IL-2, and also increased central memory T cell responses, which are required for the long-term protection against tuberculosis. Furthermore, upon adoptive transfer, these double positive T cell displayed increased anti-tubercular immune responses, as evidenced by reduction in bacterial burden than their single positive T cell counterparts. So taken together, these data suggest that these double positive T cell might provide protection against tuberculosis. So now we know that these double positive T cells are produced in the thymus and are involved in the generation of single positive T cells. So therefore, we wanted to see whether these double positive T cells that is produced in the thymus and that we are getting in the lung are different or same. So for this, we did RT-PCR for the some transcription factor that is specific for CD4 and CD8 positive T cells, and we observed that in the thymus, this double positive T cell expresses less RNX3 and TSPOK transcription factor that is specific for CD8 and CD4 positive T cells than their single positive T cell counterparts. However, in the lung, this double positive T cell expresses higher CD8 and CD4 specific transcription factor, again confirming that these peripheral double positive T cells are enough mature and carry the transcription feature of both CD4 and CD8 positive T cells. Then we know that these double positive T cells are not immature thymocytes. Therefore, we wanted to see what is the kinetics of these double positive T cell during in different organs of different organ of mice during MTB infection. And interestingly, you can see you can see that these double positive T cells are sustained even after 120 days of MTB infection, be in lung, spleen, mesenteric lymph node, and in thymus of the MTB infected mice. And then we asked whether these induced double positive T cells are MTB specific. For this, we performed a tetramer staining, and where we observed that around 6 to 7% of these double positive T cells were ESAT6 specific, which is as compared to their CD4 positive T cell counterparts. Again, confirming that these double positive T cells are protective in nature and they are MTB specific. Then we looked at the functional characteristic of these double positive T cell, and we observed that this double positive T cell has higher Th1 phenotype and lower Th2 phenotype, and there were no differences in the Th17 phenotype as compared to their CD4 positive T cell counterparts. Taken together, these data suggest that this double positive T cell might provide protection during tuberculosis, because Th1 cells and Th17 cells are important for the protection against tuberculosis. Then we looked at the cytokine level, and we observed that these double positive T cells induces almost all the pro-inflammatory cytokine that is required for the protection against tuberculosis than their CD4 and CD8 positive T cell counterparts. So now we have proved that yes, this double positive T cell has memory phenotype and also produces polyfunctional pro-inflammatory cytokine responses. Therefore, we wanted to see what is the nature of these double positive T cells during TB reinfection. And you will appreciate that for this we performed a reinfection experiment, and you appreciate that these double positive T cells were enriched in the lung of MTB reinfected animals as compared to primary infection. And again, confirming that these double positive T cell may provide protection during TB recurrence. So in order to look at the detailed nature of these double positive T cells, we perform proteomics, and proteomics of single positive and double positive T cells that was sorted from the lung of MTB infected animals, and we observed that around thousands of proteins were differentially regulated in these double positive T cells, and as compared to their single positive T cell counterparts, and around 93 proteins were differentially expressed in all 3 T cell subsets, again confirming that these double positive T cells have different proteomic landscape than their single positive T cell counterparts. So our detailed analysis revealed that there were significant changes in the metabolic, cytokine, and signaling pathways, and interestingly, hedgehog signaling pathway were down-regulated in these double positive T cells as compared to their CD8 positive T cell counterparts. As we know that this hedgehog pathway plays a crucial role during T cell development, and this hedgehog pathway has 2 states, off state and on state, and during off state, some proteins interact in such a way that they prevent the translocation of GLI into the nucleus and thereby inhibits the hedgehog signaling. And during on state, these proteins interact in such a way that they allow the translocation of GLI into the nucleus and thereby activates the hedgehog signaling. So then we looked at the positive and negative regulator of hedgehog signaling, and interestingly, we observed that positive regulators were significantly down-regulated. However, negative regulators were significantly upregulated in the lung of MTB-infected animals. That clearly showed the hedgehog signaling is inhibited for the enrichment of these double positive T cells. So on this note, I would like to summarize this finding, where we concluded that these peripheral induced double positive T cells has memory phenotype and also induces pro-inflammatory or polyfunctional pro-inflammatory cytokine that is actually required for the long-term protection against tuberculosis. So single cell transcriptomics is underway to understand the different signature of these T cell subsets, and studies are being carried out to characterize these double positive T cell in macaque and active TB patients. And now we hypothesized that having both the arms, we would like to see what is the role of these double positive T cell during HIV-TB co-infection. Therefore, we are exploring this in macaque model infected with TB and SIV. So on this note, I would like to acknowledge ICGB member, of course, my lab member, collaborator, ICGB core facility and funding partner. Thank you.
Thank you, Dr. Prakash. Do we have any questions?
I'm sorry because it's in India, it's early morning, so.
No problem. Thank you very much for your talk. And Pablo Gonzalez here. And my question is, so based on your conclusions, it seems to look like these double positives are mostly behaving like CD4s, but did you have a chance to determine whether they recognize antigens on MHC class 1 or MHC class 2 to see what is the preference of their TCR?
Yes. So basically, we did the tetramer staining for CD8 positive T cell as well, TB10.4, and we got that around 3 to 4% were TB10.4 specific, which is as compared to CD8, which was comparable to CD8 positive T cells during TB infection. So it was just single antigen. I mean, ESAT6 specific CD4 and TB10.4 specific CD8. So definitely, because tetramers are available for these antigen only. So Yes, we have other antigen like antigen 85B, but of course, we will be we will be doing staining with these antigen as well. But these are well established antigens, so we thought why not to look at the specificity for these antigen.
I see. Thank you very much.
We have another question.
Thank you very much for your talk. It was very interesting. I really enjoyed it. So I have one question about this double positive T cells. Is it specifically for TB, or is it like a general subset of T cells? And can you get them from vaccination, for example, with BCG? Do you vaccinate with BCG and you get this population on it, or not?
Very interesting question. So basically, as I discussed, that they are extensively characterized during viral infection and cancer and autoimmune diseases. But in case of TB, there are no reports. And when we made our PTL molecule, if you remember in my previous slide, so when we performed adjunct vaccination with this PTL, so enrichment of these double positive T cells were more during intranasal injection with PTL. So we thought, yes, but as compared to BCG, because in case of BCG, we did not get anything. And even in spleen, we did not get this double positive T cell population, only in lung when we were enriching with these PTL molecules. And when we, compared with the only primary infection, I mean, without any vaccination, we did not get significant difference. But when we, injected this PTL into the lung of infected animals, BCG immunized animals, the enrichment was more. And we compared with BCG, and now we are working on the enrichment of these double positive T cells. I mean, how we can enrich these double positive T cells during BCG vaccination, or modified if we are taking other modified vaccine, and if we can enrich this population, then definitely I think this will be great if to achieve the protection level than existing whatever we have existing one. Yeah.
Thank you very much, Dr. Prakash.
Thank you so much.
We now invite Dr. Alexis Kalergis to give his closing words.
Well, thank you very much, everybody, especially to the students that stayed till the end. We have a very busy and comprehensive day of flow cytometry and science with scientists from all over the world. And for our colleague here from India, for him it was like 4 a.m. in the morning. So thanks him to wake up early and then be with us. So thank you so much, and we hope we will do it again maybe in a couple of years with Professor Peñalosa. He was a great partner. Muchas gracias, jóvenes. Estuvo interesante, largo. Estamos todos cansados y especialmente a José y a Ricardo que nos ayudaron con los computadores, y también a las personas de la sala, y bueno, y a nuestra querida Antonia, que hizo un trabajo genial como conductora hasta el último minuto. Gracias.